lectures in symposia, conference, seminar, workshop, etc. Sir has also widely traveled abroad for research under many prestigious research programs and fellowships like DAAD, DST-DAAD, DST-GSPS, INSA-DFG, Germany, INSA Hungarian Academy of Sciences. He has also been visiting scientist professor at many institutions like Institute of Botany, University of Cologne, Max Planck Institute Cologne, Technical University Darmstadt, Germany, University of Central Florida, USA, Institute of Bioresources, Okayama, Japan, and Shanghai Polytechnic University, China. He undertook visits to Germany, USA, Hungary, Japan, China, UK, Italy, Spain, Belgium, and Portugal many a times. Professor Sharma has highly rich administrative experience of more than 25 years, as he's also the department chair and dean, Faculty of Science and Technology, Dean Academics, and Dean Research. Professor Sharma has also been the member, Rajasthan State Biotechnology Advisory Board, Government of Rajasthan, expert member, R&D Project Committee, DST Rajasthan, resource person, DST workshops, expert member, Bioinformatics Committee, State DST Jaipur. He has also been expert member of selection committees of many universities, selection committees of MHRD, Government of India, UPSC, RPSC, expert member, Board of Studies of more than 10 universities, Academic Council member, advisory, and other committees at several institutions like BPT Program Advisory Committee, BPT Nominee, Institutional Biosafety Committee, etc. Overall, Professor Sharma contributes the quality of a great researcher, teacher, administrator, communicator, motivator, and advisor. Sir is currently mentoring several young scientists for innovative research at Amity University, Rajasthan. With this brief introduction, I would now request Sir to kindly deliver his lecture. Welcome.
So, thanks a lot, Dr. Abhi, for this very generous introduction. All chairs of the conference, the dignitaries sitting off the dais, in the audience, both online and offline delegates of this conference, and our dearest friends from the Department of the MIT School of Bioeconomics. So I think a wonderful rainy good morning to all of you. It's a very heavy rains, in fact, which we are having. Now, in fact, on this platform, I shall share some of my thoughts and some of the work which we've been doing in the area of plant stress biology. I'll touch upon maybe just a couple of them, couple of approaches which we followed in past. And then let's see what happens. So, topic of the topic of my talk. All of you know, is it full screen? Okay, it's already full screen. So, understanding plant stress biology and what are the emerging approaches in this area. That's the topic of my talk. Now, let me at the outset be very clear. I mean, let me try to make you understand the problem, the main problem that we have in the area of plant stress biology. That we have a lot of different kinds of stresses, and the stresses are included in the category of biotic stresses, abiotic stresses, and as a result of that, of these different kinds of stresses, there are huge losses, huge economic losses, damages to the crops. So, for instance, we have biotic stress, which is essentially by infection of the phytopathogens, and they are by and large responsible for around 12% of the losses, the damages to the crop. Then we have another category of the abiotic stress, And one type or the other of the abiotic stress is also responsible for, let's say, crop damages to the extent of about 50%. So a lot, lot of this, in fact, this stress background, different kind of stresses was also discussed yesterday, the keynote address by my predecessor, Dr. Nilima. And you are, I think, those of you who attended that talk are well posted on different kind of stress that we have. So globally, in addition to that, over and above that, about 22% of the agricultural land is saline, and the drought stress, scarcity of water is contributing to another 26% of the crop losses. So it's a huge, huge loss. We are just left with around 10% of the area, which in fact is not covered by, which is free of any kind of stress. Now, we know the problem of food security. It's a major problem all over the world. It has been rather a major problem, not now. Why not now? Because of the prudent approaches which were taken by, in the, let's say, early, if I go back to early 60s, India was hugely dependent on even important, the staple food grain like wheat. And then there was development of, Mexican varieties, etc. They had come to India, and gradually, even in 70s, India was dependent on grains. It was importing grain. But now we have a situation where we are exporting grains. In fact, not only wheat, but even wheat we are exporting, and a few of them, few other cereals as well they are being exported by our country. So how the situation changed? Because the population, if you see, is rising. Population, for instance, in 2024, as we stand here today, is 8 billion. And then the estimates are that by 2037, it would be 9 billion, and 2060, it would be 10 billion. And obviously, as you know, most of the European countries, they are having negative growth rate. Even USA is slightly over 1 or 2%, something like that. So most of this development in terms of most of the rise in terms of population is coming by view of the countries like India. In fact, India is now a major contributor to the population growth. So how we are going to address this requirement, food requirement, the grain requirement, etc., of this rising population? So one approach, of course, which we know was to enhance the productivity per acre. We succeeded, we had, as I said, 60s, 70s, we have a different kind of situation, but we have a better yielding varieties. Another approach, and that is as well would be a very, very potent approach in order to address the problem of food security, is that you reduce the damages, thereby enhance the productivity, reduce the damages to different kinds of which are caused by different kind of biotic and abiotic stresses. Develop resilient crops, develop genetically engineered crops, find out what mechanisms are going on in those plants which are robust, which can grow under any kind of stress situation, because there are plants, which can grow very well under different stress situation, whereas those plants which, are not able to tolerate even, depending upon, for instance, you're aware of rice, is not able to tolerate unless you have plenty of water, and the plants would die. So, it all varies. So, I mean, one approach, therefore, would be to develop stress resilient crops. And how we do it, we have to obviously understand what kind of mechanisms are responsible that make the crop robust, that make the crop resilient. So that is the question we go. So naturally, therefore, if you talk of the plant biotechnology area, scientists world over globally, they are. It's not moving?
Yeah.
Oh, okay. It has not moved at all. तो पहले बताना चाहिए था. So this was the slide in which we discussed the data on the slide. Sharing. All right, so we are back into the talk, and so that's what I discussed the slide regarding that. So how do we do that? You know, the key question is that how do we address the problem of this stress? So therefore, you find that scientists world over working in the area of plant biotechnology or related areas, they are using all kinds of approaches to understand what goes on in the plants under stress situations. What they do is they bring the plants to laboratories, they provide stress, they grow the plant under stress, and then see what happens. That is one approach. Now we, in fact, here took a different kind of approach. We thought that let's go to the field and find out what are the mechanisms which are naturally robust. What are the mechanisms in the plant which are naturally growing under highly stressed environment of less of scarcity of water, scarcity of mineral, scarcity of light, etc., etc. So let's go to the field. So we, in fact, try to study the mechanisms in naturally growing plants, those which were naturally adapted to stress situation. So in this context, we made a study in the plant, which is a very resilient plant growing all over India, growing in all kinds of terrain, growing, in even in mountain, rocky areas. So in all, even in those areas which have very, very less mineral nutrient in them. So it's one of the plants where we undertook this study was Bhutia Hospital, which you all know by the name of Palas. In fact, it's also called as flake of the forest or tesu. In fact, you get the natural color from these plants during the festival of Holi. So we, different kind of collaborative products. We funded by TSC, funded by the German Academic Exchange Service. We studied those mechanisms. If we found the reasons underlying for this robust performance of Bhutia, that what is, let us say, in terms of photosynthetic efficiency, what kind of modifications it has done in terms of capturing light, in terms of utilizing it to the maximum, in terms of controlling the photoinhibition processes, in terms of controlling the loss by photoinhibition process, in terms of developing a high mineral ion acquisition efficiency, despite the fact that the soil is deficient in minerals. So other plants would die, but they are able to give double the capacity to greatly absorb the minerals from the soil. So multiplicity of factors. We studied enzymes also in that context, and we found that one of the enzymes, this is recombinant ATPase, which in fact is bound to the chloroplast, is one of the enzymes which in fact contribute a lot to this. We studied subunits and how the subunits change. So during this phase, what happens? So multiple factors, ecophytochemical factors, photosynthetic parameters, mineral ion parameters, related enzymes, all that we found out, which made the Bhutia robust to grow under highly adverse kind of situations. So that was, one story which I would not go into the details of this, which I would like to share with you one story. Then also, you see, these days you are able to also combine, and you have to combine, there is no option. If you are working in the area of biotechnology, you have to combine both these, the wet lab and the in silico approaches. So that's what we did. And what we did was the following. For instance, let me give you the background of this. If you talk of the plant genome sizes, let's say if we talk of the size of the algal genome, it is only Australococcus, single cell alga, it is only 10 MB. If you forget about it, if you say, no, I'll see what are the sizes in the angiospermic plants. So cardamine, for instance, an angiospermic plant, it has a genome size of 54 MB. That is one extreme in terms of size, very small size. Then the list goes on, and if we talk of the size of the diploid genomes, it is 124,000 fiddle area. Deployed. I am not saying, polyprides, because polyprides will have more number of this. But even if we talk of the deployeds, so from 54, we are 124. And if we talk of, for instance, the Paris, which is octoploid, and it has 143,000. But let's make a comparison between 54 and 124,000. Even then, you find that there is a huge variation in terms of the ratio of global volumes, and they are differing as many as 3,000 times. So angiosperm size difference is up to 3,000 times. If you talk of the gymnosperm, they are relatively consistent. There aren't that much variation. Their size around, let's say, for instance, 10,000 MB. But then, if the gene number differences, therefore, consequently, as well will be much more, from 20 to 200 folds, rather than going up to 3,000, as you see in the angiospermic plants. Now, see the structure. See what kind of growth they have. For instance, if we talk of 54 MB1 cardamine, and if you compare it with Fidelia, 124, or Paris Japonica, 149,000, you find that cardamine seem to be apparently more complex. It has, so many multiple type of flowers, leaves, mushroom leaves, etc. Having a very simple genome. So the question therefore is, and as the student of molecular biology, we are all aware this problem has been repeatedly addressed, called as the C-value paradox or C-value enigma, puzzle. What kind of dissimilarity this is. So what they concluded, and as you have yourself are realizing and seeing through the pictures, etc., that the size of genome, even if you talk of the consistently deployed organism, is not related with the complexity of the organism. They have no relationship. 54 MB is doing wonders, and 149,000 MB is looking to be apparently simple. So we all know some basic information in molecular biology that you have junk DNA, etc., and a lot of publications, a lot of work has been done about junk DNAs. And we study that. Even our postgraduate students will be studying about that. So genomes of related organisms often a strongly different size, and there could be multiple reasons for this. For instance, you have duplications of whole genomes, by polyploidization, hexaploids, octaploids, tetraploids, etc. Or replication of the invasive DNA, we have transposable elements, but reductions are also possible by recombination. For instance, we have diploid clones. But even, I repeat again, consistent diploid 30,000 times variation in size, but no difference in terms of, let's say, complexity-wise, there is no difference. Growth of the plant, there is no difference. So the question is, therefore, you have huge genomic data. Or we have, the genomic sequences for a large number of micro to higher organisms which have been completed, talk of E. coli, for instance, talk of Arabidopsis thaliana, where whole genome has been sequenced, Aurea sativa, Sorghum bicolor, we have many. In fact, if you, we have a wealth of information, and we have different online repositories of this genomic data. For instance, we have the Arabidopsis information resource, we have GTP plant genomic database, we have geodatabase. We shall talk about this geodatabase somewhat later in detail. NCBI, National Center for Biological Information, plant genome, Grameen, and many others, just to name a few. And if we go by the most comprehensive database, the big picture that stands today, or in their latest release that was on 28th August 2024, Genome's online database called as Gold version 9, it has 37,883 completed sequence genomes. Imagine it. So figure it not. Let's forget about the era of human genome project. I'll talk a couple of minutes about that also later. And in addition, 426,632 permanent drafts, they are documented. I think for the benefit of our undergraduate and postgraduate students, I will share that permanent drafts means that it has been done only once sequencing, whereas the other category is this finished draft, the finished sequences where you have to repeat it 3-4 times, something like that. So even permanent drafts we are running in hundreds of thousands, and even completed ones 37. That's a growth graph which has been shown at the bottom. How it has grown in terms, if you talk of the years, etc., how this, the growth has been made possible. Let me specifically focus on a database which primarily addresses the grasses. It's a database for plant genome, but the focus is grasses, including comparative genomics, genetic variation, gene expression pathway. You can access the public, database. There are no charges. You can access and do whatever you like in this database. So this Grameen, it is developed to the extent that you fed the raw sequence there. It gives you the phenotypic picture of the plant. So genomic data can be converted to the phenotypic data. And you have heard of, you know about the databases. We have different kinds of databases. We have, for instance, MODs, the model organisms databases, which are completely annotated, which are completely sequenced. For instance, Arabidopsis. For instance, Drosophila. For instance, rice. So these are model organism databases which are very, very often used in bioinformatics as a reference. So what this Grameen is, where does it fit? Or you have the completely open general databases of NCBI, National, this biotechnology information resource. NCBI, all of our, it's a resource for molecular biology information. So we have MODs and we have, open databases like NCBI, which are general databases. MODs, we have almost complete information about that genome. They are specific to genome, specific to that individual organism. And the NCBI open resource where you can, retrieve, you can see whatever you like. I mean, if it is available, if it is saved. This Grameen is somewhere, I would say, is in the middle of these MODs and the open general databases, because here we have many sequences which have been, for which complete information is available. But for many others, it is not. So it's providing a mix of information. So therefore, it's somewhere, transitory between, let's say, rice or Drosophila or Arabidopsis and the NCBI open source type of databases. Let's go back to 1989, which in fact is almost all the students of biological sciences are aware of it. They have to study it. That is the year when the Human Genome Project was conceived, and with the participation of some, let's say, it was a 15-year project, and global participation, many countries, including Japan, China, India, and so many. USA was the main, NIH was the main funder. They sponsored the project to the extent of 90%, but the other countries also contributed. So a global effort. It was conceived to be 15 years, started by J.D. Watson. And the human genome in that 3 billion base pairs was expected to be completed by 2003 or 2004. But then it was completed early. In 2000, I think, when President Clinton was the president of USA and the fellows involved, that is the government efforts, and the efforts by Celera Genomics, which was a private company, they undertook the task of human genome sequencing separately. They were, at war with each other, but Clinton made them sit together, and in a joint press conference in 2000, the human genome sequence ahead of time, 2 years, 3 years ahead of time was announced. Imagine now, if I talk of today, 2024, not much time is 24 years. In 24 years, see the speed that have gone in terms of the genome sequencing. In today's time, you pay some $600, go to a company, same day, they would give you the complete genome sequence of it. So imagine, I mean, so huge, huge multiple n times, I don't know how many, huge infinite number of times has been the progress in terms of the rapid genome sequencing. We had dideoxy and this time was Baxton Gilbert, and then we have this all high throughput sequencing technologies, biosequencing was the first one, and many more had come. So you see, so very rapid and very cheap, and very cheap. But then what do we do with this? That's the question. Let it come, I said 426 lakhs genome permanent drafts are there. What do you do with that? It's a junk information. Unless you make it understandable, you find out the function of these, identify the proteins, identify their functions, etc. All right, I mean, how does it matter? Nobody's bothered about it. So therefore, you have to follow the wet laboratory approaches for this. Wet laboratory experiments, they identify the functions of correctly, but then, God bless you, because, how many functions of, hundreds of thousands, I mean, so many sequences, 3 billion human genome alone, so many, organisms. So how many of them functions, proteins, 2, 4, 6, 10, 8, 12, that's all. By going wet laboratory route, it's an ocean. Where to start, where to go. So I don't think you would be able to contribute anything unless you supplement this by using in silico approaches. So although these wet laboratory approaches, they identify the functions of proteins correctly. But these are time consuming and costly, they have their own limitations. So, but functions of the proteins must be identified. And we have several bioinformatics approaches, which are called as protein-protein interaction, PPI approaches. And to quickly name a few, interlocks method, we have co-expression and co-regulation. The co-expressed and co-regulated proteins are more likely to be involved in simple biological processing. So I mean, these are different bioinformatics approaches which are applied in order to elucidate the function of the proteins, or phylogenetic profiles based on evolution of the genes, and then try to make a link between the functions of the genes, or gene label and gene cluster, that is the functionally related proteins that have the same operon. They are expected to physically often interact, which are under the expression of the same operon. And Rosetta Stone, we have protein interaction from homologous proteins in different genomes. And then finally, we have the gene ontology method. In fact, that is the one about which I shall talk for the talk, so in somewhat detail, not in detail, the approach that we followed. Now, if we talk of the serial genomes, now all major serial genomes have been sequenced. All major serials have been sequenced for their genomes. We have rice, which is the smallest one, around 500 MB, and going to the wheat. There's a lot of variation in terms of total genome size in wheat, but let's say average, I'm saying 17,000, barley 5,000, maize 3,000, sorghum 750. That was the last one to be sequenced, I think around 2016 or so. Sorghum is of interest to us. India is a country where sorghum is growing a lot. मोदी जी भी बात करते हैं मोटा अनाज की बार-बार आजकल, तो इसलिए और भी इम्पोर्टेंट हो गया millets. It grows very well in the climatic situation which are prevailing in our country, semi-arid, arid situation. So it knows how to grow there. It is also a C4 plant. C4 plants are characterized by higher productivity also. So when this sequence was made available, we were looking forward towards it. And We had a review and that's okay, it's not important. So it's the fifth most important cereal. Wheat, rice, maize, barley for food, feed, and biofuels. The plant is especially important, as I've already said, arid, semi-arid, etc. So the plant knows well how to naturally adapt to these conditions. That's the point I'm trying to emphasize. The genome has been sequenced. A small size genome sequence makes it an attractive model plant, even to study not just that it is able to grow under different adverse situations, but also from the functional genomics of C4 plants, productivity, which are more productive. So you see, this was a new, sequence that was made available, and nobody knew about the drought, especially we focused on the salt and drought tolerant proteins. We knew a lot at that time, so 2015, 16, we knew a lot about, for instance, Arabidopsis, one of the most explored, well-explored plants. But other than Arabidopsis, there were artillery plants which were known for different, or rice maybe, among cereals, if we talk, then rice. And then, about this, nothing was known, that how, what is the behavior of this plant under the drought and the salt stress. So using wet laboratory approaches, using electrophoresis, 2D electrophoresis, multi-TOF, using all these approaches, we characterize 5 highly expressed proteins. We grew the plant, we brought them to the laboratory, we grew the plant, provided stress under standard laboratory conditions, so many millimolar of sodium chloride, etc., and applied to 7 days old plants. And then, of course, we differentially expressed protein bends under salt stress using gel, then using 2D gel, etc. So then, so these 2D gel multi-TOF, and we found that these 5 proteins were the ones which overexpress. And we thought that since they are overexpressing under the high salt stress condition, they should be the suitable candidates for providing tolerance to the salt conditions. And then we've identified these 5 proteins in their molecular weight, because that could be easily done. So ATP is F1, we have FO, F1 ATPase, and F1 component is the catalytic component of ATPase. Ribosome maturation factor MP, inorganic pyrophosphatase, BET-V1, which is a birch pollen allergen, one of the most widely spread pollen allergen. At the onset of spring, many people have problems, and it's because of this BET-V1. And universal stress protein. Then we refer to the GEO database. I believe that all bioinformatic people will be aware, and it's useful to useful across all disciplines, because GEO is a consortium. It's an initiative done by the scientists involving biotechnologists, involving the computer scientists. It's a joint effort. And essentially, it focuses on the functional identification of genes. And functionally, all protein functions, they are divided into 3 categories. That is 3 domains only: biological processes, molecular function, and cellular components. So all proteins functionally in the GEO database, they are just divided into these 3 categories. And all proteins can be, depending, for instance, the location protein structurally contributing cellular process. If it's a protein which is responsible with binding, enzyme, etc., it goes to the molecular function, or involved in differentiation, etc. processes for biological function. So all proteins can be characterized based on their functions into 3 categories, and that is what this GEO database does all about. Till now, another very, very important difference. Till now, we have a lot of databases. It's not that Geo was the first database. Already we have hundreds of databases in different areas, maybe small, maybe big. But then Geo database, not only in terms of that it categorized all proteins based on their functions, but it was important, rather more important, based on some other criteria. Till now, the databases named the protein or the gene based on some name. I mean, say for example, if you talk of the PS2 protein, it will name the D1 protein, which is an important protein of PS2 involved in the photosynthesis process. That is, they were provided descriptive names, biological names. That is what I'm trying to say. Now, providing the biological names created some confusion. For example, if I provided some name working in some laboratory, since there was no perfect coordination, there was coordination because these were after all public databases, but there was no perfect coordination and no way where you could just, by one click of command, know that this protein has been named so in the database. So some other person, scientist, working in some other laboratory could give the same protein a different name. First confusion. So many times we have multiple names, biological, descriptive process, descriptive names, multiple names for the same protein. And in fact, we wrote a paper about it, which is the title is at the top in current science. Our paper only. Annotation jargon is not too late to correct it. When we noted these anomalies, then we decided that it should come out as a publication. What did this Geo did? What did they do? What was the specialty? All these terms are machine readable. So you can do a lot of computing in this. You apply all computational approaches to this Geo terms. So they are human readable, they are machine readable also. Machine would be able to interpret. So that was their specialty. And so therefore, the latest release, 17 June 2024, Geo terms 42,093, gene products 153,000, 1.537645 million, and coming from more than 5,000, that is to be precise, 5,409 species. So it's a huge database. What did we do? As I initially said, when I was trying to build up the background, that wet laboratory approach would be restricted in their, be restricted, and they can do only certain things. They are highly time consuming. So combined with in silico. When I say that we identified 5 proteins, we decided that if we now match these 5 proteins to the Geo database, what Geo database say about it? Are they corroborating with the same function as we predicted? And why Geo we did, compared with few other databases as well? Because see, what happens is that, for instance, Grammy and I talked. Now, anybody who is working on the cereals would refer to rice in order to annotate that gene. If they don't find in rice, then of course there's a novel gene or whatever. But they normally make MOD as a reference point. Rice is the MOD for cereals. So we followed what we call the SP analysis, shortest path analysis, which is working on the geoterm semantic similarities, which essentially mean that, for instance, car, bus, or SUV or whatever, they are carrying out the same function. But if you talk of the road, all the road is related to the car, but it is only related, so it's a relation, not the same function. So Geo database functions on the basis of the same functions on the basis of the function of the protein. So this semantic similarity provides functional relationship between biological process and molecular function and cellular component. So that is the picture that emerges. I will not go into the details. So you can link the proteins which we identified based on this SP algorithm with certain kinds of functions. You can match them. For example, we match with magnesium ion binding protein, which was pyrophosphatase. ATPase, inorganic phosphatase, protein, serine, furin, kinase, different beds could be correlated. They could be found in the geodatabase. So see what we are doing, we are further validating our results. We are further confirming, I mean, vice versa, in silico lab or lab in silico. You can compare the data with the databases. Molecular size of protein, go to the following in silico approach. So like that, binding, catalytic, so what all functions they were involved. Not only this, we looked for some other databases also, for instance, Interpro. Interpro is another important database, which is a host to the, it's again run by Interpro Consortium. And it is a host to many proteins. And it's again working on the function of the proteins. Database working on the function of the proteins. So you can again compare your proteins with another database, get another confirmation or rejection, whatever. So first column, you find the name of the protein, which was uncharacterized. If you go to Geo, what it says. If you go to Interpro, using Interpro scan, and then to the most informative common ancestor as per the Geo database. So find out, do they match? Similarly, you can go to another database, which is a database of cluster of orthologous proteins. It's called as Cognatus. And again, do the same thing, find out how they match. What is the molecular weight? What is the isoelectric point, PI? In sorghum bicolor, which is a, which you are trying to understand, compare it with MODs, Arabidopsis thaliana, Orida sativa, and what is the hit rate coverage. So all these parameters to further substantiate your results. Okay. So that was one towards solved. We developed a similar approach for identifying highly expressed proteins under drought stress. Did the same thing. And then we also, under drought, for instance, you deprive the plants of water and see what kind of proteins are expressed, overexpressed, and do 2DE, do maltitol, and based on this, we identified the over 6, which were overexpressed in drought. The question is that whether the proteins which are expressed under salt and the ones which are expressed under drought, are they common? Do they have a common linkage? Or are they different, that some of them working under salt, some of them working under, drought? So you can, using the same, geo databases, etc., you can explore association networks and find out whether they have some commonality in them. Because ultimately, it's not that, plant has one mechanism for salt. I mean, you have to see whether it is true or not. Maybe some are unique, some are not. So you can compare, for instance, how they are linked through geodatabase, binding, catalytic responses to metabolic processes. So like that it goes. So these were the 6 proteins and then their links, that how many of them were common to both salt as well as drought. So, I mean, this opens up immense possibilities then, because you can also try to find out the proteins which are, formed under biotic stress, with abiotic stress, develop what are the common pathways, find out some proteins which were common, which are common to all, etc., etc. So many things can be done using this. So, okay, it's all well published. And should I take 5 or how much time? Okay, 5 minutes. So, just one or two slides to, let's say, give a glimpse of the other thing which we do. Just one example, drug designing. Drug designing, we take it from the animal perspective, not from the human perspective, but with plants. So a drug discovery process is a very lengthy process. We are all aware of it, involving huge cost, $600 to $700 million. And so many steps, discovery, you have to find out target for the drug, identify the target, optimize the target, then find out appropriate drug which will act on the target, test some leads. Some of these leads would lead to development of drugs. Then they are taken to the preclinical testing, trials, etc., etc. So 15 years gone, and a lot of money is gone. Here, bioinformatics holds a great process. There are so many things that can be done, but I think maybe the last slide, because I'm not going into the details. So you see, you can develop, for instance, a lot of promise. We have, for instance, broad spectrum antibiotics, and we have narrow spectrum antibiotics. And we are all aware what is the difference. So when we go to the doctor for the first time, वो बिना test के he will prescribe कि okay, you take this. This is broad spectrum. गले में भी तब भी ठीक है, इसमें भी तब सकती है. Everything is fine. For narrow spectrum, the doctor has to go in for clinical isolation, maybe some testing, culture, even cultures or blood tests, blah, blah, blah. And then only narrow spectrum antibiotics is possible, but generally it is not done. Initially broad spectrum, which in fact is harmful, we are aware, because it plays also with a lot of friendly microbes. Same thing is happening in plant kingdom also. We have indiscriminate spray of fungicides, bactericides, which kills. Even, for instance, which would eliminate not only the crops which you where you wish to work with them, weeds, etc., but it will also eliminate the crop plants nearby, which happens with weedicides. Or if you are using bactericides in a legume plant, it will also eliminate nitrogen fixing bacteria. It's a broad spectrum. So everything under the sun is killed. It does not discriminate. But now when these days you have the lot of plant genome sequences, you have the sequences of the pathogens, that is of the bacteria, of the fungi, then can't you develop tailor-made drugs? So for instance, if I spray bactericide on a legume plant, rhizobium is living happy. Nothing happens to it. But the actual culprit, which is, for instance, Pseudomonas viriworm, is killed. So that kind of promises are there, because you can do a lot of exclusions, inclusions, find out which are pathogen specific, which are specific with pathogen, test them on rhizobium. Rhizobium is not killed. Test them on your model, pathogen is killed, fine. Identify those proteins and develop your drug. So we did this for Pseudomonas viriworm. Fascio is the last slide, I think, is a major plant pathogen, infects a wide range of plants, including the model plant Arabidopsis and other major crops, also causing a devastating disease, hello blight disease in beans, for which we worked. So based on the approach, which I just summarized orally, I am not discussing in detail, we were able to, come out with certain proteins which would work as the drug targets. So thank you so much for your attention.
Any questions, anyone? Any questions, any queries?
So no queries. I think queries can come up later also. But if you have any queries, most welcome. If they are not, the session chairs have to see whether they are running short of time. ठीक है, मैं बैठा हूँ, जब तक आप बोले बैठो, बीच में कोई query.
Thank you so much, sir, for sparing your valuable time and giving such an informative talk on plant stress biology. Moving on further, I would now like to introduce our session chair, Dr. Pushpendra Sharma, sir. Dr. Pushpendra Kumar Sharma is currently working as an associate professor at Amity Institute of Biotechnology, Amity University, Rajasthan. Prior to this, he has also worked as an assistant professor at Sri Guru Granth Sahib World University, Fatehgarh Sahib, Punjab, Chandigarh. He has earned his PhD degree from Department of Biotechnology, Punjab University, Chandigarh, and awarded a postdoctoral fellowship by DBT India in year 2010, which he has pursued at IISER Mohali. He has also been awarded with Young Scientist Project by SERB, New Delhi in year 2013, and has also has more than 18 years of teaching and research experience. His research work is focused on genomics and proteomics that mainly include metagenomics, metatranscriptomics, protein engineering, identifying and characterizing novel genes and enzymes, and pathways using metagenomics and transcriptomics. He has also been granted a patent for protein engineering of thermostable lipase for its enhanced thermostability and activity. Sir has also published more than 60 research papers, review articles, and book chapters with cumulative impact factor of more than 250, with I-10 index of 25 and H index of 20. Dr. Sharma has guided 4 research scholars for their PhD degree. In addition, Sir is also the core member of Amity Science Technology Innovation Foundation and is also the coordinator for Center for Nanobiotechnology and Nanomedicine. Alongside, he is also actively doing research and guiding several UG and PG students for their research projects. So with these few words, I would now hand over the session to Pushpendra sir.
Thank you so much, ma'am, for the nice introduction. First of all, I would like to welcome today's guest, Dr. Manoj. I must, before going into detail about his CV, I must say that Dr. Manoj is associated with me for past more than 15 years. During when I was assistant professor in my previous university, Dr. Manoj was basically, he's a brilliant scientist, and to really meet his scientific thirst, he has moved to a country like, Singapore, US, Canada, Australia. And he's today going to talk about, welcome, Dr. Manoj.
Hello, thank you, Dr.
Pushpendra. You have to really wait for a long time because our session is expanded a bit more.
Yeah, that's fine. That's fine. That's
We're simply waiting for the time. And this talk will focus on doping in colloidal nanocrystal emerging optoelectronic properties and devices, which I think is a very unique area, and we can learn so many things out of this, because this is different, because sir was talking about genome and proteome, and this is entirely a different, but yes, because this is a thirst area now into a number of applications. So before Dr. Manoj, I will call him to talk. I would read his CV. Dr. Sharma has completed his PhD in 2011 with distinction. He has worked as an assistant professor, visiting scientist, and senior research fellow at different renowned universities around the world. Manoj Sharma currently works as senior research scientist with ARC, Center for Excellence in Excitation Science at Monash University, Australia. Dr. Sharma research focuses on energy efficient luminescent solar concentrator, light emitting devices, and novel optoelectronic of colloidal nanoparticle, led by him at NTU Singapore. This work has been recognized worldwide and almost appreciated by Singapore government also. He has 13 years of post-PhD experience at the level of assistant professor, senior research fellow, program manager, scientist, and he has worked in leading world university like NTU Singapore, Monash, Melbourne, and many more. He has published many research articles in top tier world famous journal that include advanced material, nanolater, light, science and application, chemistry of many material, proceeding of IEEE, advanced functional matter. He has presented research on future energy saving technology, which include building integrated nanocrystal photovoltaics based greenhouse agriculture. And his expertise is well recognized by the research community across various countries, like including Australia. So he was also into news views at Australia. His innovative research work in Australia was being highlighted in news views, as I discussed. Furthermore, he has collaboration all over the world to carry out his research activities, making him a global researcher. He is often invited to various prestigious international conferences worldwide, and is often invited to various prestigious international conferences. He has won several awards. His major research output has significance in the field of net zero energy building, photovoltaics, and advanced quantum technologies. Thank you, Dr. Manoj. I think all of us will learn today so many things out of his work, and a different area that would basically be, maybe some of you can be importantly looking for such kind of work in future. Thank you so much. And now I would ask Dr. Manoj. Already we have, I think wasted many time, so much time, and you are waiting for a long time, for maybe around 1 and 2 hours. Please talk, share your slides and start talk. Yeah.
Yeah, thank you very much. Thank you, Dr. Sharma. Can you hear me?
Yes, yes. Yes, you are audible. Everyone can hear, please.
Can everyone hear me? Okay, all right. Okay, I try to be loud. Thank you very much, Dr. Pushpendra. It was nice talking with you after a long time. And I also thank the organizers for the conference who have invited me to present some of our latest research. So also, I think before me, Professor Vinay Sharma has presented a very nice talk on the topic of plant biotechnology and how stress is an important parameter for the crops, different crops. And although my topic is a bit different, but I try to connect like this with different audience related to biotechnology as well. As Dr. Pushpendra mentioned that these kind of technologies may have a huge impact in different diversified fields. And also it is always said that new novel ideas come when people from different research backgrounds, they basically interact with each other or brainstorm. So that's how the novel research comes. So without wasting time, so I will go to the topic of my talk, and in between, since it is an online platform, in case the audience are not able to listen to me, so please stop me in between so that I can check in case if there is a problem with the voice or the slides. So the topic of my talk today is related with doping in colloidal nanocrystals and their emerging optoelectronic applications. As Dr. Sharma has already introduced me, so I work presently at Monash University, Melbourne, and am also affiliated with Center of Excellence on Exciton Science. Basically, that is a 7-year of funding given by Australian government for high-end research on exciton science and related topics. So I selected few pictures. It gives you some ideas on the topics which I will be presenting. So I will be basically presenting the work which I have done in last 6 to 7 years during my affiliation at NTU Singapore as well as Monash University Singapore, Monash University Australia. So where currently I am working. So what actually I do, as you can see that from the picture here. So so this is my picture working in a glove box system in Singapore. So we basically do smart colloidal nanocrystals. So what are nanocrystals? So I will quickly come to this topic. So this has been in the news in last year. Nobel Prize in Chemistry has been awarded for the discovery of tiny quantum dots. So these are the three basic scientists who were awarded this Nobel Prize because of the discovery of this quantum dots. So what are quantum dots? So quantum dots are tiny particles, tiny semiconductor particles that have new emerging optoelectronic properties. So what are that properties? For example, as you can see that when we decrease the size of a particle below certain radius, then the band gap of the material changes. So now if the audience is not is from different background, they can see that from this particular image, that when you decrease the size of a crystal, then the color of that nanocrystal changes. So that was something very fascinating at that point. So how a atomic particle which has how a small semiconductor particle which has few atoms only, like for example 10 to 100 atoms, how they behave differently as compared to their bulk properties. So that is coming from a Venn diagram which in class 12th, actually most of the undergrad students who are sitting here, they might have studied that we have metals and we have semiconductors. So metals have an overlapped band gap, valence band and conduction band. However, when we go to the semiconductors, we can tune this band gap. So now when we go from bulk to quantum dot, QD is quantum dot. So what you can see that the valence band and the conduction band have different energy levels. Basically, this continuous band has been split into small atomic levels. So that has a very important implications on that applications. So how that energy levels have been split, that is a very important thing to understand. So first thing I would say, in a common language, you can see that a bulk semiconductor would have millions and trillions of atoms. So millions and trillions of atom in a cubic centimeter particle will have millions and trillions of energy states. That's why they become continuous. However, when we are talking about quantum dots or tiny nanocrystals, they basically have few atoms only. So few atoms means they have very few energy states. So that's why you can see that once you change the band gap, so the energy states are getting off, and then you can see that you can tune the band gap. So I try to explain in a simple language, but if it is not clear to some of the audience, that is fine. You can just take a message that the discovery of quantum dots was very important in last 4 decades, and that's why this has been given Nobel Prize, and what impact that has on different applications. For example, as a student of biotechnology, you might have also and chemistry, you might have seen a lot of research papers on sensors, on photocatalysis. So all these applications, along with photoluminescence or electroluminescence, all these research directions have been impacted by the discovery of these quantum dots. So you can see that these are the percentage contribution of quantum dot based research worldwide in the last 4 decades. So you can see that the role of quantum dots have increased tremendously. For example, 50% of the papers are getting published with a title name as quantum dots if they belong to photoluminescence or photocatalysis. And these have huge implications for future energy saving devices. So how many types of quantum dots we can create, or how many types of quantum confinement we can create, as you can see from this diagram. So the picture A is bulk, which is bulk solids, which we generally have in our life, day-to-day life. However, when we decrease the size of this material and manufacture it at a very small size, then we can have a various kind of quantum confinement. If we confine the nanocrystal in only one direction and allow the particle to grow in other two directions, then we say it as a quantum well. That is like you can imagine like a piece of a paper. Piece of a paper, however, the thickness of the paper has to be in the dimension of nano, for example, 1 to 10 nanometers. So then that kind of a quantum structure is called quantum well. And similarly, if we make these as a rod, in that sense, the quantum confinement has been coming in other two directions, and in one direction it is allowed, then these kind of quantum particles are called quantum wires. And if you try to manufacture a particle in which you are squeezing the particle in all the directions, then you are getting a 0D kind of a dimensional particle that is called quantum dots. And the energy levels of these three different quantum objects are very different as compared to bulk. So based upon these different energy level possibilities, they have their applications in different fields that are related to biophysics, biotechnology, nanotechnology, and different diversified fields. So coming to very basic question, so why we are so curious about this nanocrystals as the Nobel Prize has been given last year. So what is the use of these colloidal semiconductor nanocrystals? So basically they can have application in two fields. Very simple. I would say photons. Photons are the quantity of light. As you can see, we are sitting in the room. You can see the light is coming from different bulbs or LEDs in the room. And the nanocrystals can have application as a charge carriers. So nanocrystals are generally synthesized to get two things. First thing is photons, second thing is charge carriers. So where charge carriers are useful, so charge carriers can be useful for photovoltaics. For example, silicon solar cells. You all know now we have silicon solar cells on the rooftops. So how that silicon solar cell work is basically a PN junction kind of system. So when the sunlight shines on silicon, you have electrons and holes created. Now that electrons and holes needs to be extracted out. So how much efficiently you can extract or how much charge carriers you can generate, that will decide the efficiency of the device. Now, if you want to go beyond silicon, you want to make new kind of solar cells that are next generation solar cells, for example, perovskite based solar cells. So then what we need to engineer, we need to engineer the charge carriers. So that's why these are the two very important things. They may have application in different diversified fields. As you can see on the left hand side, I try to highlight application related to lasers, which has huge implications in bio based research as well as medical fields. So generally you see lasers all over the place nowadays. However, these lasers are based upon semiconductor bulks, but not nanocrystals. But however, in next 10 to 20 years, you will see that the lasers will be made from colloidal nanocrystals. So what will be the benefits of this? The photons. So for example, you can see that the lasers are red, green, and blue. Why don't we have an orange laser? Why don't we have an infrared laser? Why don't we have an cyan laser or yellow laser? So that kind of answers are given by quantum dots, because as I mentioned previously, quantum dots can tune the color of a particle with a very precise wavelength. So you can't make these kind of a fine tuning with existing technologies in the bulk. Similarly, charge carriers, as I mentioned, they have a huge implications on these kind of industries. So now coming to topic, so I will quickly skim some of the slides in case some of the audience don't find it useful. They say if they're not understanding, that is fine, because it might be a different field from their background. However, you will try to get some, I will try to give you some insights that are related with diversified fields or applications. So the title here is Colloidal Cadmium Selenide Quantum Wells. So what are quantum wells? Quantum wells are particles, they are like nanosheets. So here the quantum confinement is in the z direction, that is the thickness. As you can see on the bottom here, this is like the transmission electron microscope image of the nanocrystal having thickness of the order of precise atomic layers. So you can see that we can even calculate the atomic layers as one monolayer of cadmium, one monolayer of sulfur in that sense. So where they are useful, so by controlling the thickness of these nanosheets or quantum wells, you can tune the emission color. So this on the top, it is the photoluminescence emission spectra, so which basically shows that we can have 450 nanometers, like 500 and 550. So you can tune the emission color by changing the thickness of this quantum well. So that is very, very important thing to generate. As I mentioned that for lasers, you generally have a green or a blue. If why don't you have an in-between color? So by these kind of technologies, if we can uh, make them very efficient, these technologies can have all the colors which currently we are missing in already available commercial lasers. So they have huge implications for different security, optoelectronic applications, as well as for medical diagnosis. So another thing is we were involved in 4 years back for this particular work, where we for the first time we make some heterostructures of colloidal nanoplatelets. So what are heterostructures? Hetero means we have a core nanoplatelet, and then we systematically grow in chemistry labs. We can grow a crown or a shell on these core nanocrystals. So what this will do is this will improve the optical properties of these materials. For example, these materials, they are already good, but however, their quantum efficiency suffers. So what quantum efficiency means, when you excite this material, when you provide voltage to this material, then they emit some light. So that efficiency of that emitted light, it has to be better and better. It has to be brighter and brighter. So in order to do that, we generally grow these heterostructures. So that are basically, you can see that core crown and core shell heterostructures. And you can see that we can characterize their different colors and improvement in the efficiencies. So next kind of a control which we can try to generate from these quantum particles is by doping these nanocrystals. So what doping means? Doping is basically you are adding intentionally some of the materials, some of the guest material into the host material. So in order to modulate their properties. So what properties? It can be any properties. It can be optical properties. It can be electrical properties. So that intention is that kind of a thing we can generate by doping these semiconductor nanocrystals. So here copper is a earth abundant element. It has been shown to be doped in cadmium selenide semiconductors. So they are, after doping, they can generate different colors. So they can shift these materials emitting in the visible and can convert them to emit in the infrared, which has huge implications for bio-based research. As some of the students who are having bio background sitting in the audience, they may know that the penetration depth of the tissues is higher with infrared light. So if you want to do diagnosis for early stage cancer or these kind of applications, so you need the light to penetrate into the cell, as well as it should not damage the cell, and it should have some fluorescent markers. So for that kind of a technologies, we need a wide spectrum, and which can be controlled and have its known properties. So that's how these kind of a concepts that are, for example, with doping, we can have a Stokes shifted emission. So we can tune the absorption, we can split the absorption and emission. So here this system is absorbing, for example, in the blue, however, it is emitting in the infrared. So this work was already there. So back in 2017, so I just published this work. So I started this work 2014 when I was in India actually, with Dr. Pushpender Sharma. So then I moved to different parts actually to continued this work, and I have spent some time in IC Bangalore with Professor E. Sharma. So I learned some concepts related to these kind of topics. Then I have some affiliations with Turkey, and then I moved to Singapore. So finally, that work was published when I was in Singapore in 2017. So this was the first report where we have successfully doped copper into quantum wells for the first time. So this has an huge implications for different optoelectronic devices that we discovered in last 5 to 6 years, and it also has been followed by different groups worldwide after us. So basically, we designed a method by which, instead of, you can see that in the top, these are small, tiny spherical quantum dots. So we can also dope them into flat nanoplatelets. So what is different when we dope copper into quantum dots and nanoplatelets? So that has very huge implications. Maybe in the last part of the presentation, I will try to highlight. However, on a simple scale, you can see that doping copper in quantum wells or nanoplatelets gives 80 to 90% quantum efficiency, so which is very, very high. So that kind of a quantum efficiency means that almost all the absorbed photons have been converted into infrared photons with 100% or 90% efficiency. So that was a huge milestone in this particular field, because prior to this research, the copper doping in quantum dots, which was also very good, they were only able to get 30 to 40% quantum efficiencies. So these are some advanced characterizations. I will not go to the detail, but these are how these materials look like in solutions. They can generate different colors. So this is some elemental characterizations by which we prove that the copper has been doped successfully into the systems. So later in 2018, we also followed another research paper where basically we try to understand that how copper enters these nanoplatelets and how it is and how that is responsible for generating that highly efficient emission. So the title of that paper was also very I remember that was we selected as a journey of dopant ions. So I did some experiments on a long month scale time. Then we were trying to understand how dopant ions start from surface of the nanoplatelet and goes into the quantum wells, and then how they generate this kind of a Stokes shifted infrared emissions. So also we followed this by some elemental characterizations that you can see here, the transmission electron micrograph showing that we have successfully doped these systems. So now where these particles we have used, as I started in the presentations, so these objects are responsible for generating photons or for carriers. So here, as you can see that this is an example of a luminescent solar concentrator. So what does that mean? You might have seen that nowadays living in cities, for example, in Jaipur, even in MIT universities, I have been to MIT in Noida campus, but as you can see that we have high-rise buildings like mega cities in the world. So the space is getting lesser and lesser. So when I was in Singapore, that was very highlighted a lot, because in Singapore you can see that the population density is very, very high. So you will have people living in apartments in multi-story buildings. So how you will add solar cells on that rooftops? So you can put solar cells on your rooftop or at your home. It can be good for generating electricity for one household. However, when you have rise buildings, the top part is very, very small. So that solar cells cannot generate enough electricity to power the whole building, which is having 100 apartments or like 500 apartments. So how would you address the need purely based upon solar photovoltaics? So then these kind of a novel concepts called building integrated photovoltaics came, in which LSC is a part. So in that space, you can imagine a glass window that is semi-transparent, or I would say transparent, in which this window can generate an electricity. So for example, in this glass, we can add some smart nanocrystals that can absorb the solar light and can convert that light into photons, which will travel all the way to the edges. And on the edges here, as I'm highlighting, you can see in the slide that on the edges you can attach a solar cell. So that solar cell will absorb that emitted light and that can generate current. So basically, you can imagine if it is a 100-story building with all the glass facets, you can impart that glasses with nanocrystals and that can generate electricity. So that would be very interesting thing to observe. So worldwide people are trying to address that. So these copper doped quantum wells have a huge implications in that. So that's what we show in 2017 and 19. We did some experiments on this copper doped nanoplatelets. As you can see, these are emitting in the visible as well, like orange. Also, we can generate infrared light of it. So we did some experiments, and at that time it was showing we reported the record efficient doped luminescent solar concentrators where they can generate enough electricity when they are doped into semi-transparent glass windows. So here we are also continuing in Australia with some startup companies to further commercialize these efforts. So these are different examples of luminescent solar concentrators that we generate. So now, on another note, as in the previous talk, Professor Sharma has mentioned about some crops and how to address issues related with crops. So you can imagine these kind of a smart windows are also have huge impact on agro-photovoltaics. For example, in the space of in the field of agriculture, the concept of greenhouses has been emerging. Just imagine if we can cover the rooftop and the sides of the greenhouses with these kind of a smart nanocrystal inks, which can convert the solar light into a suitable color that the plant plant absorbs efficiently. Just imagine it is a green or it can be red for different plants. So you can increase the yield of the crop by putting these smart nanocrystals as luminescent solar concentrator films on the greenhouses. So that has a huge implications, and people are also starting up. And I hope in India also lots of groups are working on this as well. So now coming to another application of these same nanocrystals, which are doped copper dome nanoplatelets. In 2019, basically, one of my student in NTU Singapore, he took this project. For the first time, we showed that we can have a mutual donor acceptor phenomena. Generally, in biophysics, you know that for understanding different phenomena, we use donor acceptor players. So we have two materials in which one material can absorb one portion of light and can transfer to the other. So that can be used as sensor. However, in this kind of nanocrystals, we use a doped, one undoped and one doped system. So that can generate a pair of a system. For example, both can work as a donor and acceptor. How they can work? For example, the blue light of this 3 ml colloidal quantum wells can basically be transferred to 4 monolayer system, can generate green color. And the green light from this system can be reabsorbed by the same 3 monolayer to generate a red color. So in that system, so by varying the pump power, we can generate all the visible colors from green, blue to orange within a single nanocrystal compositions with a cross-check configuration system that has huge implications on the field like biophysics, in which you can probe the donor as well along with the knowledge of the acceptor. So without going into the detail, another application which we used for that kind of a smart new copper dope quantum wells is related to lasings. I know that the audience might not be familiar if they're not from physics background here. However, these smart chemistry-based nanocrystals which we synthesize in chemistry labs, they may have huge implications in physics. For example, for the first time, we show that we can generate a sustained bi-exciton emission. Bi-exciton means we have a single electron hole pair when we excite, and we can have a double electron hole pair as well. So we generated these kind of phenomena, and we demonstrated in 2021 with the demonstrations of first vertically cavitated lasers with a very record low threshold of 7 microjoule. Means at very low power we can make these nanocrystals as lasers. So that was I started in the start of this talk. You can see that how a spontaneous emission has been converted to a very sharp blazing peak. So that is a green laser we show at that time. So now, if I come to the difference between, I have said about the synthesis of novel copper doping in nanoplatelets or quantum wells, and I also demonstrated some applications of these which we have done. Also, people worldwide are following this, and they have doped different elements after following our work. For example, copper, mercury. So now, what is the difference? As you can see that in this slide, I try to highlight on the left-hand side, these copper dope nanoparticles or quantum dots, so they can generate phonon bottleneck. Basically, that in these systems, you can create charge carriers. However, the same dopant ions, when we dope in nanoplatelets or quantum wells, they can generate cascades. So in another way, the hot electron cooling is very fast, or in simple language, I can say that they can generate very efficient photons. So the same dopant ion, if it is doped into quantum dots or quantum wells, they can generate different things. For example, in this case, it is generating carriers, good for solar cells. In this case, it is generating the photons, which is good for lasers or LSCs. Then I just quickly highlight a few examples of different lasers we fabricated from these materials covering the whole spectrum. In the last few years, I'd quickly highlight that the topic of perovskite nanocrystals is highlighted a lot, and a lot of groups worldwide are working on this, and this has an huge implications in the field of solar photovoltaics. For example, a sheet of perovskite nanocrystals doped with ytterbium can quantum cut a photon in the UV into infrared light with a double efficiency. So this is novel concept was available for last 50 years. However, very recently with the discovery of perovskite nanocrystals and the ability to dope ytterbium efficiently into the systems, we can for the first time show very high absorption cross-section host that can absorb all the UV light and can efficiently convert in infrared, which the silicon can absorb. So with that, people have demonstrated some applications of these systems. So in the last few years, we have been working in this field that we are shifting the same concepts of doping and nanocrystals, but in a different host, for example, perovskites, which can generate the emission in the visible, in the orange, as well as in the infrared light. And we still have to publish some of these efforts. So we are basically trying to use these materials for different optoelectronic devices. So in the summary, I will quickly summarize what we have done. Hope it will give you some insights that for the first time we show that the doping can be done into colloidal quantum wells, and these dope nanocrystals have an implications on two important aspects. One is charge carriers, other is photons. So how we can generate the charge carriers, for example, free electrons and holes for different solar cells, or we can use these as photons for lasers or luminescent solar concentrators. It can be used for building integrated photovoltaics, or it can be used for photocatalysis. So then we are further extending these research topics into different fields, and I want to thank you all with this some selected cover articles which we published and some nice drawings by students from our group. And I acknowledge different collaborators and professors and the funding agencies in Singapore and Australia who supported us to conclude or finish this work. Thank you very much. I will be happy to answer some questions. Hope it might be interesting to the audience. And if there are some questions, I will be happy to answer. Thank you.
Thank you. Thank you, Manoj sir, for such a wonderful lecture. What I could understand out of the lecture basically is, this copper doping, etc., and doping of all this material can be used for future energy generation, definitely. And they can have many application to medical area also, I think.
Yeah, absolutely. Because, for example, as I mentioned, so these kind of lasers, they are very potent, and they have been already under development by some companies, and they have tie up with some biotechnology and medical diagnosis fields. So people are already currently using these things.
So it can also be very important for diagnostic, diagnosis of the disease,
Yeah.
for example.
Yeah.
So thank you, Manoj, for sparing your valuable time. It was, I think I saw you after almost 13 years.
Yeah, it has been a long time. Hopefully, you and your family are fine. So we will try to connect some other time offline as well. So if there are no questions, I thank the audience for
Yes, yes.
patience.
Any question, please, student? You have any question, you can ask. Any question? No question? Only genomics and proteomics? So I think, yeah, your lecture was great. It would have been more better if you could really physically present here and interact with the student.
Yeah, definitely. I think that's I resonate with you on this. Yeah, exactly. Yeah.
Really, thank you so much. And definitely we'll call you at some point if you will
Yeah.
visit India. So
Yeah, I will let you know, and then when I'm visiting India, then hopefully we can have some more interactions, and then I can interact with students, and that would be more convenient. Students can ask face-to-face questions if they have that gives them some motivation.
thank you. Thank you,
Thank
Manoj.
you.
Thank you. Thank you so much.
Thank you,
Will
everyone.
yeah, definitely the certificate and all that will be, I'll be forwarding everything, rest of the, yeah.
Yeah, that would be great. That would
Thank
be
you,
great.
Manoj. Thank you
Okay.
so much.
Thank you very much. Thanks, everyone. Bye-bye. Bye-bye.
Thank you. कितने लोग हैं पार्टिसिपेशन के लिए 10 है मैं बोल देता हूँ कितने बच्चे हैं
अटेंडेंस बोले थे आप कुछ बोलना
Okay, I'm also the session chair. Please, student who have basically registered for their presentation, please have tea and then you can come back soon because we'll be having the, it's already 12:30. So within 1 hour we'll finish. 10 student, 12 students are there. 13. आप में से कितने लोग हैं जो भी प्रेजेंट कर सकते हैं? मीन दे आर फीलिंग दे कैन बी हियर एंड प्रेजेंट आफ्टर टी, इमीडिएटली आफ्टर टी, चाय पीने के बाद तुरंत। कौन है जो प्रेजेंट करना चाहता है यहाँ पे कि नहीं है? ऑनलाइन है। प्लीज रेज देयर हैंड हु आर फिजिकली प्रेजेंट हियर। स्टूडेंट्स, एनी ओरल प्रेजेंटर, भविष्य और एंड अदर, बस सब तुम ही हो? तो 4 तो ऑनलाइन है, 5 ही हो फिर।
नहीं अनाउंस आप लोग करना, मैं आपको यू आर द सेशन नाउ वी आर मेकिंग द सेशन। पहले मैं अटेंडेंस लेके फ्री कर दूं बच्चों को, ठीक रहे ना इनको जाना।
थैंक यू। आजकल मैसेज किया ना, मैं टाइम ही नहीं मिला।
अरे कोई टीचर अच्छा Students, attention please. Now I am going to take your attendance. Okay, after that you can go. Only the presentation, oral presenter can stay here. Rest can go. Okay. BTech Food Tech first semester. Hardik Sharma. Sona. MSc Biotech first semester. Tanmay Sharma. Jyoti is not here. Natuva Suma. BTech Bioinformatics 5th semester. Harshita Sharma. Ritika. Priyanshu. Shri Krishna. BTech Food Tech 3rd semester. Shruti Gupta. Kunal. Ansh. Dheeraj. MSc Biotech 3rd semester. सर अभी जो यहाँ पे बिठाए थे उनकी अटेंडेंस दे रही हूँ बस। अच्छा सॉरी। MSc Biotech 3rd semester students attendance please. Sanjana. संजना, मानसी, कनिका, हर्षिता, नीति को छोड़ के बाकी सब एब्सेंट हैं। ऋषभ, जानवी, ये सारे ब्लॉक्स। फूड टेक 7th सेमेस्टर। रोहित सैनी, रोहित सैनी हो तुम? फिर मेरे को ना कुछ बच्चों से एक मैं अटेंडेंस में ले लूँ उसके बाद ले जाऊँ।
अरे बहुत हमको अर्जेंट काम है भाई, सर बुला रहे हैं। रुको आपके पास
तो नाम भेज देना, आई विल मार्क इट।
ओके ठीक है, मैं नाम भेज रही हूँ आपको।
ओके
हिमांशु, Food Tech 7th semester students, attendance please. Rohit Seni, Himanshu, Sakina, Mansi, and Rohit. BTech Biotech 7th semester students. Akash Sharma, Mohit Singh, Dhitika, Ashita Pari, Amisha, Dakshita Tandon, Dharmendra, Khushwardhan, Ayush Sharma, Ayush Mathur. BTech Bioinformatics. कौन सा सेमेस्टर है? और भी सोनी, गर्विता, तनुप्रिया, MHM दीपिका, रुद्र पटेल, भव्या, ठीक है, भृगु, रियांशु, ओम, युगल, रुचि, डायना, गौरव, अभिनव, ईशा, स्प्रिया, तुषार, वंश, खुशी, सानिया, श्रुति कीर्ति, पलक अग्रवाल, हाँ मैंने ध्यान दिया। MSc Food Tech first semester, Rushik, कहाँ? Please be seated. I have not finished yet. Rushik, Abhishek, Ansh, Dhruv, Vivek, Bharti, Nandita, Muskan, BTech Biotech first semester, Shashank, Manas, Shivambika, Vraj, Kartikeya, Rishabh, Gaurav, Ravindra, Tanish, Priya, Kavya, Nishtha, Khushi, Nimisha, Khushi Yadav, खुशी लोरा, दोनों खुशी यही है। हाँ तो कौन सी खुशी यादव, खुशी लोरा, भीम सिंह राठौर कहाँ है? सुशांत, महिका, सृष्टि, प्रेरणा, अर्पिता, अंशुल,
Okay, you can leave now. इनकी कहाँ गई है ये? Fifth semester. Third semester की WhatsApp पे है ना मेरे पास. I can identify you. You can leave now. MSc food में रह गया. Absent हो तुम लोग तो. मैडम MSc हाँ ये तो अंदर बैठे थे. इसका पता नहीं है ये दोनों. तुम्हारा नाम क्यों नहीं है? मैम सेमेस्टर की और बता दीजिए. कहाँ है? यही है. अच्छा यही है.
शिखा मैम
Ananya, yes ma'am. Be in fifth semester, please. Rest, please. If it is not called yet, so please wait. Ananya Jain. Please, मुझे देखने दो मेरा व्यू क्लियर करो. अनन्या जैन, परी भदौरिया, आर्या, प्राची, सूजी, भूमिका, आस्था, जलज, अवनी, रचित, सारांश, भव्य दीवान, भविष्य तो थी. आची कशिश, फाल्गुनी, माधव पांडे, निपुण गुप्ता, गवराज, अभिनव.
मैम ये सभी लोग आए थे, ये लोग चले गए लास्ट में, इतने सभी लोग ये वरने तो मैं-
अब्दुल. तो ये जब मुझे मिलेंगे तभी मैं इनकी लगाऊंगी, ठीक है? क्योंकि मैंने इनको देखा नहीं यहाँ पे. जिनको देखा तो उनको लगाती.
एक सेकंड मैं इनको मैं छोड़ देती हूँ कि मैम से एक बार मिल लेंगे.
मैम बीटेक
थर्ड सेमेस्टर की. अच्छा आज इन्फो की है मेरे पास.
मैम वो हम खिलवाए वाले हैं. मैम हमने भेजा था लिख के.
आपने भेजा है?
नहीं आपको दिया है.
इसमें दिया है.
मैम छोटा
तो आया नहीं है.
कभी नहीं आया बीटेक बायोइन्फॉर्मेटिक्स 7 सब कभी नहीं आया.
इन लोगों को भी नहीं आया. तो इस पे लिख दो अपने अपने नाम. यहाँ पे लिख दो अपनी क्लास लिखो. एक साइड में 7 से लिख दो. एक बार ये लिख दो. ठीक है. तुम लोग वहां बैठ के लिख लो बेटा. मैं ना पोडियम खाली कर दूं so that I can begin the oral presentations. Okay बेटा. वहाँ लिखो.
मैम क्या लिखना है?
नाम लिखो अपना. क्लास ऊपर लिख दो और नाम लिखते जाओ. हाँ.
मैम इससे ऐसे एक बार मैं स्टूडेंट हूँ तो मेरा आज फर्स्ट डे है तो मेरे भी एडमिशन लगेगी अटेंडेंस लगेगी.
हाँ बिल्कुल लगी. ठीक है. मुझे बता दीजिए कौन से कोर्स में आप.
मैम बीएससी बायोटेक्नोलॉजी.
तो 2 मिनट बेटा वेट करो. आई विल सॉल्व योर प्रॉब्लम. ओके.
मैम इसको कर
ये अभी हाँ मुझे ही दे दो. इनके फोटो खींच के ग्रुप में डाल देंगे टू मार्क द अटेंडेंस. भविष्य इज प्रेजेंट. अच्छा शी इज
सारे ऑनलाइन है मैम.
ओके ओके. सो
ऑनलाइन ही है.
तो फिर आप कर लीजिए शुरू सेशन.
मेरे को पता नहीं ऑनलाइन होने मुझे पता ही नहीं. आपको पढ़ रहे थे हम तो
नहीं मेरे पास कोई लिस्ट ही नहीं आई है.
ये प्रेजेंटर्स हैं. फर्स्ट
अच्छा इसमें file
प्रेजेंटर.
में ठीक है.
हां फर्स्ट प्रेजेंटर ऑलरेडी लाइन में है.
ओके ओके.
विल स्टार्ट.
तो ये
नहीं 12:30 का टाइम दिया है.
12:30 का टाइम.
रवि दर्शनी फ्रॉम
ये एक बार देख लेते हैं पार्टिसिपेंट्स कितने प्रेजेंट है ऑनलाइन.
रवि दर्शनी.
ये ऑडियो जा रहा है.
कर दिया.
Hello. Participants, I am calling your name for oral presentation. So please give me your attendance by raising your hand if you are present.
Ravi
Ravi Darshini,
Present, ma'am.
are you present?
Yes, ma'am.
Okay, thank you. Next is Nikki John Kamapilli. Nikki John, are you present?
Sorry.
Prachi Balyan.
Yes, ma'am.
Okay. Nikki John, are you present? Manali. Manali, are you present?
Yes, ma'am.
Okay, thank you. Tina. Tina. Tina Patidar. Tina Patidar. Haq Nawaz. Haq Nawaz, are you present?
Yes, ma'am. Yes.
Okay, thank
Yes,
you.
ma'am. Yes, ma'am.
Pooja Ram. Okay, thank you. Chandni.
Present, ma'am.
Okay, thank you. Subhiksha.
Present, ma'am.
Okay, thank you. Naveen Prakash. Naveen Prakash.
Present, ma'am.
Karthikeyan.
Online है सारे.
Karthikeyan.
Karthi present, ma'am.
Okay, thank you.
इसको बोलो मैम.
Kirtana.
Present, ma'am.
Thank you. Jiva Bhati.
Present, ma'am.
Okay.
इसको बोलो मैं
So
first
the sequence is confirmed now. So I will call your name one by one and you have to give your oral presentation. You will be provided 5 minutes for the oral presentation. Okay. So be ready with your presentation. And 2 minutes will be for question answer session.
Ravi, बोल
So first presentation will be of Ravi Darshini. And I would like to introduce the chairperson of the session, Ms. Dr. Pushpen Sharma and Dr. Akhilesh Kumar. So both are present here. I would like to welcome you, sir. So Ravi Darshini is going to present. Ravi Darshini, please share your PPT.
Yeah, ma'am.
जो कॉमन प्रेजेंटर उसमें से एक नहीं प्रेजेंटर सिर्फ एक नहीं इसका नाम भी दो
हाँ, मैम इसके दो टॉपिक हैं.
मैं तो नाम ले ली ऑल आर प्रेजेंट नहीं मेरा नहीं
Hello, Ravi Darshini.
Yeah, ma'am.
Ravi Darshini, can you hear me?
Yes, ma'am.
Share your PPT. There is a link below to share your PPT. Are you able to share now? Share your PPT.
Hello,
Ravi
ma'am.
Darshini, are you connected through Pooja Nandini?
No, ma'am. Ma'am, can you see my screen?
I'm not able to see your screen. I'm not able to see your screen.
Yeah, ma'am, I'm working.
Have you shared the PPT
Yeah,
in the
ma'am.
email?
Yeah, ma'am.
You have shared the PPT. Okay, just give me a second then.
Hello, ma'am. Now you can see my screen.
Yes, your PPT is visible now.
Okay, ma'am.
Can you show the first slide?
Yes, ma'am.
All the participants, please keep your PPT ready, and there is an icon right after the react button. That is, we can see the participants. After the participants, you can see chat button, and after button, there is react and then share. So all of you will have to share from the share button. Okay, so please be ready with your presentations. Open the presentation and then do the sharing once one person is done. And we have shared the order already. Okay. Yes, everyone. Okay. Okay, then you can start. Ravi Darshini, please start and put it in full screen mode. You have strictly 5 minutes and 2 minutes for question answer session. Okay.
Okay, ma'am.
Yes. So start with your full screen. Full screen is yeah, that button. Yes. Go ahead, please.
Good afternoon, everyone. This is Ravidarshini, currently pursuing my MTech Food Technology at Congo Engineering College. So the research topic I have chosen was the studies on the extraction of crude iodine from red seaweed, which was Gelidium amansi, and its potential application in fortification of food. And I was guided by Mr. Arun Joshi, Assistant Professor in Congo Engineering College. The main aim of this project focuses on the extraction and fortification of iodine from the red seaweed Gelidium amansi in the food product to develop a protein iodine-rich food product. The main objective was to select an uh, suitable extracting method for extracting iodine from red seaweed and to fortify those extracted iodine in food and to evaluate the stability and quality parameters of iodine fortified food product. So first of all, a red seaweed. Here I have chosen was Gelidium amansi, which was grown commonly in the region of southern region of India, in Tamil Nadu areas. So it
Ravi
was a
Darshini,
yeah,
excuse me,
ma'am.
Ravi Darshini, your slide is not moving. Please move your slide. It is still sitting in the
Ma'am,
first slide.
ma'am, I have been moving my slide, but
Okay, you
it
haven't
was
moved your slide. You only have 5 minutes. So go ahead, please.
ma'am, no, ma'am, I have been moving my slide, but it was taking longer to.
Okay, now it is moving. So you're in the
Okay.
third slide right now.
Yes, ma'am.
Okay, go ahead.
Yeah, ma'am. So first of all, red seaweed is an rhodophyta, which is a multicellular marine organisms. In this Gelidium amansi, which was also known as Umtuga sari, it is an important species of red algae, commonly harvested in the shallow coast. It is a rich source of antioxidant, which includes polyphenols, phycobilly proteins, and contains proteins, minerals, trace elements like vitamins, and also essential fatty acids. So the requirement of iodine is 150 microgram per day. It can be obtained from a single gram of red algae. It was the most abundant element in red seaweed was iron and iodine. So the iodine amount in the Gelidium amansi remains comparatively high than the other plants found in the nature. So commonly consumed iodine was usually fortified with salt. So however, a group of people was not advised to eat salt because of certain health issues and concerns. So they were high people who have high blood pressure and pregnant women who have high blood pressures and hypertension. So iodine fortification other than salt in food will be useful to meet their daily iodine requirements in order to enrich iodine in their regular diets. So for this, I have chosen three methodologies for this process. First was drying. It was the cleaning of Gelidium amansi, which was obtained from the seashores in Tamil Nadu areas, and then microwave drying of Gelidium amansi, and then grinding into fine powder for further process. And then second method was extraction, which is a main process to obtain iodine from the dried powder of Gelidium amansi. So here I have opted for ultrasound extraction for the efficient extraction of iodine, and also it does not destroys the other components and natural bioactive component present in the Gelidium amansi when comparing with other extraction techniques. So here I have chosen water as a solvent, and extraction was carried out for 15 minutes at 40 degrees Celsius, and then I have obtained a crude iodine extract. Then after extraction process, I have chosen RSM for optimizing the composition for the food product I have taken. Here I have chosen a millet based premix, uh, pancake premix or waffle premix for daily consumptions. So the RSM was used to optimize the ingredients, and then fortification of iodine, which we have extracted, was done, and then quality analysis of the premix to ensure the stability and the quality parameters, and finally the iodine fortified premix will be obtained. So to ensure the quality and stability, several analysis was performed. For example, water activity, moisture, ash, sensory, iodine content, microbial analysis, bulk density. So while comparing with the control sample and with the optimized sample, the water activity rate was 0.54, and the moisture content was 2.97 percentage, and the ash percentage was 2.96 percentage. By using 9-point hedonic scale, sensory analysis was performed, which we have obtained 8.9 points. And for iodine content, the analysis was given for uh, identifying the iodine content. And microbial analysis, total plate count method was used, which showed less than 10 counts. And then bulk density have to be performed, and these are the analysis performed still now in the developing stages. So I would like to acknowledge here. So I would like to thank my guide for his guidance and unwavering
Okay, thank
support.
you, thank you. You can stop now. I have a question to you.
Yes,
Can you tell me, I mean, iodine extracted, what exactly, how it is differing from other iodine sources?
yes, sir. So usually iodine was chemically developed, and it was involved in the food. So here we are obtaining it and extracting it from the natural source, which is a red seaweed, which was usually commonly growing nowadays in the seashores. So this will be as a natural source of iodine, and this can also it was present in higher amount in the red seaweed while comparing with other natural ingredients which have iodine. So there will be a minimal loss while in processing. So it was the main aim is to obtain the natural.
Ravi Darshini, the question is basically, so many iodine is available in the sea, right?
Yes,
So
sir.
extracting it from the seaweeds and then basically, getting it purer form, actually, it's a very, it is going very costly, actually, not like that. It's a costly process in production, right? You are purifying it, extracting it.
Yes, sir. Here I'm while we are going for.
Okay. Thank you so much, Ravi Darshini. All the best. Okay, next pull.
Our next presenter will be Dr. Nikki John. Is she present? Dr. Nikki John, please let us know if you are present.
Yes, sir.
Okay, so please, ma'am, you can start sharing your presentation.
Okay, just a minute.
Yeah, sure.
Is my slide visible?
Yeah, it is visible.
Okay. So my topic is on, good morning, everyone. My topic is on sustainable approach for the use of fruit pomace for the development of a biofilm. So myself, Dr. Nikki John Kannambali, along with Dr. Tangavel, professor from Karuna University. So basically, when we move on to the introduction, our main aim is to monitor the food quality, which is basically a critical role in maintaining the food safety and human health. So as we all know that food freshness is evaluated by the pH values associated with the food. The pH sensing indicators, what we use also gives a distinguishable color change variation. And the main source here, which we're using is biofilm is made up of carrageenan, which is a water-soluble sulfated polysaccharide that is extracted from red edible seaweed. And it also has a strong rigid gel biocompatible material, and it is also non-toxic. It is stable and again, it is very stable compared to all other, compared to the biofilm when we look into a wide range of pH. So here again, we have incorporated anthocyanin, which is extracted from great pomace, as well as extracts from the juice what we have obtained. We have extracted the anthocyanin from it. And then the main, as we all know, the component present in anthocyanin is cyanidin-3 glucoside, which is present as anthocyanin pigment, which is responsible for the color change, basically from acid, neutral, as well as the premium change. So the pH change in the color basically associated with pink, varying from bluish green to greenish yellow. And the major objectives of this research is, first is extraction of anthocyanin from great pomace. Secondly is development and evaluation of the properties of this anthocyanin-based film. And third one is application of the film in the monitoring the food spoilage. Hope the sites are visible and it is moving. Yes. So the first process is extraction and characterization of great pomace anthocyanin. So here we have taken the great pomace, which is dried pomace powder. We have extracted it using ethanol, and then we have concentrated using a rotary vacuum evaporator. We have obtained the grape anthocyanin. We have checked into the UV spectrum of the anthocyanin, as well as we have looked into the quantification of anthocyanin. So by checking into the visible spectrum, we could understand that there is a reddish peak to pink to greenish yellow, which was observed in this pH range of varying from 2 to 11. And as you can see from the picture, that is a graph itself, you can understand that around the range of 525 nm nanometer, in the range of pH 2 to 7, there was a maximum absorption, which again shows the presence of flavium cation. And similarly, there is a peak around 618 nm, which again increased with the pH range when the pH increased from 6 to 11, which shows the formation of the carbonyl pseudobase. So here in the picture, you can see the color variation of anthocyanin extract, basically the grape anthocyanin extract, when we subject it to various pH change. So from 2 to pH 2 to pH 11, you can see the color change from pink to purple, then towards the greenish yellow side. We have also checked into the HPLC as well as FTIR, which had shown that when HPLC was performed, which shows the presence of cyanidin-3 glucoside and cyanidin-3 rutinoside, which again proved the presence of the anthocyanin component present in the particular functional group. And when you studied about the functional group, these are the basic components with the P, which shows the stretching vibrations of glycosidic linkages. We have carbon-carbon stretching vibrations, as well as we have OH stretching and the vibrations. References are also given. These are the basic components of a natural biopolymer film. So basically for the preparation of biopolymer film, we require a film forming material, a functional ingredient, as well as an additive. The film forming material which we have used is carrageenan. Functional ingredient is anthocyanin, and an additive which is used is glycerol. So these are all the properties of the various materials which we have used. Carrageenan I've already explained. Glycerol is a common plasticizer which is being used, and it is also hydroxyl group which is present and responsible for the intermolecular interactions. So that is a major reason why we have taken glycerol, and it is one of the best plasticizer which syncs with all the film forming material. This is a basic method of preparation of development of the film. So here you can see the control film as well as a film with incorporation with the anthocyanin. So here in the control film, we have incorporated carrageenan powder, glycerol, as well as ascorbic acid in order to maintain or neutralize the pH. And we have formed a control film, and the other one is a anthocyanin incorporated. So here we have varied the concentration of anthocyanin and checked which is appropriate. This is the anthocyanin response of the anthocyanin film if when it is subjected to pH change. So here you can see the film also showed a change in the color when there is a change in the pH. And this is a color value also, the LAB values, which showed in the difference. This is the characterization of the carrageenan anthocyanin film by FTIR. So we have done both carrageenan as well as the anthocyanin film, and these were the particular functional groups which were present. And we have also done on the moisture content, tensile strength, elongation of
Can you
break.
please move fast because time is,
Okay.
5 minutes only.
We have also proceeded with that, and we have done the scanning SEM analysis. So SEM analysis, you can understand clearly that what is the dope material which is being present. And here you can clearly find the difference in
Conclusion. Can you please come to the conclusion?
Okay, so this is a conclusion where in which we have checked into the food spoilage for the milk, where in which it showed depending upon the change in the characteristics of the milk, the pH also changes, acidity changes, which again showed a color change in the film, which indicates whether the milk is spoiled or not. How is the quality of the milk? The same thing we have tested in the milk booth or the society, which again showed that purple color indicated the milk sample was fresh, whereas a pink color indicated that it was slightly spoiled.
Okay, my question is to you. Did this particular film develop is basically looking for only for the milk or multiple foods material?
It can be done for multiple foods. I've done the study only for milk, one particular food.
So only the basically you're talking with respect to the pH, the microbial system that might be growing and causing the food spoilage, right?
Yes. This is just a detector or an indicator to show food spoilage. Immediate, a rapid detection. Microbial spoilage can be seen, but that again takes days or time to know the result. But this is a quick indication.
Okay, right.
Dr. Nikki, there's another question. So can you tell us what is the stability of the product? Like
you can see the last type, the stability of the film. So here, which was found as refrigerated condition in the dark, the film was stable for 21 days. But when we kept in the room temperature and in the light, the stability was less. The color of the film again changed.
All right. So, thank you so much. Thank you so much, Dr. Nikki. We will move on to our next presenter.
Thank you.
Prachi?
Yes, sir.
Yeah. Can you please start your presentation?
Yes, sir. Yes. The other one is presenting, so I can't.
Yeah. Dr. Nikki, could you please close your presentation?
Yeah, sure.
Sharing your slides. Yeah. Prachi, please go on.
Yes. Is it visible?
Yeah, it is visible. Please make it full screen.
Yes. Good afternoon, everyone. Myself, Prachi Balian, research scholar from Vanasthali Vidyapith. My title of presentation is Phytochemical Analysis and Antioxidant Potential of Nyctanthus arbotristis, which is a wild and underutilized plant. Background, plants have been used by mankind since antiquity as foods, medicines, and industrial raw materials. So many phytochemical studies are done on many plants. So these phytochemical compounds play an important role in the adaptation of plants. So these plants can be recognized as a great source of natural antioxidants, such as plant polyphenolics like flavonoids, tannins, phenols, etc., which possess free radical scavenging properties. Nyctanthus arbotristis, which is popularly known as Parijat or night jasmine, belongs to the family Oleaceae. This plant is also called as tree of sadness. It has also been credited with many therapeutic properties like antiviral, anti-leishmanial, antifungal. The water-soluble portion of alcoholic extract also shows anti-inflammatory activity. So the present study comprehensively investigates antioxidant activity, total phenolic and flavonoid content of successful extract of leaves of Nyctanthus. Material method, chemical reagents from high media sigma SRL, and the plants were collected from Vanasthali Vidyapith. Extract preparation, plants are thoroughly washed with distilled water and dried at room temperature. The extraction was done by sausalite process. Qualitative phytochemical analysis was also done to detect the presence of alkaloid, steroid, carbohydrate, etc. Quantitative analysis was done by TPC, TFC, and antioxidant acid. TPC will be determined by Collins or calcium method. Total flavonoid content was determined by aluminum chloride colorimetric method. Antioxidant assay was determined by using 3 different assays, DPPH, NOSA, and metal chelating assays. Statistical analysis also done. So here are the results on, of qualitative analysis. As we can see, the methanolic extract of this plant shows in comparison to the chloroform and distilled water, shows major amount of carbohydrate, alkaloids, phenolics, and flavonoid contents. The quantitative analysis in the graph of TPC and TFC, we can see the methanolic extract shows a high amount of phenols and flavonoids. And in the antioxidant activity, which was performed by 3 assays, DPPH, NOSA, and metal chelating, IC50 value of methanol in 3 of the assays are low. As we know, the lower the IC50 value, the higher the antioxidant activity. So we can say the methanolic extract of these plants in comparison to the chloroform and water shows high antioxidant activity, which may be also due to the presence of high phenolic and flavonoid content. So here are the discussions. Nyctanthus contains carbohydrates, alkaloids, phenolic compounds, flavonoids, etc. The leaf extract shows high phenolics and flavonoid content, and we concluded that Nyctanthus is rich in antioxidant compounds, boosting their usage as a source for the creation of food supplements with antioxidant qualities or as food additives to increase the antioxidant content of food products. So we can say it is a promising source of bioactive chemicals that we can be used treat a variety of ailments and make lucrative nutritious drugs. So here are the acknowledgements. Would like to express their gratitude to Professor Inaditya Shastri, Vice Chancellor, Vanasthali Vidyapith, and also funding agency. Thank you. Hello. Yes, sir.
Prachi,
Yes, sir.
do you have any animal system for testing the antioxidant activity of these compounds?
No, sir. I have done only on the plants.
So are you planning for something like testing the antioxidant activity in animal system as well?
No, sir. Actually, I have work only on plants, not animals. I'm from the plant science field, so not on. Not
So
from
what's your long-term objective of this study?
Sir, my long-term objective of this study will be to include this plant as we can say that this is an underutilized plant. So in marketing strategy, so that it can be known to many peoples, because in traditional or tribals, people use this plant as a food, which has a very nutritious properties. So for that.
Okay, fine. Thank you. All the best.
So Prachi, you can stop presenting.
Stop kar diya.
Yeah, please. So our next presenter will be Dr. Nikki John again from Amal Jyoti College, and she will be presenting on plant-based meat alternatives. So over to you, ma'am. Are you present?
Sir, my other co-author will be have the same paper presenting in the afternoon session.
All right.
Okay, thank you.
So let me call Miss Manali from Andan University. Yeah. Could you start presenting the PPT, please?
Yes, sir.
Yeah, it's visible. Manali, please make it full screen.
Okay, sir. Sir, is this visible?
Yeah, it is visible. Can you please make it full screen? There's a scroll button at the bottom. Yeah.
Sir, is this okay?
It is not full screen right now. So can you switch between the slides? So I think there's a little internet lag here. Okay, you can present in this mode only. Okay, please.
Okay, sir. Okay, sir. So my project title is Development of Dietary Fiber Induced Milk-Based Drink. And why we have to take dietary fiber for to take the optimal body function
Please
and
be a
foods
little
that
loud,
are
Manali. You are not clearly audible here, so can be a little
okay,
louder.
sir. Sir, foods that have less amount of dietary fiber can have adverse effects on our gut health and weight gain and cholesterol levels also. So, dietary fiber mean it is in plant-derived carbohydrates and are not digested within our gut, and it can be classified into two, which is based on its solubility, which is an soluble dietary fiber and insoluble dietary fiber. And in India, the dietary fiber intake is very low, and the DRA is between 90 to 38 gram per day. It can be vary among age and sex. Despite this recommendation, fiber intake in most of the countries and especially in India is very low. So I took diet fiber as an main theme of my project. And according to FSSAI standards of dietary fiber in India, the dietary fiber source product contains at least 3 gram of fiber per 100 gram for solids, and for liquids it is about 1.5 gram. And the main aim of my project is to develop a nutritious and functional beverage by enriching a milk-based drink using dietary fiber. And the objective is to formulate and develop a milk-based drink enriched with dietary fiber, and to examine the suitability of stabilizers to maintain its cloud stability, and to study the physiochemical and sensory and stability of the dietary fiber enriched milk-based drink. And the raw materials I have used are among three, which is first one is milk, which I have prepared in local market, and then satin husk, which is an underutilized plant, which is an Indian herb, and it is commonly found in India. It works well. It works well in weight loss, diarrhea, and importantly in gut health. Use of satin husk in our diet for about 3 weeks may lower blood cholesterol levels. And the stabilizer I choose Capacaraginin, which is a natural hydrocolloid which is obtained from red seaweed, and it is an off-white color and neutral odor and taste. It is an underutilized stabilizer for milk-based drinks, which is especially about for dietary fiber enriched milk-based drink. So I took this Capacaraginin, and the methodology is, first, we have to prehydrate the psyllium husk and for make it more soluble with the milk. So the prehydration must be with a ratio of 1:15, and we have to preheat the milk at 40 to 50 degrees Celsius, and we have to mix the prehydrated psyllium husk and stabilizer in the preheated milk, and we have to homogenize the milk. I have done two-stage homogenization at 1500 to 2000 psi, and we have to done pasteurization to kill the microbes, and I have done at 80 degrees Celsius for about 5 minutes, and we have to store the milk for about at 4 degrees Celsius. These are the trials I have made. And the fifth trial, which is about 20 gram of silent husk and 0.3 gram of capacaraginin, has came out very well, which is uh, the responses are the pH is good and the titratable acidity also good, and the viscosity is about 18.6 at 30 rpm, and then overall acceptability is also 8. And I have um, and I have and the and in results and discussions I have made for stability analysis for the stabilizer, and the cloud stability is verified for verified under turbidity process using centrifugation method, and the optimized product I have taken for 5 days, and then throughout the 5 days I have taken pH titrability and then cloud stability and viscosity, and it is good. And the conclusion is for capacaraginin is more effective in increasing viscosity and might contribute to more to acidification also. And then cloud stability is also good. We can say it by when the turbidity low, the cloud stability is good for beverage products. And the viscosity is higher because it may be due to the silent husk, because it has the gel forming capacity and water absorbing capacity also. And we all know that the capacaraginin also have thickening effects, so it may lead to increase the viscosity when we store for about long days. And the acknowledgement is I would like to thank Dr. R. Bhaskar for his guidance and support, and Kongu Engineering College for providing necessary resources, facilities, and engagement. I would also like to thank Amity University to giving me this great opportunity. Thank you, sir.
Thank you, Manali. I have a question. Does the flavor of milk change after addition of these
No, sir.
additives?
No, sir.
So is it
No,
exactly
sir.
same?
Saline has no flavor. Yes, sir.
Okay. And the viscosity also, although you have shown in your result, does it increase too much?
So viscosity is
Like, does it increase too much? So that
so
it becomes
viscosity is
completely different from the
slightly changed.
slightly.
Slightly changed due to the gel forming capacity of the silent husk, sir.
All
It
right.
has water absorbing capacity, so the viscosity slightly changed.
Okay. Okay, Manali.
Okay, sir.
You can
Okay, sir.
stop the sharing. So the next presenter is Tina Patidar. Are you present with us? Miss Tina? Okay. If she is not present, I think we can move on, and next presenter will be Mr. Haq Nawaz. Are you with us?
Yes, yes. Am I
Can
audible, sir?
yeah, you are audible. Can you please share the screen?
Just give me a second, sir.
Please start the presentation.
Is it visible, sir? No?
Yeah, it is visible. Please make it full screen.
Okay.
Yeah, it is
Is
visible.
it now full screen, sir?
Yeah, it is visible. Please start.
Okay, okay, sir. So, okay. Good afternoon, everyone. My name is Hafiz Rafiq, and I'm right now pursuing my PhD uh, under the supervision of Dr. Mir Hashim Rasool, and in the Department of Physics, Islamic University of Science and Technology. Uh, so, uh, the title of the presentation is Rare Earth Activated Inorganic Phosphor Materials. So basically, we are working on development of the phosphor materials for their different applications like LEDs and like displays. So I have chosen to synthesize them, characterize them, and then check their photoresistance properties for different applications. And in this paper, I have chosen explicitly for solid state lighting applications. So the background of the presentation would be that I would be using certain terminology in this presentation. So I would like to acquaint you people regarding the term. So there is a term luminescence, which is simply means spontaneous emission of light by the substance when excited by some source. And there are so many types of the luminescences that depends upon the excitation source, because the excitation source could be X-rays, it could be electrons, it could be light, and it could be neutrons. It could be there are so many types of the actually luminescences. So I have chosen here 5-6 types. And the photoluminescence is the basic thing we are working on, because we are using the light as a source for the photoluminescence or for the luminescence purposes. And the photoluminescence is basically divided into 2 types. One is the fluorescence and another is the phosphorescence. Fluorescence is simply the instantaneous emission when there is the excitation source, and there is the instant emission. And when the excitation source is removed, then the emission stops. But there is another term which is called the phosphorescence, which is actually, in simple terminology, we call it a delayed emission, which says that even if you remove the excitation source, the emission persists. And we're actually working on the phosphorescence. So there is an important term which is the phosphor, which is simply any inorganic material. Luminescence can be shown by different types of the materials, inorganic materials and organic materials. But phosphor term is explicitly used for inorganic materials, which exhibit as luminescence, and it consists of a basic host and an activator. And then the background would be that there are so many actually phosphor materials, and there are different formulas for them. But we are working on a specific formula, which is ABP, alkaline metal, alkali metal. Sorry, there is a correction. Alkali metal, and B is an alkaline metal. Having 2 valency, and the first one is the monovalent. And there is a PO4, which is called the phosphate group. And there ADPO4 family have been prepared in the literature, like lithium strontium phosphate and doped with the europium, as I already mentioned. Am I your internet is unstable? Am I audible, sir? Hello. Hello.
Yeah, you are audible. Please continue.
Sorry. Okay. So lithium strontium phosphate was developed, and sodium calcium phosphate was developed, and lithium zinc phosphate was developed, but it was doped with the manganese. So I have checked their application when I doped with the samarium. The basic criteria or basic fundamental things that in the past we were using to develop the LEDs was we had a gallium indium nitrate blue LED chip, and we put a phosphor on that phosphor material, and it was a way to prepare the LED chips. But then the recent development is we use a UV LED chip as a source, and then we use 3 different phosphor materials on the top of it, and it gives a white light, because red, blue, and green combine together to give a white light. So in case of or compound lithium zinc phosphate doped with the samarium, it shows actually orange red emission. So we can use that orange red phosphor material in place of this red, even though there are materials, but we're just trying to explore its applications. And for this preparation, we have included lithium zinc phosphate doped with samarium, and because of their large band gap, and one of the band gap is one of the criteria for the development of the LED LED bulbs. And because of its high physical stability, we have experimentally detected that its stability, because it should be stable at very high temperatures, and lithium zinc phosphate is very stable at high temperatures. And because of its ease of preparation, the preparation was not that tough. Even though the preparation was a lengthy process, but wasn't tough. There are simply steps. We followed the steps. We mixed the ingredients. The ingredients I have shown here, and then we heated at 1100 degrees Celsius for 24 hours. And basically we were able to develop this. And then XRD we have done, and XRD confirmed that the material has been synthesized, and we have done the retail refinement analysis, having a chi-square approximately 2, and we have done the SEM analysis, showing all the elements present equally, or we can show in a uniform basis. And this samarium has been shown, the distribution of samarium has been shown, which shows there is a smooth and uniform distribution, which is quite a good sign for the research. And why the rare earth or the samarium has been chosen? Because of the FF transitions. FF transitions are shown by the rare earth elements because they have electron in the F shell and S subshell, and when the electrons are there in the S subshell, there happens to be a FF transition. They are famously called as the FF transitions. They have been proven by the dike. You can see the samarium having different electronic energy levels, even though there is only one electron present in the samarium 3 positive, but still there are different energy levels because of the FF transitions, and we can prove these transitions through LS coupling incorporation. And we have done the excitation, even though we have done so many analysis, but for the this presentation point of view, I have taken photolysis excitation spectrum. So when we excite this material on 402 nanometer excitation, so there happens to be many FF transitions. And these FF transitions belongs to samarium. And after that, we have done the emission spectrum analysis or photolysis emission spectra. This was very important, and it shows 3 different transition lines: 564, which shows greens, 602 nanometer, which is orange, and 650 red. And when these 3 emissions combine together, they give the result as you can see on the right here on 500, almost in the orange red region. And these were the transitions involved I showed here. And since it is showing orange red emission, so we can use it here as a red component while developing the UV LEDs. So thank you so much.
Please speed up, and you have to finish within the time. Please come to the conclusion.
So I stop it. Thank you so much. Thank you.
Yeah, you can come to the conclusion.
So okay. So conclusion is I calculated so many things regarding this compound, like CI coordinators, how concentration changes the coordinators of the emission, how the temperature behavior of this material was, and color purity, how much is the color pure of this compound. So I have developed all these, I have calculated all these things, and this paper has been already published. And I would like to thank you, IUST, for the financial support and also the research facility provided by National Institute of Srinagar. And thank you.
Okay, so can you go to the slide where you have shown the this phosphor, right? Go back. Yes. So yes, next one. The next one slide, please. Yes. So what is the use of this? The new scenario you're talking. I mean, what is the main? How it is going to be different from the rest previous one?
Okay, so the reason for developing this material was that there has not been that much of work on the lithium zinc phosphate, and since I was new to the PhD, so I chose this material because of its ease of synthesis. That was the first motivation, and I've heard and I've gone through the literature, and it was showing physical ability. So I checked this is
your application. Listen to me, please. First, your application will be focused on luminescence, right?
Exactly, luminescence, white LED luminescence.
Okay, so yeah, that's what my question is actually, how it is different from the, because already so many materials are available for the luminescent studies.
Yeah, like quantum dots
Yes,
these
yes.
days, they are quite cheap. They are, carbon quantum dots. They are not polluting for the environment also. So how can you justify like using the rare earth?
Obviously, there are so many.
Yeah. Hello.
Hello.
Yeah.
Okay, so obviously there are so many methods, because there are so many materials that being getting developed for the LED application. But since phosphor is the basic thing that modern new technology is actually based on. I already showed in this. This is the new scenario of creating the white light using different phosphor materials on the top of the LED chip.
Dr. Vijay, student over. Are you a student of Dr. Vijay? Dr. Vijay in NIT, Srinagar.
No, actually, Doctor. No, he's one of our colleague, but I'm not his student.
Okay.
But I have.
Okay.
He has been guiding us as well. His PhD student is Dr. Irfan. We are working with each other.
All right, Acknowledge. You can log out, please. So,
Now.
yeah. Thank you.
Okay.
And
Thank you.
next presenter is Miss Pujala from Anna University.
Yes, sir.
Yeah. Miss Pujala, can you please start sharing your slides?
Yes, sir.
Miss Pujala, please share your slides.
I have, sir.
Yeah, please. Yeah, you can start presenting.
Myself Pujara from Kong Engineering College.
Can you be a little louder, please? You're not audible clearly.
My topic is on the development of the buckwheat string hopper enriched with the calcium. The main introduction is to enrich the string hopper with calcium represents the target approach, which enhances the nutritional profile than the traditional products. This food with calcium, which main aims to the deficiency of the calcium intake, which is inadequate. Then this combines the traditional food ways with healthy eating to nutritional food that are familiar and appealing to the consumers, improving the public health. The main objective is to optimize the formulation of the buckwheat string hopper enriched with calcium. Focusing is to find the best combination that maximize the calcium properties and maintains the acceptability of the sensory. Then the methods, I have using the buckwheat flour, red rice, sesame seed flour, pepper and salt. These are the raw materials or make into the dough, and it is squeezed into an string hopper. It is steamed for 10 to 15 minutes and packed in the vacuum packaging. The multiple trials has been conducted to optimize the sensory and the calcium. These are the trials I have carried out. There are 13 trials with the variation of the buckwheat flour, red rice flour, sesame seed flour. Then salt and pepper is at same composition for all the trials. Then trial 2, which has been carried out for only buckwheat flour and the red rice flour. Then sesame seed flour, salt and pepper are in same composition. The trial summary is trial 1, 4 and 7 has the good flexible strands and softer in texture. Trial 7 has good mouthfeel and overall acceptability. Trial 2, 3, 5, 6 has not found that string hopper has a good texture and it is finer and easily broken. It is not also an appealing appearance and clumped together. Overall, trial 8 has the best flexible strands and good in texture. Then trial 9, 10, 11 has good mouthfeel and texture. Trial 12 breaks easily, not good mouthfeel. Then trial 13 strands are not good in texture. Then these are the analysis has been carried out. I have carried out sensory, then color, flame photometry for calcium, then texture analysis. Result, it has increased the calcium content compared to the traditional string hopper up to 21% of the RDA. Then sensory acceptability quality of the color and texture has enriched the ingredients. Then compared to the traditional sensory properties, it has been maintained for the buckwheat string hopper also. It is in product optimized composition. Then discussion. The enriched string hopper has a promising alternative for consumer improved calcium intake. It has multiple non-traditional flours. The product maintains the flavor, texture and appearance than the conventional string hoppers. It offers the both health and the sensory satisfaction. I thank my college and the guide for the support and the guidance.
Yeah, thank you, Ms. Pujara. And what is the common name for this tissue? What is the common name?
String hopper.
Yeah, for string hopper. What do you call it in the local language?
Idiyappam.
Okay. So yeah, I think very insightful work. And
Thin noodles. It looks like a thin noodles, which is thin.
all right. Okay. So you could have put a picture of the dish just for the audience to relate it. Okay, thank you so much.
Thanks.
So our next presenter would be Ms. Chandni.
Yes, sir.
Yeah, Ms. Chandni, can you please start your presentation?
Yeah, sir. Wait a minute.
Please make it full screen.
Is it visible, sir?
Yeah, it is visible. Please make it full screen.
Okay.
Please continue.
Good afternoon, all. This is Chandini from Food Technology, Congo Engineering College, and it is guided by Shri Mohan ma'am, Assistant Professor. My work is on the development of ready-to-cook sweet dumplings enriched with protein and fiber by utilizing quinoa. The main aim of the project is to prepare ready-to-cook sweet dumplings that is enriched with protein and fiber, which is suitable for all age groups. The objective is to develop the ready-to-cook product, to optimize the composition of the dumplings, to evaluate its physicochemical properties, textural and sensory analysis of sweet dumplings. The main scope of the work is to provide a healthy alternative and for fried and junk foods, to demands the convenience and innovation. It is naturally enriched product and less gluten. So also by adding pumpkin seeds, it is rich in protein and antioxidants. Coming to ready to cook products, it acquires large food market, and it is basically convenient for everyone to use. Then quinoa, it is called as Chinopodium quinoa. In Tamil, we'll say Seemai, which is a major source for the dumpling, and it is a gluten-free alternative, high source of protein, energy rich, and it is also called as superfoods. Then coming to pumpkin seeds, it improves heart health. As we all know, it is very good for blood pressure, etc. And then palm jaggery, it is added for sweetness, and it strengthens immune system, improves digestion. So it is prepared by steaming, and it gives a quick and easy meal solutions. This is the recommended daily allowance for protein and fiber according to FSSI. For 10% RDA of 100 grams of solid, and for fiber it is 3 grams. So this is the methodology. The quinoa seeds and pumpkin seeds were roasted at 60 degrees Celsius and grinded and powdered. Then a pinch of salt, cinnamon, and were added with the jaggery syrup, and it is kneaded. Then appropriate using molding will make it into appropriate shapes. And it is packed in pouches, and it is packed in Ziploc pouches. It is kept at freezing for minus 18 degrees Celsius for 24 hours, and then it is marketed retailly. Coming to trials, I have taken 9 trials. From that, 25% of all the components were used, and it shows a good overall acceptability. So from the trials, when quinoa and pumpkin seed flow were higher, it gives harder texture, and when it is low, it gives oily texture. So trial 9 was selected, and this is the final optimized composition. I have kept salt, cardamom, cinnamon as constant. So these are the trials I've made. So for the above optimized composition, the moisture content was water activity, protein, fiber, cooking time, hardness using texture analyzer, and sensory analysis were carried out. From this, higher protein and fiber content was shown for the optimized sample. And then from the conclusion, it gives a overall balanced nutrient profile, and it maintains the traditional taste and texture for the dumplings. Also, ready to cook is nutritional superior, convenient, and product which goes with the modern dietary trends. So final optimized product gets a score of 8.5. Yes, sir. So I thank my guide, Ms. Mohan ma'am, and the head of the department, Dr. R. Bhaskar, sir. Thank you.
Thank you, Chandani, for the presentation. My question to you is basically the protein content you're talking. Just go through the slide, please go back. Yes. What do you mean by control and dumpling sample here? And how did you?
I couldn't get.
How did you?
I couldn't get you.
Yes. How did you got the protein enhanced in the dumpling sample? Did you add that pumpkin, right?
Yeah, so pumpkin seed were added, and quinoa flour were added. Quinoa 4 is a pseudo cereal, and for protein estimation, I have used gel dal method.
Okay, so and the control was not containing the pumpkin seed, right?
No, sir.
Okay. I mean, did you basically got the, in what percentage? If you talk like, that in what gram out of the total gram, like
100 grams we have calculated, sir.
so very heavy, okay, content. What about the digestion? Because now you are playing with the natural food, right? No? Did you got some information about digestion system?
Yes, sir.
Digestion, the people eating this particular, the food, for example, modified food. What about the digestion? Did you got some data into that also?
Still further research has to be done in this area. So
Because digestion is a concern, right? When you are supplying the food with so many, high content of protein, the digestion issue may be there, right? So is it?
okay, sir, it is mainly suitable for young peoples and fitness freak peoples, and for them,
Right.
protein and fiber may contribute a lot for their health benefits.
Right. All right, Chandni, you can please log out.
Yeah, sir. Thank you, sir.
So next presenter is Miss Shubhiksha.
Yes, sir.
Yeah, please start sharing your slide.
Okay, sir.
Yeah, it's visible. Please start. Miss Shubhiksha, we are not able to hear you.
Sir, am I audible?
Yeah, now you are audible. Yeah, please start.
My topic is formulation and optimization of herbal pastry. Myself, Subiksha, I'm PG scholar from Engineering College. Pastries, it is a soft confectionery product, are widely acceptable for their texture, shape, and taste, and can effectively deliver the natural antioxidant from plant sources and, by maximizing the health benefits. It is a small solid preparation made from the sugar substitutes mixed with the other active ingredients such as herbs. The diabetic friendly qualities of the pastries are preserved by the use of erythritol and xylitol, as they are low calorie and zero calorie sweeteners without increasing the blood sugar levels. Herbal pastry combining raw turmeric, ivy gourd leaf, and Indian borage extract promotes the oral healing and with anti-inflammatory, antibacterial, and immune healing properties. The objective of the study to optimize the formulation of herbal pastry and to find the best combination which has maximum antioxidant properties, polyphenol content, and which also maintains good sensory characteristics. Next method, the pastries are prepared. Puree is prepared by washing, sorting, steam blanching, and grinding the turmeric, ivy gourd leaves, and Indian borage leaves to create the purees. Then mixing of ingredients by heating xylitol and erythritol and water to 75 degrees Celsius, and then addition of citric acid, gum arabic, and the puree is added at the last stage. Then gelatin is added, and also corn flour is added. Then it is kept in the starch mold and kept in dehydrator for 16 hours. For about 46 degrees Celsius, multiple trials were conducted to optimize the sensory attributes, and also antioxidant activity and polyphenol content were estimated. These are the preliminary trials carried out, in which the puree and water content is varied, and all other ingredients are kept as same for all the 5 trials. In trial summary, the trial 1 has lower polyphenol content as it has lower extract values, and the antioxidant activity was also less, but the texture was good. In trial 3, it has optimum polyphenol content and has moderate overall acceptability. Trial 5 is optimized product as it has higher polyphenol content and antioxidant activity with a good sensory score.
These are the analysis that I have carried out, that the sensory, then followed by color analysis, then texture analysis, then antioxidant activity, and also polyphenol content has been estimated. These are the results. The results show that there is a significant interaction between the extract and the water, influencing both the antioxidant activity and the sensory properties of the pastilles. Antioxidant activity with a higher extract content leads to greater antioxidant activity. However, it decreases with the increase in water content. The sensory scores were improved with increased extract levels, but there is a slight decline at very high extract levels. This is the optimized. This is the combination of the optimized product. That is the trial 5. Then discussion. The optimized herbal plastically shows significant improvement in both functionality and sensory appeal. Higher antioxidant activity contributing to its potential health benefits, particularly in oral care. It can heal the oral ulcers, that is mouth ulcers. The product texture is optimized with high hardness and reduced chewiness. It provides more consistency. The use of xylitol and erythritol as sweeteners ensures that the pastilles is suitable for people who are having diabetes. It without raising the blood sugar levels. The combination of the raw turmeric, ivy gourd leaves, and Indian borage leaves in the pastille provides a strong inflammatory and wound healing properties. We extend a sincere thanks to Kongu Engineering College and my guide for the support and guidance.
Thank you, Saviksha. So, what was the sample size for the sensory analysis? Right? You did the sensory analysis. So,
yes,
what is the sample size? Do you understand my question? How many persons participated in the sensory?
Around 25 members.
How many? Sorry? Can you
25.
repeat? 25. So, like, is that considered sufficient? Or any comments about this? Should we have more participants, or the sample size should be larger?
Sir, I can't understand what you're
So,
asking.
what I asked is, how do you determine that 25 number of persons participating in the sensory analysis?
Sir, I have I have used a sensory sheets with attributes, sir. In that attributes, I have asked certain questions, and the panel members, based on their answers, I have calculated the overall acceptability of the samples. Sir.
Okay. Thank you, Sabiksha. So,
thank you, sir.
Please log on. So, our next presenter is Mr. Naveen Prakash.
Yes, sir, I'm ready.
Can you hear us?
Yes, sir. Did you hear me?
Yeah. Please talk a little bit louder. You are not audible clearly.
Sir, I am audible, sir.
Yeah. Please keep the microphone close to your mouth.
Yes, sir. Yes, sir.
Yeah. Please start sharing your slides.
Sir, is this PPT is visible, sir?
Yeah, it is visible. Please make it. Yeah, you can start presenting.
Yes. My study and perform the analysis of palm oil using Hunter Color Lab and develop the chart for oil quality. My project aim is to develop chart for color analysis and chemical analysis in palm oil to perform color analysis methodology of fresh palm oil to maximum reused palm oil using Hunter Color Lab equipment to perform the chemical test for fresh palm oil to each frying cycle to create a color chart that correlates color parameters with oil chemical analysis like saponification, iodine, peroxide, and acid value. Project scope: develop a color chart that visually represents oil quality based on the established correlation. Introduction: oil frying is one of the most popular habit in all the industries and also in houses and also various fast food centers. The major mistake in that area was reusing oil for a number of hours. The reusing cooking oil leads to formation of polycyclic aromatic compounds, which leads to heart disease and secretion of bad cholesterol and leading to cardiac arrest in some cases. This was the methodology. First, deep frying of tapioca chips at 190 degrees Celsius palm oil. Color analysis. After frying, oil sample was taken and it was taken color analysis using Hunter Color Lab. After taking color analysis, frequently chemical analysis like saponification, iodine, peroxide, and acid number was carried out. Saponification number, it measures the presence of triglycerides, which are the common signs of degradation. Iodine number measures of degree of unsaturation in oil. Acid number, it represents the amount of hydroxide to neutralize 1 gram of sample, which contribute to rancidity. Peroxide, it is the common indicator of oxidation. Mainly peroxide was formed due to oxidative rancidity while frying, and this was the chart was prepared. I will take 6 frying samples. According to that, simultaneously for each frying, I take a color analysis using Hunter Color Lab and also the chemical test for each frying cycle. According to the analysis, it has been showed that according to the frying, the chemical values, peroxide, acid, saponification are kept in increasing. By using this chart, it can used for an auditing. If the food safety auditor was gone for an audit, means this chart will helps. Normally, the oil sample was given to the food testing laboratory. It should take approximately 3 to 4 days for the result. By using this chart, we can take the oil sample and get the color from Hunter Color Lab. Using this chart, according to the color reading, we can get the chemical analysis results like saponification, iodine, acid, peroxide. These are the ranges, and and also the FSI specification of palm oil was saponification number 195 to 205, iodine value 54 to 62, and acid number not more than 6, peroxide value not more than 50 milli equivalent per gram. Per kilogram, sorry.
You stop your presentation, please. I can have one question. Go back, please.
Yes, sir.
In the previous slide, please. Yes. All basically your graphs are similar in appearance in all cases, like saponification, iodine values, and peroxide and acid. I mean, even the y-axis and x-axis, right? Check it. They are running at the same value. If you look in, so how did it happen? Because all methods are different actually. If you see, peroxide is enzymatic method, saponification obviously is iodine based. So how did you got this clear, the same kind of graph in all these tests?
Sir, this bar graph will easily understand for all cases. So this type of design was used, sir.
This is not possible at all. Same variety of experiment, the same data, it's not possible at all.
Okay. Okay, sorry, we are running a little bit short on time. Please come to the conclusion. No, resulting conclusion.
Sir, discussion. The changes in parameter measured expected degradation process occurred during frying. Monitoring these parameters can help determine the oil should be replaced to maintain the food quality and safety. These are the result and discussion, sir.
All right. So thank you, Naveen. Can you please log out?
Okay, sir. Thank you.
The next presenter is Mr. Karthikeyan. Karthikeyan R.
Yes, sir.
Yeah, please start sharing the presentation. Yeah, please make it full screen and start presenting and try to stick to the time.
Yes, sir. Topic is development of nutritional bar using lemon peel powder and rice bran powder. Introduction. This project is based on the development of nutritional bar using lemon peel powder and rice bran powder as the key ingredients. Normally, both lemon peel and the rice bran powder are the frequently discarded product, like lemon peel are the frequently discarded and the byproducts, and the rice bran also the byproduct of rice. These both lemon peels and the rice bran powder are rich in dietary fiber, micronutrients, bioactive components, and this project aims to develop a nutritional bar using these two as the key ingredients. Objectives to formulate a nutritional bar using lemon peel powder and rice bran powder, and to optimize the nutritional bar, and to assess the sensory acceptability, and to determine the nutritional profile of the final product. Scope, byproduct selection and acquisition, and potential byproducts, potential functionalities include, and developing methods to convert the byproduct into a food product. This literature review is based on the both byproducts, lemon peel and rice bran, and its functional properties, nutraceutical properties, and followed by the rice bran. It also tells about the rice bran stabilization process, which is helpful for the human consumption. And this is a literature summary. Literature summary tells about the unique properties of the lemon peel and rice bran powder. This is the methodology. This is the project process flow chart and the trials. Process flow chart means first dry ingredients will be taken, and jaggery syrup preparation will be made, followed by mixing, sheeting, shaping, and finally baking, freezing, or cooling. We can use any of the method to prepare a bar. And these are the trials. I've taken 10 trials to optimize this product. And these are the trials and analysis I've taken. I've taken water activity. Minerals, I used flame photometer for analyzing the minerals, as and followed by the sensory. These are the results. I've taken the 10 trials for total optimization. Based on the trial 6 has the good sensory and nutrition content. So I've taken this as the final optimized product, and these are the test values of the control peanut bar and the nutrition bar. Control peanut bar means normal peanut solid cake. Nutrition bar is a product. This is discussion, and the water activity of the nutritional bar is significantly lower than the control bar. It indicates the lower moisture content, which is helpful for shelf-life stability and preventing the microbial growth. The moisture content is a little higher than the control peanut bar. This might be the usage of rice bran powder and lemon peel powder. The ash content of the nutrition bar is higher than the control peanut bar, which this suggests the higher mineral content in the nutrition bar is likely due to the presence of both the rice bran powder and the lemon peel powder, both offer which is rich in minerals. In control peanut bar, no minerals like potassium, sodium, lithium, nothing is detected in the flame photometer, but in nutritional bar, so both sodium and potassium are detected at significant levels, and the levels are equal to the daily intake level. This indicates that the incorporation of lemon peel and both rice bran and lemon peel powder, it enhances the mineral content of the nutritional bar. Finally, I would like to acknowledge the support of my food technology department of Engineering College and my guide, Dr. Sadil Kumar sir, for their support. That's it, sir.
Hello, Karthikeya.
Yes, sir.
So I have a query from you. So you have used different sensory properties. You have find out different sensory properties, but one of your graph you have shown that actually there is no error bars are added. So you have taken only one replicates or many replicates for these samples.
Sir, sorry, sir, I can't understand. Sir.
So go back to your graph which you have shown, showing different sensory properties here. Go back to your slides.
Sir, which slide, sir?
No, before that one. Yeah, this slide. Where you have shown that all sensory properties, water content, everything. Yeah, this one. So you have shown moisture, pressure, everything, but you haven't shown any error bars here. Because how many samples have you used for your study?
Sir, maybe I can tell about the protein content and carbohydrate content also, but my project is I'm concentrate only on the mineral content and rather than the protein or carbohydrate content, which is normally will be equal in the both control bar and the nutrition bar also. But I'm concentrate more of the mineral analysis and then that. That's why I used.
I'm having a simple questions. How many different samples have you used for all these studies? So, okay, Karthikeyan, one more question. What can you conclude from overall conclusion from your study? Can you give in one line?
Sir, your voice is not properly audible. Sir.
I'm asking, can you give one line conclusion of your study?
One line vision of your study, sir.
Okay, fine, Karthikeyan.
Sir, objective, sir.
Can you conclude your study in only one line? Why you have conducted this study and what are your takeaways from this study? Okay, I think we are running short on time. We are calling the next candidate, who is
Miss Kirtana N. Miss Kirtana S.
Yes, sir.
Can you hear us?
Yes, sir.
Yeah. Please share your slides. Yeah. Make it full screen.
Is the screen visible, sir?
Yeah, it is visible. Please make it full screen. Don't zoom it. Make it full screen. The button left to it. Yeah, right. Yeah, please start.
Good afternoon, everyone. Myself, I'm Kirtana Srinivasan. I'm a PG scholar and I'm currently pursuing for technology in Engineering College. And my guide is Dr. A. Sudha. And my work is on studies on developing grapefruit extract cocoa balls. So the main aim of this work is to optimize the formulation of cocoa balls by evaluating how ultrasonication affects the grape juice extract. The scope of this is to capitalize on market demand for unique refreshing drinks, highlighting the nutritional benefit of grapefruit as it is rich in antioxidant and vitamin C, crafting an innovation that combines the fruit flavor with boba's fun, and to leverage the popularity of boba culture and growing interest in new flavors. And the main objective of the study is to study the effect of ultrasonication on grapefruit juice based on its physicochemical properties, to optimize grapefruit extract cocoa balls formulation, to evaluate its sensory profile and consumer acceptance. So this is a basic introduction about the work. So basically grapefruit, as we all know, it is rich in vitamin C and antioxidant, and it has a tangy sweet flavor with refreshing citrus essence, which enhances the flavor, citrus flavor in it, and it is mostly preferred in beverages and desserts with novel innovation in the recent days. And spherification is a technique that forms liquid spheres with gel-like outer layer and liquid center, offering a unique burst in mouth experience. By infusing grapefruit extract, these boba balls are being made. The process involves combining grapefruit juice with a gelling agent and dropping it into a calcium bath where the spheres are being formed. And the liquid is being entrapped inside, and this is the methodology. Initially, the grapefruits were purchased from the local market. It was peeled and blanched, and it was blended, and it has undergone ultrasonication, and the filtered extract was optimized using the physicochemical analysis, and the grapefruit balls were formed. And this is the effect of ultrasonication on the juice extract. So I have undergone 3 different times. So it is like 10 minutes, 20 minutes, and 30 minutes of sonication time with temperature of 20 degrees Celsius. And these were the analysis that we carried out. pH, bricks, titratable acidity were analyzed for the sample of control and treated sample of 10 minutes, 20 minutes, and 30 minutes. And these were the results obtained. And the color values were checked. And in the chemical analysis, antioxidant activity and the vitamin C of the juice were performed. And increased Brix value altered the color properties. And there was slightly a decrease in pH, which is due to the breakdown of organic acids. And titratable acidity and Brix varied due to the release of the acidic compounds present in it. And the studies on sonication improves the juice extraction and maintains the higher nutrients level compared to the convenience method. However, change in pH, Brix, and titratable acidity had an influence on flavor and shelf life, as it requires careful optimization. And this is a trial composition. Ingredients used were grapefruit juice extract, sodium alginate as a gelling agent, and salt. As we all know, grapefruit is light bitter in taste. The salt was the best suitable rather than sugar. So I opted for salt and calcium lactate and water. And these were the different trials. And among this, the optimized sample has undergone the analysis like hardness, swelling capacity, vitamin C, total antioxidant, and sensory. Based on all this analysis, it has been concluded that the developed boba had, when the sodium alginate levels were in a lower concentration, it showed no hardness with increasing sodium citrate levels. Hardness varied between 204.3 and 395 compared to the commercial range. It was around 300 to 400, and maximum swelling capacity occurred with higher sodium alginate levels and minimal immersion time. And enhanced antioxidant activity and liquid entrapment was observed, and vitamin C content remained unaffected even though with the ingredient variations, with peak absorptions at 0.3% of sodium alginate and 0.2 sodium citrate. The sodium alginate concentration and swelling capacity, among the other factors, had a significant impact on boba. And I extend my thanks to the department and the head of the department and the other authorities. And I would also like to extend my thanks to my guide, Dr. Ishita, for her valuable support, and special thanks to the coordinator. Yes, sir.
You can please stop. I have a question. What are the benefits of grapefruit extract for health?
So actually, grapefruit is rich in vitamin C and antioxidant, and it is a seasonal fruit. It is not being available throughout the entire period. So by making a grapefruit boba balls, people can taste it throughout the entire period, not only in the particular season.
All right. So what do antioxidants do to the body? Why are they so beneficial?
Antioxidants help in boosting the immunity of the body.
Okay. All right. Yeah, there's one more question from Dr.
One more question.
Yes, sir.
My question is, it's an interesting study that sonication is altering the food property, right? So
Yes, sir.
how did it happen? Can we talk about that? How sonication can be, altering the food content or the quality of the juice?
So due to sonication, there is the effects such as cavitation, which is happening inside. Due to sonication, the heat generation will be happening which will be minimal compared to the other methods. And when I undergone the study over here, I compared with the control and the treated sample. It showed improved properties in terms of vitamin C and antioxidant, whereas the color was affected slightly.
Okay, okay, thank you, Krishna. You can please log out.
Yeah, thank you.
And our next presenter is Mr. Jiva Bharti.
Yes, sir.
Yeah, can you please start sharing your slides?
Okay, sir. Okay.
Yes, please start with it.
I am Joe Bardi. Topic is
Yeah.
development of sustainable biodegradable film using food waste. Introduction. The project investigation the develop of biodegradable film using fruit waste. Sustainable materials that can replace conventional plastic are required. Food waste is a significant problem in many countries, and it can be converted into valuable products, including biodegradable films. Biodegradable films offer several advantages such as reduced environmental impact, improved soil health, and the potential application of packaging, agriculture, and consumer goods. The project aims to develop novel edible films by utilizing of food waste as premises of biopolymers. The objective: develop biodegradable film using food waste and to study physical properties and mechanical properties of the developed edible film, to study biology of the developed film. In this method, films are prepared by casting method. Polymer of fruit husk cellulose, potato peel starch, kefir peel, and agar agar was dissolved in under distilled water and add 50 ml of glycerol. The solution was heated at 80 degrees Celsius for 20 minutes. The solution was cooled at 25 and poured into clean petri plate. The petri plate were dried for 48 hours at 50 degrees Celsius in the oven. These are the trials I have done. Totally, I tried. Totally, 7 times I had. These are the samples, trials and analysis. Result. These are the analysis I had been carried out, which tells that thickness, tensile, elevation, moisture content, biodiversity rates is higher than control sample, where water absorption is lesser for biodiversity sample. Discussion thickness of the biological film, higher thickness compared to control film, which might influence the film structure and density. The tensile strength of biological film, higher tensile strength control film, making the biological films more robust and less prone to tearing. Elongation of the biological films, higher elongation than the control film. This improve the film flexibility and resilience, making them less brittle. Moisture content of the biological film, slightly higher moisture content due to hydrophobic nature of additional component which might absorb more moisture. Water absorption of biological films, lower water absorption of the control film. This film improve the barrier properties against water penetrations. The control film and biological films, comparable biology within 25 to 30 days. This film do not significantly affect their biology. After all, sincere thanks to Kong Engineering College and Ms. Mona Priya ma'am, Assistant Professor, Kong Engineering College for Technology. This support and guidance. Thank you.
Thank you, Jivavati. You can please log out. And our final presenter will be Ms. Tina Patida. Can you hear us? Tina, can you please hear us? Tina Patida? Right, so Miss Tina Patida is I think having some issues with the internet. And so we will finish this session and we'll break off for the lunch and we will meet again after 15 minutes. Right. Thank you, everyone.
Okay, so we are closing this session and we had a very nice talks and presentation of the students. We'll be meeting after some time. Thank you so much. Thank you all.
So I had a doubt. People who have logged in and they have presentation to be given after, say, this lunch session, should they stay logged in?
Can you please repeat your question?
Yeah, I am Dr. Ragini Singh from AIMS Rajkot, sir. I have the presentation
Yes, ma'am.
scheduled. So should I stay logged in or should I log out from this?
Hello, Ragini. Dr. Ragini, this is Vijay.
Yeah, hi Vijay.
Yeah, this is the session, this one which you have joined us right now. We are closing for 10 minutes. Okay.
So can I keep logged in? Because we have this internet issues, patchy internet issues. So if I'm logged in, so I would prefer to stay logged in then.
Okay, then keep logged in, no issue. Okay.
Okay, thank you.
Okay then. Okay, continue then. Okay.
Thank you. Thank you.
Okay, thank you. Hello. Hello.
Hello.
Dr. Ragini.
Yeah, hello, Dr. Vijay, please.
Yeah, Dr. Ragini, we are just sharing the PPT.
Yeah.
Just let me know whether you can see it.
Okay.
Yes, it is. It's online from our end. Can you see it now?
Yeah, I can see.
Okay. Then so I think we can start. It is fine.
Yeah, just a
Yeah.
second.
Okay. So let Dr. Ravneet Chuk ma'am to introduce you. Okay.
Yeah.
Okay then. Okay. Thank you. Thank you for joining.
Thank you, Vijay. Thank you. Am I audible?
Yeah. You are perfectly audible. Okay. You're audible. Absolutely fine. Okay.
Thank you. Thank you.
Thank you.
A very good afternoon to one and all present here. I, Dr. Ravneet, on behalf of Amity University, Rajasthan, once again feel privileged to welcome you all as we have gathered here in the afternoon session of this conference entitled Sustainable Development in Applied Science, Engineering and Technology, coming under the 4th International Convention on Green Environment, Technology and Entrepreneurship through Innovation. I welcome our keynote speakers, Dr. Ragini Singh and Dr. Alok Shiomati Tripathi, Organizing Chair, Dr. Vijay Chaudhary and Dr. Arpit Srivastava, Session Chairs and Co-Chairs, Dr. Karun Chandalia and Dr. Arpit Srivastava, faculties, student participants and delegates from across the globe. So before we start with this session, I would first like to introduce our keynote speaker, Dr. Ragini Singh. So Dr. Ragini Singh earned her doctoral degree from the Gujarat Cancer and Research Institute in Ahmedabad, India in 2012. Her groundbreaking research entitled Genomic and Proteomic Signatures in Oral Cancer focused on the development of biomarkers for assessing the risk of oral cancer. Dr. Singh's work delved into the polymorphism of xenobiotic metabolic enzymes and cell cycle regulators, highlighting their roles as susceptibility markers in oral cancer. Additionally, she also dedicated significant efforts for understanding the mechanism of tumor recurrence and metastasis associated with this malignancy, contributing to the development of biomarkers for aggressive tumor traits. Notably, she also reported novel mutations in the p53 gene known as the guardian of the genome. Dr. Singh currently serves as an associate professor of biochemistry at the All India Institute of Medical Sciences in Rajkot, Gujarat. In this role, she continues to advance her research and academic endeavors. Dr. Singh has authored and co-authored 35 scientific publications in reputable journals, demonstrating her significant contributions to the field of cancer research. Her research interests are diverse and impactful, focusing on the cancer micrometastasis, circulating tumor cells, and circulating nucleic acids, including circulating tumor DNA and also microRNA. Through her work, Dr. Singh aims to enhance the understanding of the cancer biology and also contribute to the development of innovative diagnostic and therapeutic strategies. Her dedication and contributions make her a leading figure in the field of biochemistry and cancer research. Ma'am has joined us online for this session. So with these few introductory remarks, I would now welcome Dr. Ragini Singh to deliver her keynote talk.
Am I audible?
Yes, ma'am, you're audible.
Thank you. Thank you, Dr. Ravneet Chugh and Dr. Vijay Chaudhary for the very kind introduction. Good afternoon, everyone. At the start, I would like to thank Founder President Dr. Ashok K. Chauhan, Chancellor Dr. Aseem Chauhan, Vice Chancellor of Amity University, Rajasthan, Professor Amit Jain, Dean Research and Director, Amity Institute of Biotechnology, Professor Vinay Sharma, and Organizing Chair of ICGETEI 2024, Professor Manju Kaushik, for giving me this opportunity and having me here before the August gathering. Since we are based on the outskirts of Rajkot, so the internet connection is a little patchy. So I would just take the liberty to switch off my video mode so that the continuity stays. Today I will be talking about a molecule, lipoprotein little a, its role in atherosclerotic cardiovascular disease. Basically, this talk is little deviation of my core area of research, but at AIMS, we have sort of focus on research. We have planned to deviate towards the diseases which are non-communicable in nature. So our focus has not only been the cancer research, so we have also started working on atherosclerotic cardiovascular disease, the problem which is very prominent in our region. So for today's talk, I will be talking on the lipoprotein little a in atherosclerotic cardiovascular disease. I'm going to briefly walk you through the history of discovery of lipoprotein little a and the role it plays in cardiovascular disease, and the recent discoveries which are providing a new twist to this old age saga. So next slide, please. Next slide, please. Can we have this in a full screen mode, please?
Hello.
Yeah, can we have this in a full screen mode, Dr. Vijay?
Yeah, it is in the full screen mode right now. Is it visible to you?
Yeah, it is visible. Okay, fine. There may be some issue at my end.
Maybe it is a bit low speed over there. It is full screen over there.
Okay, thank you. So these are the objectives of my talk. And firstly, I will be providing a brief overview of LPA structure, metabolism, and measurement. This will be followed by the role of lipoprotein little a in the pathophysiology of atherosclerotic cardiovascular disease. The third objective will be the clinical significance, reviewing the clinical significance of elevated lipoprotein A levels, both in the primary prevention as well as secondary prevention settings. So primary prevention settings, the patients who have not experienced any episode of cardiovascular disease, and the secondary prevention settings is patients who have had the episode of cardiovascular disease. And lastly, I will be focusing on the impact of lipid-lowering therapies on lipoprotein little a and the new targeted drugs in the horizon. Next slide, please.
Is it visible now? We are in the next slide.
No, we are not. I'm not able to see. Next slide, please.
It is on this slide 3 right now.
Okay, but here at my end, it is slide number 2 only.
It seems like it is very slow over there.
Yes, we have this problem. Should I share my screen? Should I try with that?
Maybe you can try once if it works.
Yeah.
Or maybe then it will be more difficult because then, from your end again, it will take that much time to come over here, no? Like the way it is taking time to reach there.
Yeah.
So if you can keep your laptop on, I'll be changing the slides, and if there is any difference, we will somehow communicate with each other. Right now we are on third slide.
No, it's still showing me the second slide, which Dr. Vijay. Sorry.
They'll do one thing, I'll stop sharing and share again, okay?
Yeah, that will be better.
Is it visible now?
Yeah, it is visible, but I hope you are seeing it in a full screen mode.
Yes, yes, it is in the full screen mode.
Fine, fine. So let's begin with the first objective, the overview of lipoprotein little a, or LPA it is commonly called as, its structure, metabolism, and measurement. So this molecule was discovered by Dr. Berg in 1963, and he recognized this molecule as an antigenic variant of low-density lipoprotein, which is LDL. And we all know that LDL, which is in a common language termed as bad cholesterol, is pathogenic and causative for atherosclerotic cardiovascular disease. The figure on the left is a schematic presentation of this molecule, and I will take a deeper dive into it in the next slide. So next slide, please. Yeah, thank you. So this is the zoomed-in version of the schematic, and I would like to spend a couple of minutes to review the structure of this LPA molecule. The spherical structure in the orange color, which is shown here in the center, is the LDL-like particle. And as you can see, the green structure, which traverses pretty much across this orange structure, it is serving as a structural scaffold, and this is termed as ApoB100, and it is serving as a structural scaffold for all the lipids which are contained. And the green one, they are called as, they resemble the LDL-like particle. But a special feature which makes this molecule a little different from covalently bound to this LDL-like particle. Now, this additional structure, it is peculiar in the sense that on the left side in the gray color is an inactive protease domain, which is followed by amino acid sequence called as kringles. Kringle is basically the shape of these amino acid domains resembles the Danish pastry, which is called as kringle, and hence they get the name. So these amino acid sequence, they are called as kringle repeats. So starting from the bottom, we have kringle 5 followed by kringle 10 to 1. And then if you can see, which are termed as kringle 4 repeats. So an important point here is to note that this kringle other two kringle units which are of importance are the kringle 4-9 domain, which is linked to the oxidized phospholipids, which is present inside the spherical molecule. And these oxidized phospholipids, they are termed to have a kringle 4.
Hello, Ragini, Dr. Ragini, can you hear me? Hello, Dr. Ragini Singh.
Yeah, Vijay, is it?
There is a kind of a voice break in between. So is it possible for you to join maybe through the phone, or maybe we can continue like this, whatever you suggest?
I have joined through phone only. I'm using my personal
Okay, then let us continue like this. Okay, then.
Is it again means there is break in the voice?
Right now it is absolutely fine. Once in a while there is some break.
Okay. So the figure on the right shows the evolutionary lipoprotein molecule, and if you can see, both of them, they are having inactive protease domain on. So in ApoA molecules, we have Kringle 4 and Kringle 5 only, whereas in plasminogen, we have 5 different types of Kringle domains. Next slide, please. So the schematic here shown in this figure shows talks about the LPA synthesis and the clearance. The major organ that is involved in metabolism of LPA is the liver. And here you can all see that this is a structure. This is a structure for a hepatocyte, and within the nucleus, we have the LPA gene, which is coding for LPA mRNA. And this LPA gene is located on chromosome number 6. And through the process of transcription, it produces mRNA. This mRNA, after transcription, gets translocated into the cytoplasm, and these ApoA molecules will get folded in the Golgi apparatus and will be secreted in the cytoplasm or towards the cellular surface. So where they will form an assembly with LDL-like particle to eventually form the LPA particle. So this is an area of research where the assembly of these particles is taking place. So once this LPA molecule has been formed, it is released into the circulation. For its clearance, again, the hepatocytes, they have the receptors, dedicated receptors, the LDL receptors, the scavenger receptor B1, and the dedicated receptors to plasminogen because of the structural homology between ApoA and plasminogen. Now, once this particle is again back into the hepatocyte, it is recycled or degraded within the lysosome. Next slide, please. Now, this schematic here shows the importance of LPA gene and its role in modulating the LPA levels. So I will walk you through the schematic, and this is taken from a recent publication of 2021. So we have this LPA gene on chromosome number 6, and the two alleles are there on the chromosome number 6. So allele 1 is termed as small ApoA allele. Allele 2 is termed as large ApoA allele. An important point to remember here is that both of these alleles, they will contribute to the final lipoprotein A concentration in the circulation. So basically, this is an example of autosomal dominant inheritance. And in this example, which is shown in the figure, we can see that small ApoA allele. Why this is termed as small ApoA allele? Because of the presence of only smaller number of Kringle 4-2 subunits. So here in this case, it is having 10 subunits. Whereas when we are talking about the large ApoA allele, it is having 25 Kringle 4-2 subunits. Now, both of these alleles, they are contributing to the plasma concentration of LPA. But because of the laws of molar concentration, the smaller allele will contribute to higher concentration of the plasma, which is evident here in the figure. So the smaller allele is contributing to somewhat 60 milligram per deciliter of LPA, whereas the larger allele is contributing to somewhat 10 milligram % of LPA concentration. But the final concentration of LPA in the circulation is roughly 70, which is sum of the product of both the alleles. One more important thing to bear in mind is that the 90% of the LPA levels, they are genetically determined. Next slide, please. So moving on to the measurement of lipoprotein little a. So we have two commonly available assays that are used in clinical practice. The first set of assays, they are termed as mass assays, and they report the LPA level in milligram per deciliter. The second class of assays are the particle assays, which report the levels in nanomole per liter. Of these two, the recent guidelines suggest that particle assays are better in the sense that they are giving the total LPA numbers in the circulation. And we all know that the total LPA number is directly related to degree of atherosclerosis. The mass assays, they are insensitive to ApoA also. And we have just learned that this ApoA can be small in size or it can be larger in size, and that is impacting the final concentration of LPA in the circulation. Next slide, please. Now I'm going to briefly go over the role of LPA in atherosclerosis pathophysiology. And I would like to start by mentioning that LPA influences atherosclerosis through multiple non-redundant pathways. Next slide, please. These pathways, they are illustrated in the schematic. The exact mechanism needs to be elucidated further, but one of the key mechanisms by which LPA is thought to cause atherosclerosis is that this molecule is considered proatherogenic. The reason for this is because it has got similarity to LDL-like particle, and LDL are the causative agents for atherosclerosis. So these molecules, what they do is they will bind to the vascular endothelium. They will upregulate the adhesion molecules. They will cause increase in the SMC proliferation. They will lead to formation of foam cells and increase in necrotic core formation and lesion calcification, ultimately forming the atherosclerotic plaque. The second mechanism by which LPA is thought to cause atherothrombotic disease is that this particle is considered to be prothrombotic, and this traces back to its homology with ApoA and plasminogen, and that this particle decreases plasminogen activation, decreases fibrin degradation, and increases platelet responsiveness. And the last non-redundant mechanism by which LPA can cause atherosclerosis is by promoting inflammation. So oxidized phospholipid, which is present in this molecule, play a key role in driving the inflammatory cascade in atherosclerosis. And then there is increased expression of interleukins and chemokines, particularly interleukin-8, that promotes atherosclerosis. Next slide, please. And now I will be talking about the clinical significance of elevated LPA level. Next slide, please. I would start off by briefly discussing the clinical practice guidelines, which are available at this point of time, and whatever their recommendations regarding checking LPA levels are. The first one to go in here is the guidelines suggested by American Heart Association and American College of Cardiology. These guidelines were released in 2018, and these guidelines suggest that LPA concentration should be measured in subjects who have family history of premature atherosclerotic cardiovascular disease, personal history of premature atherosclerotic cardiovascular disease with no major risk factors. The next one to go in here is the guidelines from European Society of Cardiology 2019, which was released a year after the above guidelines. These guidelines have expanded the role of checking LPA. These guidelines recommend that LPA should be checked once in a lifetime of a person, and individuals who have extremely elevated LPA level, that is greater than 180 milligram per deciliter, they would have important implications for discussing the cardiovascular disease risk, because in these individuals, if we have a discussion regarding their 10-year risk towards cardiovascular disease, they can be they're gaining towards the cardiovascular disease can be curtailed by use of lipid lowering therapy at the right time. Now, similar to the American guidelines, the European guidelines also recommend assessing LPA in individuals who have family history of premature cardiovascular disease. So what we understand about premature cardiovascular disease is presence of the disease in individuals who are less than 55 years of age. Now, most recently, the National Lipid Association, pretty much the same year, that is 2019, they released a scientific statement solely focusing on LPA, and they have further expanded the subjects in whom the checking of LPA is reasonable. So these patients where the LPA should be checked, that include individuals with family history of premature ASCVD, personal history of ASCVD, individuals who have primary hypercholesterolemia, that is LDL concentration greater than 190 milligram per dl, individuals who have borderline 10-year atherosclerotic cardiovascular disease risk ranging from 5 to 7.5%, individuals who have less than anticipated LDL cholesterol lowering, even with lipid lowering therapies, and individuals with cascade screening in families with hypercholesterolemia. Next slide, please. Out of all these guidelines, I would like to highlight the 2018 cholesterol management guidelines. Now, this schematic looks a little busy, but I will walk you through some of the key aspects in the next few seconds. This schematic is particularly focusing in individuals of the primary prevention group. So these are the individuals they may be the individuals who have LDL cholesterol concentration greater than 190 milligram per deciliter, individuals with diabetes mellitus falling in the age bracket of 40 to 75 years. So this American Heart Association guidelines, they recommend estimating the 10-year cardiovascular disease risk by making use of a pooled cohort equation. So pooled cohort equation. So we have individuals with 5 to 7.5% who fall in this category, they are at borderline risk. Individuals with 7.5 to 20% category, they fall in the intermediate risk category. Now, in these individuals, there should be a risk discussion with the clinician, and also the assessment of risk enhancers, which are shown on the right side of the slide. So this column represents the atherosclerotic cardiovascular disease risk enhancers, and these are commonly assessed during the process of history taking. But I would like to focus your attention at the bottom of this column, where apart from the other markers associated with increased risk, I have highlighted the LPA levels of greater than 50 milligram per deciliter assessed by mass assays and greater than 125 nanomole per liter assessed by particle assays. So if you have a measurement of LPA in the primary prevention settings, which is higher than this threshold, then you can consider the use of lipid lowering therapies in such individuals. Next slide, please. And if we use this cutoff of greater than 50 milligram per deciliter, there is an estimated 1.43 billion individuals across the world who have elevated LPA levels. And as you can see, the burden of elevated LPA varies by the continent as well as by the ethnicity. The highest proportion of individuals with elevated LPA burden would be in Africa. Nearly 30% of the individuals, they have elevated LPA, followed by South Asia, where 25% of the individuals have elevated LPA. So this points out why India is experiencing a very high cardiovascular disease burden. Next slide, please. So the graphs show here the population distribution of lipoprotein a, and the reason why this is important to consider is because we have already learned about the threshold, which is defining the higher and lower LPA levels. But what is important to remember is that distribution of LPA is showing a skewed pattern in the graphs. So this study was conducted by Warwell and colleagues in the year 2016. They enrolled some 500,000 individuals with equal representation from both the genders. So if you can see from the left side of the graphs, so this LPA distribution is showing a skewed distribution pattern with tailing towards the right. And this is in stark contrast to the LDL cholesterol distribution in the population, which is showing a near normal distribution. Now, what is this slide implying? This is suggesting that nearly 1 in 4 individuals, they will be classified as having elevated LPA level. Next slide, please. Another important thing here is, as I've mentioned earlier, LPA concentration varies by ethnicity. So this data is from 2015 study, MESA study as it is popularly known as, which is multi-ethnic study of atherosclerosis. In this study, the individuals were grouped in cohorts belonging to different ethnic groups, that is Blacks, Caucasians, Chinese, and Hispanics. So if you can see the distribution of LPA pattern, it is somewhat similar what we have discussed in the earlier slide. It is skewed and tailed towards the right. But what is evident from this slide is that the median levels of LPA, it is higher in Blacks, 3 times higher than the other ethnic groups, that is Caucasians, Chinese, and Hispanics. The other 3 groups, that is Caucasians, Chinese, and Hispanics, they are having somewhat similar median levels. Next slide, please. So we have talked about the role of LPA in primary prevention settings. Here we'll be comparing the LPA levels and cardiovascular disease risk in both primary and secondary prevention settings. So this is a study by Patel et al. Published in 2021, where he enrolled somewhat 400,000 individuals. He has used the molar assay for cutoff, that is 150 nanomole per deciliter. What they have done, what the authors have done, they have divided the subjects into two groups. One who were not having any atherosclerotic cardiovascular disease at the enrollment. The other group who had already experienced the episode of atherosclerotic cardiovascular disease at the time of enrollment. So on the left part of the graph, you can see the distribution of LPA. Again, it follows the same pattern, skewed and tail towards the right. And in the second graph, it is shown that the individuals who have elevated LPA levels, that is greater than 150 nanomole per liter, they were at higher risk for incident cardiovascular disease events in comparison to individuals who had lower than 150 nanomole per deciliter. So this part we are talking about is the primary prevention settings. And when we see these this type of comparison in the secondary prevention settings, what we observe is that the individuals who have higher levels of LPA, that is greater than 150 nanomole per deciliter, they are at higher risk of recurrent cardiovascular disease events. Next slide, please. So now we move on to the last objective of my talk, which is impact of lipid lowering therapies on LPA levels. I will start with discussing with the most commonly prescribed medicine that is used across the world to treat hypercholesterolemia, that is statins. Next slide, please. This study was published in Lancet in 2018 by Ville et al. And the results are based on 7 different trials conducted for study of impact of statins on risk for cardiovascular disease. So they found, what they have shown is that they have calculated the hazard ratio based on the LPA levels. And what they found was that the hazard ratio was showing somewhat overlapping effect even in individuals who were taking the statins for treating hypercholesterolemia versus the individuals who were not taking statins, that is placebo. So what it implies is that individuals who are receiving statin therapy, but if they have high elevated LPA levels, they are at higher risk for cardiovascular disease risk. Now, this brings me to the controversial paper. Next slide, please. Which was published in 2020 in European Heart Journal by Sam Simikas. So the results of this study are based on 6 different trials. And the authors, they wanted to demystify the impact of statin therapy on LPA levels. Now, why I'm saying this paper is controversial, because this paper, the results of this paper based on some roughly 5,000 subjects, they said that statin therapy, which is used to treat hypercholesterolemia, it is associated with slight increase in the LPA levels, roughly 11%, and thereby increasing the risk towards cardiovascular disease. So the possible explanation which the authors have given, the hypothesis the authors have suggested, is that the individuals who are not receiving statins on the left part of this slide, so this is a figure for liver, individuals who are not taking statins, there what is happening, the ApoB100, which we talked during the structure of LPA, the ApoB100, it is being utilized for formation of LDL as well as it is being utilized for formation of LPA. Whereas the individuals who are on statins, the statins will bring the LDL concentration down, and this will help in recycling of ApoB100. Now what will happen? The availability of ApoB100 for formation of LPA will be higher. So this leads to increase in the LPA concentration. Next slide, please. So moving on to, we have just talked about one class of drugs which is used to treat hypercholesterolemia. So now we are moving on to another class of lipid lowering agent, which is PCSK9 inhibitors. So briefly, I would like to mention the results of 2 key trials. First one is the 4-year trial. So what they did was they studied the PCSK9 inhibitor, evolocumab, for reducing the LPA levels. what they did was they enrolled some 25,000 individuals, measured their LPA level at the baseline, divided them into 4 groups. Q1 represents the group which is having the lowest LPA level, and Q4 represents the group which is having the highest LPA level. So this drug was given to these individuals and the LPA levels were assessed after 48 weeks. And what it was found that evolocumab, which is a PCSK9 inhibitor, was able to reduce the LPA levels in all the 4 groups, but the degree of reduction was higher in individuals who already had a baseline LPA level which were higher. Next slide, please. Next, yeah. So similar to the results of 4-year trial, Odyssey outcomes also give the same kind of conclusion, but the inhibitor which they have used, the PCSK9 inhibitor which they have used here is the alirocumab. On the similar lines of the previous study, they also measured the baseline LPA levels, divided the individuals, somewhere 18,000 individuals, into 4 groups. Again, Q1 represents the lowest baseline LPA levels, Q4 the highest LPA levels at the baseline. And this drug was administered and LPA levels were again assessed after 4 months. And it was found that the LPA level has gone down in all the 4 groups. Again, the degree of reduction was highest in individuals who had higher levels of baseline LPA. Next slide, please. So I will now move on to the new class of drugs which are there in the trials, which is use of antisense oligonucleotides for targeting the LPA levels. So this schematic here shows the role of antisense oligonucleotide. So once this antisense oligonucleotide is administered, it will enter the cytoplasm of the hepatocytes, bind to the mRNA of LPA gene, form a duplex with this mRNA. Once this duplex is formed, the enzyme ribonuclease H1 will degrade this duplex and eventually no protein will be produced and LPA level will go down. Next slide, please. So next, please. So here I will be talking about the different trials which are making use of these antisense oligonucleotides to lower the LPA levels. The first trial, this is the graph. These are the results from phase 1 trial, which have used this antisense oligonucleotides. So what they have done is the investigator has used 3 different doses of this antisense oligonucleotide, that is 100 milligram, 200 milligram, and 300 milligram for 6 weeks. And as is evident from the graph, the LPA levels were measured at 6 weeks. So the reduction in the LPA levels with 100 milligram was somewhat 40%. With 200 milligram, it was some 60%. And with 300 milligram, it was some 80%. Next slide, please. Now, this phase 1 trial was followed by the phase 2 trial, and similar results were obtained in the phase 2 trial as well. In the phase 2 trials, the investigators had 2 cohorts, cohort A, who had high LPA concentration, and cohort B, who had very high LPA concentration. This drug was administered again in 3 phases, and LPA was measured after 4 weeks, and roughly 66% reduction in the LPA levels was observed for cohort A and roughly 70% for cohort B. So a touchdown was observed at 4 weeks. Once the drug was withdrawn, the levels of LPA started to rise. Next slide, please. So this is the drug pelacarsen, which is in the phase 3 trial. It is being conducted across. It was conducted across 240 locations, published in New England Journal of Medicine in 2020. Similar results were obtained and reinforcing, corroborating the findings of the previous 2 trials. The variations, variations of the drugs were used at different time intervals, and the results were reduction in the LPA levels. Next slide, please. The key takeaways from the talk is that the lipoprotein little a is an LDL-like particle with ApoA produced in the liver. This molecule is an independent causative agent for atherosclerosis, and there are several non-redundant mechanisms by which it is causing this. Now, elevated LPA level is associated with atherosclerotic cardiovascular disease risk, and the recent clinical practice guidelines, they are moving towards broader LPA measurements, and it is the right time when we should introduce the measurement of LPA in the lipid profiles. PCSK9 inhibitors and palacarsins, they reduce the LPA levels, and these are the new therapies in the armamentarium of the clinician for lowering the lipid levels. Still, for palacarsin, the phase trial will confirm the hypothesis. Thank you so much, and thank you for bearing with this patchy network. My sincere apologies to the organizers for this. That's all from my side.
Thank you so much, Ragini ma'am, for such an informative talk on lipoproteins.
Thank you, ma'am.
Besides all the technical glitch you were able to handle so flawlessly. Thank you so much, ma'am.
Thank you, ma'am, for bearing with
Chaudhary just had to go urgently for some work. Otherwise, she would have thanked you personally. Thank you so much, ma'am.
Thank you, madam. Thank you, madam.
Thank you so much, ma'am.
Yeah.
Moving on further, I would now like to welcome our next keynote speaker, Dr. Alok Shivamurthy Tripathi. Sir is currently working as professor and head of Department of Pharmacology, IRA College of Pharmacy, IRA University, Lucknow, UP. He completed his PhD from Birla Institute of Technology, Mesra, Ranchi. He has 16 years of teaching and research experience. He also has 52 research papers in various national and international journals with cumulative impact factor of more than 100. 14 book chapters and 1 book are also on his credit. Presently pursuing a research work funded by ICMR under extramural ad hoc scheme worth rupees 37 lakhs. His major research focuses on diabetes and its complications, drug interaction, neurodegenerative disorders. He has also attended 2 international conferences at Japan and Dubai, which was funded by SIR, New Delhi and ICMR, New Delhi. Moreover, he is also working as reviewer for many high-impact journals, and he's also working as editorial board member for more than 5 journals. So with these few words, I would now request Sir to kindly deliver his lecture.
Thank you very much, ma'am. Before the start of my lecture, I'm really thankful to Ashok Dr. Ashok Chauhan sir, Dr. Asim Chauhan sir, patron of Amity University. I am thankful to Dr. Vinay Sharma sir, who is a director in the Department of Biotechnology. I am thankful to Dr. Arpit Srivastava, for giving me an opportunity to present my research in the present conference. Ma'am, am I able to share my slide? Because it is showing on screen, I am not able to start screen sharing.
Okay, sir, just let me check. Sir, you can now share your screen.
Okay, ma'am.
Yes, sir.
I hope my slides are visible to everyone.
Yes, sir.
Yeah. Thank you, ma'am, for the nice introduction. Before start of my session, I really wish to tell you about myself that I am a pharmacologist. So the most of the work what I had did is over the preclinical studies. Means we had done all the work over the animal. So may I start, ma'am?
Right,
With the
sir.
presentation? Yeah.
You can start.
The topic that I'm going to deliver today is the role of nutraceutical and functional food in health management. Uh, Nutraceuticals, it is actually a combination of two words, nutrients and pharmaceuticals, which are food component that are thought to have a health benefits, including the ability to prevent the diseases. The common examples of nutraceuticals are vitamins, minerals, amino acid, fatty acids, and phytochemicals. It has been well documented that nutraceutical improves the health, delays the aging process. It will also helpful for the chronic diseases management. And by taking chronic use of nutraceuticals for a long period of time will also increases the life expectancy. Whereas functional food is concerned, any food material uh, which there is an additional ingredients to the existing ingredient of the food, which has been considered as a functional food, and functional food are used for the health benefit. If you'll see the global market size of the nutraceuticals, it is around 280 billion. It will reach 280 billion by the year USD by the year 2025 and 26, with an average rise of 8% per year. And if you will compare it with the Indian market size of nutraceutical, which is around 3 billion USD only. And that's why it has been expected in the near future that our global market of nutraceutical in India will rise exponentially in the coming years. Nutraceuticals and functional food has a several health benefit. And that's why it is used as an adjuvant therapy. Now, adjuvant therapy means if suppose we are using for the treatment of any diseases, a conventional therapy, what are the drug available in the market, to get the synergistic effect, we used to give nutraceuticals with in combination with the conventional drug. And several research shows that it shows a synergistic effect with the conventional drug. Research in the pharmaceutical sciences is majorly focused on the beneficial effect of bioactive molecules, which are actually comes under the nutraceutical heading. And our lab has also performed several these kind of research work. She is Miss Alka. She had performed her PG from the Amity University, Lucknow, under my guidance only. And she is pursuing PhD. Her dissertation work is on eogenol, promotes the immune function and survival rate in celiac ligation puncture-induced sepsis model by targeting A2A receptor to regulate oxidative stress pathway. Friends, maybe all of you know about it. The sepsis is a systemic infection or inflammation which causes multiple organ dysfunction, and due to which, if you will observe, the patient, those are been hospitalized in the ICCU, the maximum death, major cause of death of those patients are sepsis only. Probably you know about it recently. COVID-19 had been gone. COVID had been gone. Most of the patients, those are been died in the COVID, is because of sepsis. So sepsis is one of the biggest challenge in the field of medical sciences, how to deal with the sepsis. Because the conventional drug, those are available for the management of sepsis, are either targeted by increasing the immune system bit a while, or by reducing the concentration of microorganism in the blood. So we had taken over the eogenol, which is one of the major content of aromatic oil extract of clove, which is one of the food product we generally used to take. And it is eogenol is a flavoring agent, which we used to add in the meat, cakes and teas. It is present in several other aromatic spices like cinnamon also. Eogenol is reported to have several medicinal property like anti-cancer, anti-inflammatory, antimicrobial, antioxidant and neuroprotective. And our study had been seen the effect of eogenol against the sepsis. In the methodology, we had taken Wistar rats. For this study, we had taken male Wistar rats and we had divided the rats into 5 different groups. One group is sham, another one is sepsis group, third one is a standard in which we will induce sepsis, and then after we will give any standard drug. Imipamine is another standard drug. Eogenol 25 mg per kg and eogenol 50 mg per kg are the 2 different groups we had taken. The first of all, whenever we had taken the rats, we will induce sepsis in the rat. This is what the method for the induction of sepsis in the rat. What we will do, first we will anesthetize the rat. After anesthesia, what we will do, we will put it into the inverted position and we will make an incision to the rat abdomen. The incision should not be more than 2 centimeter, 1 to 2 centimeter only incision we have to do. And we have to take out the celiac region, which has been present over the ileocecal junction. We have to tie a thread. You can see in the pictures, I had been clearly mentioned over there. There is a thread has been tied over the celiac region, and then we have to puncture it with the needle. This needle of a different gauge we will select depending upon the severity of sepsis we want to induce. If for mild sepsis, we have to take 20 gauge syringe. If we have to induce moderate kind of sepsis to the rat, we have to use 21 gauge syringe. And if we want to induce severe sepsis, then we will use 18 gauge syringe. Whereas for the sham operated group, we have to just anesthetize the animal. We have to do the incision. Then thereafter, we don't have to do anything. After putting the needle in the celiac ligation puncture, it should be perforated. Perforated means we have to press the cecum so that the fecal material should go outside. Once it will be done, we have to put all the cecum inside the body of the rat, and then we have to put the stitches. This is how we have to produce sepsis and by celiac ligation puncture. After that, we had done estimation of first thing, that is survival rate. Sepsis is having the first and foremost parameter that increase in the survival rate that we had observed over there. We observed the inflammatory and oxidative markers. Plus, we had observed the infiltration of inflammatory cell in the lung, and we had checked the histopathology study. For further evaluating the exact mechanism of action, we had gone for the in silico activity. In silico means where we will use computer, and depending on the computer's result, we will going to postulate the result. In silico, we had done network pharmacology study where we will select the 2D smiley, matlab structure of drug we will select of eugenol from the PubChem database. Then we will select the target protein identification for the eugenol's target. Target means the proteins to which our drug will bind and will show the action. That target we will select from the Swiss target database and binding database. By adding these smiley on this database, we will find a list of targeted protein. These targeted protein we will take in a separate Excel sheet. Then after that, we will take another database, which is this GenNet database, in which we will put sepsis as a disease and different gene which are been variated during the sepsis. That list we will also observe. These both the list we will put into a particular software, which is a Veni 2.1, to assess what are the common gene or common protein which are involved in the disease and the protein and the drug target. Means this will identify exactly where our drug targets comes under the sepsis also. Then the common target which we will get it, we will form a protein-protein interaction network out of it with the help of string database, and then we will perform SR plot method, gene ontology, and KEGG enrichment assay. These all process will going to identify us exactly on which pathway our drug has been acting for the treatment of sepsis. And second thing by which we will identify which particular target is there to which our drug will bind to reduce the chances of sepsis or that particular disease. Another one is after that, the selected targets, like for our study for eugenol, we observe there are two targets. One is A2A receptor, which is nothing but adenosine 2A receptor, and SDASET. These two particular protein to which eugenol will act. Then first we will take the eugenol's structure from the particular database, drug target database. Another one, we will take the proteins A2A receptor and SDAC8 from the protein data bank. And by using the Schrödinger software, in which a GLEAD is a model of Schrödinger software, we will put and we will see the interaction between eugenol with the A2A receptor and eugenol with the SDAC8 receptor to prove the effect of our drug. What we had observed when we had given eugenol, you can see here the pictorial diagram, graphical diagram is there. You will observe that there is a significant reduction in the survival rate in the sepsis group, whereas with the treatment group there is the effective prevention in the there is a significant increase in the survival rate. Whereas we had seen the different weight of different tissues like lung, liver, and spleen organs, we had observed that the weight of each organ is been reduced significantly in the sepsis group, which are been improved with the treatment. Then thereafter, we had observed the different oxidative stress parameters. One of the parameter is MDA, malondialdehyde. The level of malondialdehyde is been increased a lot. In case of sepsis, significantly increases as compared to the sham group, whereas eugenol treatment will reduce it significantly. So with the GSH, it's entirely opposite. And with these results shows that our drug shows a significant antioxidant activity in the case of sepsis. Then after we had observed the different inflammatory markers like tumor necrosis factor, interleukin-16 and C-reactive protein, out of which tumor necrosis factor alpha and interleukin-16 we had checked in both lung tissue and BALF. BALF is the liquid which has been present in the lung tissue only. We had checked it and we observed that the level of these inflammatory markers are significantly increased in sepsis group, which are been reversed with the eugenol treatment. Then thereafter, we had checked about whether there is an infiltration of neutrophils in the lung or not. We observed that there is a significant increase in the neutrophil in the sepsis group, which has been reduced with the eugenol treatment. We had performed a histopathology study also, and we checked the lung injury score, and we observed that during the sepsis, lung injury is one of the major thing, and lung injury score has been raised significantly, which has been reversed with the eugenol treatment. In silico study we had done, we had put the 2D smiley into the target database. Swiss target database has been used to identify the targets for eugenol, and we observed around 54 targets over there to which our drug has been binding in our body. Then thereafter we had checked the different sepsis gene from the DiseGenNet. We observed that it is around 1450 plus gene are involved in the sepsis. And when we had put both the protein and gene together, we found only 14 are the common target proteins are there, which are there binding for the eugenol for the treatment of sepsis. By taking these 14 targeted proteins, we had created a network of protein-protein interaction. And with that, we got a nodes over there. And these nodes we had plot for the SR plot study, and from where we had observed this particular pathway, which has been showing neutrophil extracellular trapping formation. From this, we had concluded that our drug is been targeted for the neutrophil trapping capacity is been increased by eugenol by acting over two particular protein, A2A receptor and SDAC8. We had done a docking study for this, and we observed that the docking study results are very nice over there, and hydrogen bonding is also been there. By these results, we conclude that yes, the eugenol enhances the survival rate in CLP-induced sepsis by reducing inflammation and oxidative stress, and also altered histopathological alteration in lung and spleen tissue by targeting specifically A2A receptor and SDAC8 receptor. If you will see that particular what we had done in the graphical pictorial diagram, graphical abstract, we had induced the sepsis through the CLP model. Then we had checked different inflammatory markers in the different tissues of the rat, and that oxidative stress parameter. These all shows that the eugenol is significantly reducing the oxidative stress and inflammation in the different tissues. Moreover, network pharmacology suggested that that drug has been binding over the A2A receptor. A2A receptor is having a function over there that it enhances the trapping of neutrophil. All of us knows about that oxidative stress is one of the biggest reason to have a number of diseases. But oxidative stress in the neutrophils or WBC cells are good to digest the xenobiotics. So our drug eugenol it increases two things of neutrophil. One is the engulfing capacity of xenobiotic, that any foreign material has been coming, the neutrophil's capacity to engulf it will be increased over there. And another one, the elevation of free radicals inside the neutrophils. If the free radicals will increase in the neutrophils that will help in the scavenging capacity of neutrophil to the foreign materials. So this is what our result, and this particular paper is under revision and in the International Immunopharmacology Journal. The another topic that I wish to present in front of the students, that phytochemicals improve the gram-positive bacteria in microbiota of human could be used as a prebiotic. Before going to start with this particular topic, we should know about our GI tract contain lot of different microbiota, microorganisms. If you will count that microorganism, more than 5 billion microorganisms are present throughout our body. And the highest concentration is present in our GI tract specifically. If you will see the lactobacillus, bifidobacterium, bacillus, and like that E. coli, Saccharomyces, there are so many bacteria are present in our gut, and these microbial flora are being called as gut microbiota. This gut microbiota has having several beneficial effects for our body. They help in the digestion purpose. They used to control the GI motility. They regulate the gut immune regulation also. They help in the metabolism process. They also help into the metabolic function. They help in the neurological function by targeting gut-brain axis also. As far as factors affecting a gut microbiota, there are different factors over there like diet, stress, pathogen, exercise, medication that affect the gut microbiota. But more specifically, our topic has been dealing with the aging. As aging is one of the biggest reason to affect the gut microbiota, which result into alteration in the combination of gut microbiota. That has been called as dysbiosis of gut microbiota. Gut microbiota dysbiosis result into alteration in the function of different organs. One of the major organ which we are going to discuss here is the gut-brain axis. If the gut microbial flora get altered over there, that result into stress, anxiety, depression, schizophrenia, cognitive dysfunction, and autism-like diseases. And that's why it is important to study about the gut-brain axis. So we are dealing with that further. Aging is one of the biggest parameter to alter the gut-brain axis. If you will see the person become in the old ages, the gut dysbiosis occur. And if you will see the gut microbiota, there are gram-positive and gram-negative bacteria, both are present over there. As we become older and older, in the gut, the gram-negative bacteria quantity increases, whereas the gram-positive bacteria's quantity used to be reduced. Gram-negative bacteria on their cell membrane contain a particular protein that is a lipopolysaccharide. This lipopolysaccharide is a inflammation-causing agent is there. Moreover, as we become older, the permeability of the tissues are also been increases, and because of that reason, bacteria may enter into the blood of that old person. And as the bacteria enter into the blood, the LPS level is also constantly increases in the blood of a particular person who is having older age. So if it is increasing in the blood, LPS is having a property that it can cross the blood-brain barrier and it can cause the neuroinflammation over there by activating immune system, and that result into neuroinflammation and neurodegeneration. I wish to discuss with all of you a particular pathway which will be affected over there because of the LPS. A particular essential amino acid is there, tryptophan, whose metabolism will occur over there, and the metabolic product is kynurenine. This kynurenine further metabolizes by three enzymes: kynurenase, kynurenine 3 monooxygenase, and kynurenine 3 transaminase. You can see kynurenase and kynurenine 3 monooxygenase both are been forming a particular metabolite, that is a quinolonic acid. This quinolonic acid will act bidirectionally. One, it activates the microglia cells, which are defense cells of your brain. Because of activation or overactivation of microglia cells, the inflammatory cytokines will increase and that result into neuroinflammation. Moreover, quinolonic acid is also having effect over the glutamate receptors. It activates the glutamate receptor. Glutamate is an excitatory neurotransmitter. Probably all of you know about it. Recently, that Maggie controversy has been occur over there. Maggie contain an Ajinomoto. Ajinomoto is nothing but a glutamate which causes excitotoxicity in the children and that result into neurodegeneration in the children. The same thing, this quinolonic acid is an agonist of it, which activates glutamate receptor, enhances the intracellular calcium level, which further result into neuroinflammation. And if it has not been controlled, that result into neurodegeneration. But how it has been balanced in the normal person? Because at the same time, when these two enzymes are acting over there, kynurenine transaminase is also acting over there, which converted into kynurenic acid, which is an antagonist of glutamate. It reduces the activation of microglia cells, and because of that reason, neuroinflammation is reduced, neurodegeneration is reduced. In a normal person, both are acting equally. But if you will go with the old age people, the LPS quantity get increases in the brain and blood. And because of that reason, kynurenine 3 monooxygenase level is being alter over there. It has been increased over there because of LPS. The more the kynurenine 3 oxygenase will be there, more quinolonic acid will form, and that further result into neurodegeneration and neuroinflammation. I hope everyone got this particular mechanism, how particularly LPS is responsible to cause neurodegeneration. There are different ways by which we can manage the neurological disorders with the gut dysbiosis, which are associated with the gut dysbiosis, with the ketogenic diet, functional food, antibiotic. Probably you know one functional food is there, Yakut, which is a probiotic. So probiotics are being used over there. Probiotics and prebiotic are the two terminology. Probiotic means usually alive substances are there, like gut microbiota itself, some microorganism we will add into a particular food, and that food, if we are taking, it will be help for maintaining the gut biosis, which is nothing but Yakut, like your cheese, like your yogurt, like your butter. These all are contain lactobacillus, which act as a probiotic for our gut. Whereas prebiotic are the chemical which are been present in the food materials, like it is present in the different banana, apple, garlic, onion. These prebiotic are actually help the probiotic to grow in a balanced manner, so that we will have a better effect of probiotics. So we will discuss with the prebiotic. Prebiotic is having a function that it changes the intestinal microbial flora concentration, means that disbalance which had occurred, it will balance that. Another one, it inhibit the carcinogens which are been formed inside the body. It is having an effect over the immunomodulation. It improves the nutrients absorption, specifically vitamins absorption, and it inhibit the pathogens. This all result into ultimately, if you will see, it maintain the gut flow microbiota, and which help in the health benefits. There are different phytochemicals which has a role as a prebiotics, like polyphenols are the secondary metabolite which are present in number of plant and food materials like coca, coffee, dry legumes, fruits like apples, berry, nuts, olives, and some vegetables like cabbage, tea, wine, where polyphenols are been present over there. These polyphenols absorb very small in a small quantity from the intestine, and that's why they are been helpful for the growth of microbiota, because they less absorption, they will be reside over the local area of the gut. And these polyphenols are been proved to have a beneficial effect over the flora. There are four main group of the polyphenols like flavonoids, phenolic acid, stilbenoid, and lignans. Phenolic berry extract inhibits the growth of gram-negative bacteria. Means berry extract, if you will take into particular food material, or in case of any functional food, we will add phenolic berries. Berry extract. It is having a function that it reduces the growth of gram-negative bacteria, and it can increase the growth of gram-positive bacteria. It has been well proved over there, and that's why berry extract could be uh, one of the content that we can use to use as an a nutraceutical for the management of neurodegenerative disorder as a prebiotic. Another one is the inulin, a compound which is extracted from the chicory roots. It is a fructans, which is a great importance in the commercial purpose, and is proven to have a prebiotic function. If you'll see inulin, it has been present in number of food materials like yogurt, cereals, dessert, nutritional bars, beverages, ice cream, and other. You will observe that a table has been given over there. The concentration of inulin per 100 gram of that particular thing. You can see onion pulp is also containing 1 to 7 gram approximately per 100 gram of pulp of onion. Like that, in the different food material, different plant products, we can get this particular inulin, which is having a function that it can act as a prebiotic. Oligosaccharide, which is another non-digestible carbohydrate, dietary fibers is there. Oligosaccharides are found in number of food products like milk, honey, sugarcane juice, soya bean, mustard, fruits, vegetables like onion, asparagus, sugar beets. Like that, number of boom shoots, tomato, wheat, like that, different food materials in which we can observe oligosaccharide is present over there, and oligosaccharide is present in the different form also. As isomalta oligosaccharide is there, fructo oligosaccharide is there. Like that, different types of oligosaccharides are present over there, and the researcher had been found that these oligosaccharides are having beneficial effect over the gut microbiota, and specifically research has been suggesting that the oligosaccharide is having a role over the gram-negative bacteria's growth, that many of them are reduces the gram-negative bacteria's growth, and many of them will increase the gram-positive bacteria's role. So if we will add into the probiotic a prebiotic which can increase the growth of gram-positive bacteria, we can treat the neurodegenerative disorder or neurological disorder which are being associated with the gut dysbiosis. Another one is a polyunsaturated fatty acid. There are different rich polyunsaturated fatty acid diet which improves the immune function, blood pressure, cholesterol, triglyceride level, and cardiovascular function. In humans, it has been observed that the lactobacillus, which has been present in our curd, yogurt, the lactobacillus, which is a probiotic, it enhances its adhesion with the gut, is been increased with the polyunsaturated fatty acid, and that's why it is useful for the treatment of gut-brain axis associated neurological disorder. Soya isoflavone is also there. It has been observed that soya isoflavone, specifically, if we have to say that diatzin is the one of the major content of it. Diatzin has been well proven have an effect over the gram-positive bacteria, that it enhances the growth of gram-positive bacteria. Another one is the anthocyanin, which has been present in the number of vegetables and fruits like blueberry, raspberry, cauliflowers. Anthocyanin is also present in the potato, black rice, and number of chemical substances. It has been present. It has been well proved that anthocyanin is having an effect over the bifidobacteria, effect over the growth of gram-positive bacteria like bifidobacterium, lactobacillus species. By these particular studies, we had observed that if we will add prebiotic into a particular food substances, then we can able to manage a number of diseases, specifically the diseases which are been associated with the gut-brain axis dysbiosis. Thank you, ma'am, for patience listening. Herewith, I conclude my topic.
Thank you very much, sir.
Thank you,
It's
sir.
Dr. Arpit here.
Yeah, thank you, sir.
Sir, for enlightening it with your knowledge. And I thank you very much for accepting our invitation on short notice. Thank
And
you.
I'm also very thankful, sir, for giving me an opportunity to present my topics, my lecture in the international conference. Thank you, everyone.
Thank you.
Thank you so much, sir.
Thank you.
Thanks a lot.
I hope I am audible. Dr. Karun, sir.
Ma'am, hello, ma'am.
Hello. Sir, are you there?
Yes. Hello, Dr. Arpit here, ma'am. It will take 10 minutes for him to join. Dr. Karun will take 10 minutes. Okay.
Okay, so shall we start the session, sir? Arpit, sir?
हाँ मैम, yes. मैम सभी oral presentee को join करा देते हैं. Sir will take 10 minutes.
10 minutes.
Yes, ma'am.
Okay. Okay, so we'll do one thing. We will call out the names of the oral presentees so that they can be
Yes.
ready with their PPTs.
Yes. Yes, yes, ma'am.
Okay.
Okay, ma'am.
Oral presentation students who are online. Hello, can you all hear me?
Yes, ma'am.
Okay, so I'm taking
Yes,
the
ma'am.
names of students who will be presenting in this session. First, it will be Pooja.
Yes, ma'am.
Second, it is Smita or Sudar.
Shweta, ma'am.
One second. Shweta.
Yes, ma'am.
Then Varshini. Varshini Devi, are you here? Varshini Devi.
Ma'am.
Varshini Devi, can you hear me? Are you online?
Yeah, ma'am, I can hear you.
Varshini Devi. Varshini. Okay, then the fourth one is Darshini B. Are you here, Darshini? Darshini.
Yes, ma'am. Yes, ma'am, I'm here.
Okay, next is
Yeah.
Meili. change. Mavli, Maivli.
Present, ma'am. Present, ma'am.
Okay, so I'm repeating the names once again. Pooja Nandini, Shweta, Varshini Devi, Darshini, Mavli, then Kalaivani.
Yes, ma'am.
Kalaivani.
Yes, ma'am. Here, ma'am.
Okay, Irene Varghese. Irene Varghese, are you online? Irene Varghese. Miss Irene Varghese, are you online? Dr. Anuradha Balda. Dr. Anuradha Balda, are you online? Dr. Abhimanyu Sharma. Dr. Abhimanyu Sharma. Dr. Ashok Kumar Yadav.
Yes, ma'am. I am available.
Okay, sir. Then Dr. Deepti Singh. Dr. Deepti Singh. Nikki Bhandari. Miss Nikki Bhandari. Miss Nikki Bhandari. Miss Krishna Kumari.
Yes, present, ma'am.
Ma'am, Ankur Agarwal.
He was unable to join.
Ma'am, Ankur Agarwal present here.
Who is present?
Ankur Agarwal.
Ankur, okay, let me see whether you are in this slot or not. Okay.
Yes, ma'am. Yes, ma'am.
And Krishna Kumari, is she here?
Yes, ma'am. I'm here only.
Okay, and Nikki Bhandari? Nikki Bhandari? Nikki Bhandari, no. Ankur, your name is here, okay? Ankur, your name is here.
Yes, ma'am.
Then Manisha? Kaushik?
Yes, ma'am.
Then Asmita? Dhruv? Okay, so many are absent. Arpit sir, can you hear me? Hello, Arpit sir, can you hear me? Okay, everyone, please wait for a few minutes for the session chair to join. He's going to join and then we'll start the session, okay? And
Hello.
everyone, yes.
Hello. Ma'am, I have a doubt, ma'am. Is my name is there like Punit Gupta?
What is your name, please? Repeat.
Punit Gupta. Punit Gupta. PUNIT.
No. No.
Ma'am, Devanshu
I'm
Sharma.
calling the names once again. Pooja Nandini, Shweta, then Varshini Devi, Darshini, Mavli, Kalaivani, Irene Varghese, Anuradha, Abhimanyu, Ashok Kumar Yadav, Deepti Singh, Ankur, Manisha, then Kaushik, Asmita, Dhruv, Nikki Bhandari, and Krishna Kumari.
Ma'am, I want to know, is my name there? Chahat, ma'am.
Chahat, yours is tomorrow morning.
Tomorrow morning.
And ma'am, Devanshu Sharma.
What is your name?
Devanshu Sharma.
Devashish.
Devanshu Sharma.
Devanshu tomorrow. Devanshu again tomorrow.
Okay, ma'am.
So,
So,
this side.
Ma'am, Nikki Bhandari is present.
Nikki Bhandari is present. Okay, fine.
Ma'am, Sneha this side, ma'am. My name is also not there.
Sneha Prabha, are you here?
Yes, ma'am, I'm here.
Sneha Prabha, yours is today, okay?
Yes, ma'am.
Ma'am, Punit Gupta. Tanish Khandelwal. Ma'am, Tanish Khandelwal. I'm also.
What is your name?
Tanishk Khandelwal.
Tanish Khandelwal tomorrow. Okay, do one thing. Can you Tanish Khandelwal and then
Ma'am, Punit.
Devanshu, Tanishk Khandelwal and Devanshu.
yes, ma'am.
Yes, ma'am. Yes,
You
ma'am.
both of you, can you stay till the end?
Yeah, ma'am.
Okay. Okay. If you can stay till the end, then we will take your presentation at the end. Okay. Okay, ma'am. Okay. Okay.
Ma'am,
Okay then.
I also want to ask like my name, Punit Gupta.
Puneet Gupta. Your name is not here, beta, not here. And Puneet Gupta, it is tomorrow actually. But yes, if you are online, then we can take today itself. But
Okay, ma'am.
we'll have to wait till the end.
Okay, ma'am. Okay, ma'am.
Okay,
All
so
right.
Puneet Gupta, Tanishq, and Devanshu.
Ma'am,
Yes, ma'am.
Chahat, can you wait up to the end?
Online, everyone online. Nobody offline now. Okay, so these three students also they can join, but these three will be taken at the end. Okay.
Ma'am, my meeting is scheduled
Okay,
for
ma'am.
tomorrow morning. Can I wait up to the end? Ma'am, Chahat.
Not able to hear you properly. May I know who is speaking?
Ma'am, this is Chahat. Can I wait up to the end for the meeting?
Chahat.
Yes, ma'am.
Chahat, do you want to give it today?
Yes, ma'am.
Okay, so you are also here online?
Yes, ma'am.
Okay, so we'll do one thing. We'll keep you also at the end. Okay.
Okay, ma'am.
So you have to, hang around because it will be a little bit late because we are starting the session late, so it will be a bit late. So any of you who are taking MET bus in any of these names which we have taken right now, who are going to join today, whose name was given as for tomorrow. Chahat, Puneet, Devanshu, and Tanishq. Any of you taking the bus?
Yes, ma'am.
Yes, ma'am.
No, ma'am.
No,
All of you will
net-
be online,
No,
keeping
ma'am. No,
online,
ma'am.
right? Yes, ma'am. We are online. Okay, so we'll continue your sessions after we finish the ones which are giving first now. Okay? Okay, ma'am.
Okay,
Hang around
ma'am.
for a longer time. Okay?
Okay.
Okay then. Thank you. Sir को share करेंगे. Give it to me. Bye-bye. So all the participants kindly wait for a few minutes and then we will start. Okay, we'll announce when we start. Just wait for five minutes.
Hello. Moving on further, I would now like to introduce our session chair for this session, Dr. Karun Chandalia, who has joined virtually. So from 2015 to 2021, Sir served as the Managing Director of Pali Milk Union. He achieved significant milestones, including establishing a complete cold chain in the milk procurement system through the installation of bulk milk coolers at dairy cooperative societies. He also introduced electronic testing systems and online monitoring at all societies, enhancing transparency and profitability. The dairy plant's processing capacity was expanded from 0.6 lakh liters to 2.5 lakh liters per day, while milk sales grew from 38 KL to 65 KL per day. Under his leadership, the unit became the second largest seller of ghee in the RDF across Rajasthan and also introduced a new range of indigenous milk products and desserts. Sir successfully implemented food safety and quality management systems and also transformed the unit's financial standing from a position of loss to accumulated profits exceeding 10 crores. Prior to this, as Managing Director of Jodhpur Milk Union, he achieved a more than 100% increase in milk procurement, expanded sales of milk, ghee, and other products, and also oversaw the complete renovation of the manufacturing unit by implementing key quality and safety systems. His earlier roles at Udaipur Milk Union from 1995 to 2013 and RCDF Limited 1984 to 1995 involved leadership in plant operations, quality control, marketing, finance, and inventory management. Additionally, he also holds key positions in professional networks such as Secretary of the Indian Dairy Association Rajasthan chapter and President of the College of Dairy and Food Technology Alumni Association. As a visiting faculty member for several institutions, he has extensive teaching experience in dairy technology, food technology, and also management. His key skills include the rehabilitation of sick units, financial and administrative management, production and project management, food safety management, and quality management systems, where he also serves as a lead auditor for ISO 9000 and ISO 22000 certifications. With these few words, I would now extend a floral welcome to Sir for this virtual session. Dr. Karun, sir.
Thank you. Thank you. I am here.
Thank you so much, sir. Yes. We are now handing over the session to Dr. Arpit as we are starting with the oral presentations.
Thank you.
Thank you, sir, for joining us. Thank you so much.
Thank you. Thank you.
Thank you very much, sir, for joining. And now I would like to request the first participant to please start the presentation. Dr. Vijay, can you please announce the name of the first participant?
Yeah. Let me just stop the screen sharing so that they can share.
Okay. Okay, ma'am.
Yes, I have stopped the screen sharing. Okay. And the first participant is Pooja Nandini. Pooja Janani.
Pooja Janani, yes, ma'am, I'll be
Janani,
sharing.
yes, sorry. Not Pooja Nandini, Pooja Janani.
Okay. Pooja, you just have 5 minutes. Okay. Just concentrate
Okay,
on
sir.
your work. Okay. And your
Okay, sir.
conclusion.
Okay,
Okay.
okay, sir.
Please go ahead, Pooja.
Okay, thank you, ma'am. So, good evening to everyone. Myself, Pooja Janani from Congo Engineering College. Currently, I'm pursuing Masters in Food Technology, and my project work has been guided by Dr. A. Sudha. So, the title of the project is Development of Fig Ice Cream, a novel dessert with ancient roots. So, the main aim and objective of this product is to develop an unique ice cream flavor using fig powder. Since the fig powder has sweetness, it will also be partially replacing the added sugar. And also fig is an seasonal fruit. So by making it as a powder and storing it, we could utilize it in throughout the year. And also the health benefits of the fig fruit will be utilized by this. So first, the fig fruit is made into powder by collecting the fruits and considering the feasibilities. The fig powder has been sun dried for 1 week, and also tray dryer has also been used, and it has been sieved, grinded and sieved. So this is the basic ice cream processing method. By using this method, both the control sample and the treated sample were made. So when it comes to the processing factors, aging time and blending speed has been considered. So these are the preliminary trials which have been done. So as a stabilizer, carboxyl methyl cellulose were taken, and as emulsifier, glycerol monostearate were taken. Here only the percentage of added sugar and fig powder has been varied. So with the help of 9-point hedonic scale, the sensory evaluation was conducted, and the trial F1 has been chosen. So with the help of the preliminary trials, the temperature has been optimized. So the reason for optimizing the temperature is because when we add fig powder in milk, sometimes it causes curdling. So to overcome that issues, the temperature has been optimized. So next, the texture were analyzed. That is, the hotness and the adhesiveness of the ice cream has been analyzed using spherical probe. And overrun of the ice cream has been analyzed. That is how much air has been incorporated while churning before freezing. So in the result and conclusion that when we add fig powder, the temperature should be optimized to overcome the curdling issues, and the hotness of and the adhesiveness of the ice cream depends on the ice crystal formation. And by optimizing the processing factors, that is blending speed and the aging factor, we could analyze the texture and overrun of the ice cream. So I would like to acknowledge my sincere thanks to Congo Engineering College and my sincere thanks to Dr. A. Sudha ma'am for her support and guidance throughout my project. Thank you. Sir? Am I audible, sir?
Yes, yes.
Yes. Yes, Pooja.
Yes, sir. Sir, if you have any questions, please do ask.
What is this temperature optimization?
So, when fig powder is added in milk, the temperature should be kept in 50 degrees Celsius so that we can avoid curdling off.
Before that, have you pasteurized the ice cream mix?
So it is actually a pasteurized milk only I have taken. Already it has been pasteurized.
Now, why do you think that it will curdle the milk?
So since the fig powder has acidity, that will lower the pH of the milk.
If how much quantity you are adding to this fig powder? Fig powder, how much quantity of fig powder you are adding?
So trial F1 has been chosen. So fig powder has been added in 15 gram.
15 gram.
Yes, sir.
What is the resultant acidity?
So when we kept in 50 degrees Celsius, there is no curdling, so.
No, I am just asking that you are adding fig powder to the milk. Okay.
Yes, sir.
And naturally, if you say that it is acidic in nature, fig powder, then
Yes, sir.
it will increase the acidity of milk. है ना?
Yes.
वेरी
Yes, sir.
सिंपल। ठीक है, अगर हाई एसिडिटी का कोई भी अपन इसमें इंग्रेडिएंट डालेंगे तो एसिडिटी बढ़ेगी, घबराने का
Yes,
बात
sir.
नहीं। मैं आई एम आस्किंग फॉर माय इंफॉर्मेशन ऑल्सो, है ना।
Okay, okay, sir.
तो कितना बड़ा एसिडिटी
Sir, I haven't
नोट
checked
किया
that.
गया?
So, no,
Note-
sir. I'll work. Okay, I'll
Okay.
work on that, sir.
Okay. Secondly, we are calling it ice cream, है ना।
Yes, yes, sir.
आइसक्रीम का मिनिमम फैट परसेंटेज कितना होता है, वो कैसे मेंटेन
Sir,
करोगे?
sir, I have taken 6% of fat and 9 gram, 6 gram of fat and 9 gram or 9% of SNF. The
Anyway,
milk which I have taken.
it is a very good your presentation, but
Yes.
my advice is go through the standards of ice cream required,
Okay, okay, sir.
है ना. But
Okay,
yes,
sir.
you are using fig, it is very good. Okay,
Thank you,
you.
sir. Thank you so much, sir.
Okay. कैमरा तो मैंने
Okay, Shweta, your screen is visible now, so you can proceed.
Okay, sir. Sir, am I audible?
Yes, you are audible.
Okay, sir.
Please share your PPT in full screen mode, slideshow mode, and
Okay, sir.
start presenting.
Okay, sir. Good evening to all. Myself Shweta. I'm from Kongu Engineering College, and my topic is studies on drying characteristic of amaranthus polypnoid. It can be guided by Johnny Blessing Manoj, Assistant Professor of Kongu Engineering College. So the aim of this project is to study the drying characteristic of amaranth. The objective is to study the drying characteristic of amaranthus polypnoids by using different methods, that is microwave drying, fluidized bed drying, and tray drying, to find out the best dryer for production of dried amaranthus polypnoid based on the yield of bioactive compounds, that is antioxidant, polyphenol, and flavonoid. The scope is utilization of low cost and nutritious amaranthus, and extension of shelf life of the amaranth, and also amaranth powder can be added to any food substances. So the introduction is drying is a crucial process for preservation and enhancement of shelf life. It reduces the moisture content and water activity of the amaranth, and it helps to increase the shelf life and reduce the microbial growth and spoilage. So it is easy for transportation and storage too. And amaranth polypnoids is a tropical amaranth. It also called as a tropical amaranth. It is a nutrition-rich leaves. So this is a methodology of the project, that is amaranthus polypnoids has been dried by using tray dryer, microwave drying, and fluidized bed, and the dried sample has been powdered, and the bioactive compound has been analyzed for the powder form, and the optimized sample has been selected. Okay, these are the drying rate curves for microwave drying, and it is for fluidized bed drying, and for tray, for microwave and fluidized, and also tray. The temperature for tray and fluidized is same, that is 40 to 60, and for microwave we have used 5 different watts.
And these are the readings of tray, fluidized bed, and microwave drying of color values, that is yellowness index, whiteness index, LA, B, del E values, and the water activity and moisture content of the sample. And these are the values of total polyphenol content and flavonoid content and antioxidant content of the samples. So by consolidating the samples, we have come to the conclusion that is the microwave dryer at 180 watt seems to has the good color value, and but as we compare the total polyphenol, flavonoid, and antioxidant content, we have microwave drying at 450 watt has the good color, moisture, and bioactive compounds range. So we have considered 450 watt is the optimized best sample. Thank you, sir. Sir?
Okay. Okay. Can I ask you something? Why you have chosen fluidized bed drying?
Because I'm done the comparative studies on tray and fluidized. In India, tray drying and fluidized bed is used. For tray dryer is suitable for farmers, and fluidized is best for industrial process. So I have selected fluidized.
Okay. Sir, do you have any questions?
She's talking about shelf life. है ना?
Yes.
Yes.
Shelf life, if we compare this tray drying and this microwave drying, है ना?
Yes,
तो
sir.
इसमें आपको माइक्रोवेव ड्राइंग में कितना फर्क पड़ रहा है?
What, sir?
What is the difference?
Difference of shelf life when we dry this amaranthus and tray drying versus microwave drying.
Yes, sir. Yes, sir.
What is the difference? I think you have tabulated it.
Actually, the moisture content and
Shelf life. We are talking about shelf life, no?
Sir, I didn't understand. Can you tell me?
Shelf life. What is shelf life? Do you understand?
Actually, sir, I didn't study the shelf life, accelerated shelf life study. But as we compared with the fresh.
What was your objective then?
Sir?
Objective of your experiment?
It is to optimize the best sample by
To
the
study
yield
the
of
drying
bioactive.
characteristics of amaranth.
Yes, sir.
So you have not gone for this shelf life study. Right? Drying, crucial process for preservation and enhancement of shelf life. So you have not studied the difference. Okay. So, then why should we go for this microwave drying? Any purpose?
So because it has the high yield of bioactive compounds, that is called high yield of flavonoid, polyphenol, and antioxidant range as compared with the tray and fluidized bed, right? So,
What about efficiency? Efficiency of drying? In which process it is better?
so microwave is best, sir. Microwave 450 watt is considered as best because it has the high retention of these compounds. So,
Nowadays, people say that this we should avoid using microwave in edible products. Nowadays, people say that we should avoid using microwave drying for direct edible products. माइक्रोवेव यूज करने के लिए अभी मना करते हैं कि कम किया करो. Can we think for solar drying, solar tunnel drying?
And also, I think vacuum drying can be done of that.
Yes.
That was the best because you can keep the color also intact in that vacuum drying. Okay. So We just
Okay,
have.
very good presentation. Thank you.
Thank
Thank you,
Thank
sir.
you.
Thank you, sir.
Next, the Varshini Devi.
Yes, sir.
Okay. So be ready with your presentation.
Yeah.
Yes, PPT is visible. Yes,
Am I
start
audible?
presenting.
Yeah.
Yes.
So my project is about development and career pulse.
Varshini, you are not audible.
Sir.
Hello. Next, Dharshini. Please be ready. I think she is having some issues.
Sir, am I audible?
Yes, but your voice is distorting. It's not clear.
Yeah, 1 minute.
There is network issue, I think. Next, Dharshini. Varshini, can you please join afterwards?
Yeah, sir.
Okay. Next, Dharshini is online. Dharshini.
Yes, sir. She's in.
Okay. Dharshini, please. Can you share your screen? Varshini, just stop presenting.
Yeah, sure, sir.
Dharshini, please be ready with your PPT. Dharshini, can you hear me?
Yes, sir. I can hear you, sir.
Okay, so please present now.
Okay, sir. So is my screen visible?
Yes, it is visible. Please put it in slideshow mode and start presenting.
Good evening, everyone. My name is Darshini. I'm from Kongi Engineering College, Department of Food Technology. My topic is about development and optimization of fermented oat milk using water kefir grains as a functional milkshake, which was guided by Mr. M.S. Shivaswamy, Kongi Engineering College. The aim of this topic is to develop and optimize a fermented oat milkshake using water kefir grains for a fermentation process that is rich in probiotics, protein, with improved texture and flavor and potential health benefits for gut health and immunity. Scope meets the rising demand for a dairy alternative, acts as a functional beverage. Water kefir grains advances fermentation technology by offering new applications in food processing and microbial fermentation. It supports sustainable food production practices. Objective: to develop fermented oat milk using water kefir grains to study the physiochemical properties and sensory evaluation of the fermented oat milk
to develop milkshake using fermented oat milk by using response surface methodology. Introduction. Like water kefir grain is a cluster of yeast, microorganism, and enzymes which is used for fermentation, which is used in this process. The study focuses on developing and optimizing a novel fermented milkshake using water kefir grains, which finding aim to contribute to a sustainable plant-based milkshake optional that aligns with consumer preference for healthier, innovative beverages. This is the methodology where the oat is soaked and drained, where oat milk is extracted.
Darshini, can you please go to your results and conclusion?
Okay, sir.
We are running out of time.
Okay, sir.
So it will be better if you focus on your results.
Okay, sir. This was the result where the fermented oat milkshake is stored in a period of time like day 1, day 7, and day 14. These are the analysis where pH and titratable acidity are interlinked, where pH is increases where the titratable acidity lowers, where viscosity starts to decrease day by day, and proteins and carbohydrates are also has slight changes. Therefore, day 1 milk is chosen for preparing milkshake, where RSM this was the process which was involved. Then using RSM, milkshake was prepared, and this was the 3D surface graph where which I'll discuss now, where the this analysis reveals significant interaction between cocoa powder and peanut powder across several factors like viscosity, pH, antioxidant. Like intense changes in the cocoa powder concentration can alter viscosity, pH, and antioxidant activity, but the magnitude of these changes is influenced by the amount of peanut powder. Therefore, the interaction is crucial for optimizing the product formulation to achieve desired characteristics. This was the result, like using RSM, the desirability of was 0.938, and the optimized product amount was cocoa powder 5 gram, peanut powder 2 gram, and xanthan gum was 0.1 gram. This was blended with 150 ml of fermented oat milk, and preservative adders was sodium benzoate, that was 0.1 gram. Thank you, sir. Hello, sir.
Hello. Yes, sir, please ask some questions.
You just defined what is a response surface methodology.
So it's a methodology where we can do different types of trials, where in some places we need to do more trials. Instead of that, this software helps us to do minimal trials and gives us an optimized products by evaluating and giving graphs.
Any details about this methodology?
So here I use central composite design, sir. In this, we can vary 2 ingredients and we can give more responses. Then it gives more runs.
Okay, go ahead. Thank you.
Okay, Darshini, one question from me.
Okay,
What
sir.
is difference between box bank and this CCT design?
In central composite design, we can alter 2 ingredients, and box Benton we can vary 3 ingredients.
Okay. Okay, thank you.
Thank you, sir.
Hello. Hello. Next participant, Mavli Chandrasekhar.
Okay, sir. Firstly, are you doing the presentation? So
Please
I'll
start
start.
presenting.
Okay, I'll start.
Yes, it's visible. Start presenting.
Okay. Good evening, sir. So my topic is about the development of the closure pattern prefix and evaluation of its quality parameters. Prefix, according to the SSI, is nothing but it's characterized under the food products that require further preparation for consumption. So it's a standard flow that's been here. It is also a composite flow that's been used for the making of the dosa. So according to FSS, so the mixed flow is the product that's been obtained by heating, grinding, and the blend millets or the pseudomonads or by breaking the flow applied from the treating beds. So here the objective is to formulate the dosa pattern prefix and analyze its quality parameters. The method here that is being used is that the raw materials has been bought as crop, dehulled pearl millet and the dehulled black flour that is being taken and it is first seen with the picture of high quality and then it is being guided individually flows. According to the FSSC, the mixed inlet flow to size of about 45 meters, right? Here we have been seeing to the size of 40 meters to get the uniform size of the flow. So here, according to the trial, ratio according to that. So the multiple trials have been conducted to know about the constitution of the data. Here the flows have been taken and a pinch of salt for about 50 was done and 7 trials were made. From this, what we understood was when the background is being in a lesser amount, we didn't get the consistency of the dosa and the buckwheat flour, the smell was more dominant. The higher amount of the background flour to about 50 gram per 50 gram gave a good ratio of the dosa consistency, and also it was the right proportion rather than a very higher amount of the bucket flour or ball millet. The right ratio of the background flour, the right consistency for the spread of the batter. That is that trial 1 and 2 did not give a good picture as the diagram is very less. And also the smell of the bucket is very strong. In trial 3, 4, 5, there also was a consistent shape and it was also preferable, but still we could see that the smell of the bucket floor was more dominant. And when the background is lesser and also the consistency of the smell was completely so in the trial 6 and 7, the amount of the background was significantly and it gave a good spread in the good consistency. So the analysis was made with the iron, that is infrared analyzer for finding the moisture content. The color spectrophotometer was used for knowing the color of the flow. So since the buckwheat is being used here, the color could in slight with slight compared control that was the rice butter flow. Since why rice flow was used because that is the high so since the bucket has the higher amount of that had a slight lightness in its color, it was more preferable compared to other. Then the water activity was analyzed using the portable water activity analyzer. The pH was seen basically for the batter to know the pH which should play a vital role to understand the fermentation of the batter. So the results were being analyzed where the moisture was in a very lesser amount since the composite flow is about 8 to 10% of it. We could find that the moisture content was between 6 to 8, which is more preferable for the flow and also and the lightness of the color of the floor was also lesser, and the difference, the color difference was little greater than 6, but still find that it similar with the control rice butter flow. And what happened was also about 0.6, 0.5, and it also denotes the shelf life. And the pH was seen about 6.28. So when added curd for in a 1 spoon of curd, we could find that in 1 hour there is a change in the pH for about 5.25 to 5.2, which is a reasonable range for ferment of the dosa and also the acceptable one was for the ingredients of 18 grams bucket flour, 1 minute flour of about 50 grams, and back gram flour of 50 grams. And also it had a very good taste, and the smell of the buckwheat and the quality was not much dominant in this trial one, and that was being used for the optimization. So the minimum and maximum range of the moisture content was between 5.2 to 10.6. And also the black gram this from this level we can understand that the matches and also it is very lesser amount of the moisture content was being identified in the flow that was being used for the project, and we could see that moisture content could lead to spoilage and the multi-degradation. So this also proves that ours was
Lovely.
very lesser.
Just go to your results. Just summarize. Your time is gone. Okay, time is already.
Yes, sir, that's it. So I we extend the thanks to our competition for some stars.
I think there should be a graph of the results. What you have shown. Okay, so a graph should be there or a table should be there. So it should not. It is not presented in a very good form.
Okay.
Sir, do you have any question?
No, it's okay.
Okay.
Yeah, thanks.
Calavini.
Hello. Sir, Vashni Devi is presenting right now, sir.
Okay, you can continue.
Am I audible to you, sir, right now?
Yes, you are audible.
Yeah. So my project is support development and optimization of career paths using Ashwagandha. This project is guided by Mr. M.S. Shivaswamy, Assistant Professor, Senior Department of Food Technology, from the the main project is to develop Ashwagandha career pairs. So it is a herbal leaves. So it's a herbal based now. Higher amount of phytochemicals in it, so it and it acts both prebiotics and probiotics, so that it can improve the gut health and as it boosts the health benefits. And by interacting with other ingredients, it comes to the customized option based on the consumer health. We can go with the various targeted formulation from this chemicals.
Can you go on your results?
Yeah, sure, sir. So in this project, I have varied the sodium management. So the main principle of this project is about a purification process, which involves in the preparation the development of the caviar pulse. So in this I have varied the sodium alginate, which is the level of 0.2 to 0.5 gram. So how the sodium alginate is, it has a gelling characteristic. So how it is affecting the caviar property. So that thing I have studied. For that, first I have conducted the extraction of ashwagandha from ashwagandha root powder, which is about 5 grams of ashwagandha powder, and with the 50 ml of water extraction, I have gone with the ultrasonication bath in order to get a higher yield and with a higher amount of phytochemicals. And comes to the pH value, the caviar can able to form with the pH range of which is more than 4 and in between 6. Comes to the extraction. Before extraction, the pH value is around 5.90, and after it is about 5.84 the extraction from the extraction of ashwagandha. And after that, formation of the caviar pulse with the trial 1, 2, and 3 and 4. Comes to that, we can able to in this picture itself, we can able to see before blending, these are the 4 trials, and after blending, we can able to see how the gelling characteristic affect that liquid. And here the images of the different trials. So trial 1 and trial 4 doesn't have that much. It doesn't have that much effect on that. Sorry, it has more effect on that caviar pulse, because in the first trial, which is of 0.2 gram of sodium alginate, the formation of the caviar pulse is the size is very smaller when compared to the trial 2 and 3, and comes to the trial 4 with the sodium alginate of 0.5 gram. The size is getting bigger, as well as the texture is so hard, and it is elongated in shape. It is
ये पानी का नहीं करता आपका ही
मैंने like it.
Sir, am I audible?
You can continue.
Yeah,
Yes.
yeah, sure, yeah. So you can be able to see the size, which is ranges from 0.5 to 1.7. That is sample 1 to sample 4, and in the sample 2 and sample 3. So in this project, I have gone with the basic verification process. So I have aimed for the smaller size of caviar pearls. So the sample 2 and the sample 3, which has a spherical in shape, and comes to the sample 4, which is elongated in shape, and the sizes ranges from 1.5 to 1.7. And comes to the analysis, I have gone with the color analysis and texture analysis. From the previous result, we can able to see that sample 2 and sample 3 has a good shape and sizes. So that I have gone with the texture studies for texture and color studies for sample 2 and 3. So in the color analysis, I have done for sample 2 and 3, and for texture analysis, I have gone with sample 2 and 3 in the hardness. Awesomeness, gumminess, and springiness. And the next is storage of caviar pearls. So I have stored the caviar pearls in different temperature, that is room temperature, and in refrigerator room temperature open condition, and another one is closed in a closed container. The next thing is I have stored the caviar pearls in a sugar solution, so we can able to compared with that open and like normal temperature when kept in refrigerated temperature. And the next one is refrigerated temperature with a sugar solution. It shows that the caviar pearls in this sugar solution with the Brix value of 22 has a shelf life which is more than 1 week when compared to the normal condition and the refrigerated condition. Comes to the discussion, the pearl characteristic. So based on the size and shape, I can able to conclude that sample 2 with the 0.3 gram of sodium alginate exhibited the most favorable shape and size. And comes to the analytical evaluation of sample 2 and 3, the color and texture properties shows that sample 3 has a hardness which is more than that of sample 2, and the storage stability, so that room temperature in refrigeration and in sugar solution under refrigeration condition. The caviar stored in sugar solution maintain the quality for over 1 week. And so from these tests, we can able to conclude that sample 2 demonstrated an optimal balance of textural properties and shape. And the highlights are the potential of ashwagandha in functional caviar pearls, and how sodium alginate concentration influence the physical properties and overall quality of the sir, that's it.
Okay, thank you. Similar suggestion for you. I think tabular form should be better. Okay.
Okay. Yeah,
Okay,
thanks.
for presenting the results. Okay, of that color and all.
Yeah, sure, sir.
Irene Varghese.
Sir, Kalaivani is here, sir.
Kalavani. Yes. Okay, Kalavani.
Okay, I am presenting right now.
Okay.
Sir, is my screen visible? Is my screen visible, sir?
No, not yet, Kalaivandi. Please start. And second, maybe you can do this way. You can unshare and share again. Okay?
Yes, ma'am. Yes, ma'am.
Yeah, most of the time then it works.
Is it visible, ma'am?
No, it's still not visible.
You're presenting from
Also,
phone, Kalaivandi. Yeah, it is your screen of your phone is visible now, but not the PPT. So please share your PPT. Yeah, it's visible. Go ahead, please.
okay, ma'am. Okay, my topic is development of dark chocolate by incorporating spirulina achar. It's guided by Dr. V. Chitra Devi, Professor, Department of Technology, Congress Engineering College. So it's a small introduction about the dark chocolate. We all know that dark chocolate is nutritionist. It contains antioxidants, anti-radical, anti-carcinogenic properties. And we have incorporated the spirulina achar, and blue-green algae. It contains about 60 to 70% of protein. And here we are incorporated the spirulina char in the dark chocolate. Preparation of spirulina char. The spirulina char is prepared by hydrothermal carbonization of spirulina powder with water. It is loaded in the autoclave reactor with water in the ratio of 1:10 and maintained the heat for 30 minutes in 250 degrees Celsius. And at least 10 gram of spirulina powder yields up to 6 gram of char. And this is the methodology. After obtaining the spirulina char, we are grinding it in a mortar and pestle, and it's a usual chocolate preparation methods: double boiling, melting of butter, addition of dry ingredients, and mixing well it, and again grinding of the chocolate and molding and freezing it for 24 hours and demolding. It's the optimized ratio. We have obtained the optimized ratio by performing several trials. On the basis of sensory analysis, we have taken this ratio, and this is the images of the sample and the control, where the control doesn't contain any spirulina char, and the sample contains approximately 1% of spirulina char. And this is the analysis we have done. The texture analysis and the parameters goes by the probe used is needle probe and pre-test speed 1 mm per second, test speed 1.1 mm per second, post-test speed 5 mm per second. And the instrument we have used is a plus connect stable microsystems, and the size of the sample is 2.5 centimeter into 1.8 centimeter, 0.5 centimeter. Sample type chocolate, model penetration test method, and it's the color
Can you go
analysis.
to your results, please?
Yes, sir. Okay, these are the results for the texture analysis. The values for different tests such as hardness, springiness, cohesiveness, gumminess, and cheviness are obtained. So for the sample one, and trials are done in triplicates. For the first trial and the second trial, the hardness values and other values are similar in and around the range. But in the third trial, we can see that there is a drastic change in the hardness and other values. So the interpretation goes by the change in the other values is due to the melting of chocolate in room temperature. Since the chocolate does not undergo the process called tempering, so it melts faster in room temperature. So for that reason, we have stated the references such as dark chocolate stored at a higher temperatures, above than 20 degrees Celsius, starts to melt, and it should be stored in 6 to 12 degrees Celsius. And proper melting only results in the hardening of chocolate. And next here goes by the color analysis and sensitivity analysis. Here color same we have done duplicates and the values are presented. So the L value, the lower L value indicates that the sample is darker in color, and the flavonoids and the other substances in dark chocolate such as flavonoids reduces the colors of red and green in the chocolate. And here the sensory analysis is described in a graph. We can see that the sample scored less than the control in the sensory analysis. The reasoning goes by spirulina can impart a distinct flavor that may align with consumers' expectation for chocolate, and also it indicates that the customers prefers taste, which is taste over the quality and health of the product. So that's it, sir. And we thank our college and our guide for this opportunity. Thank you, sir.
Okay, one question from my side. Why you have uses char for the incorporation? Already spirulina has been incorporated into chocolate into many
Yes, sir.
researches.
Sir, this trial we are starting because there is a process in which they are extracting biofuel from the spirulina, sir. So we have started this to check whether the spirulina char can be incorporated in the chocolate. Since spirulina char is a byproduct of the biofuel synthesis, sir. The spirulina char
Okay.
we here have used is edible one. This is the preliminary test for that. Next, we will be performing the biofuel synthesis. After that, we will be doing the heavy metal test and other tests to prove that the char is edible. And after that, we will be incorporating. So this is first the preliminary test to test whether the sensory attributes just complement each other.
Have you done the protein test of this?
Yes, sir.
Can you show me the results of protein?
Actually, I didn't include it in the PPT, sir, because its
Okay,
quantification is going on. We are
that
not.
should be included because how much protein is still left? Okay.
Yes, sir. Yes, sir.
Okay, next.
Thank you,
Next, Miss Snehaprabha. Miss Snehaprabha, can you please join?
Yes, ma'am.
Okay,
Ma'am,
please share your screen.
Yeah,
excuse
sure.
me, ma'am.
Yes.
It was iron next in the list.
Yeah, Iron is next in the list because there is some issue with Sneha Prabha. She will not be able to join after this. That is
Okay,
the
ma'am.
reason, Iron. So please bear with us.
Okay.
Okay.
Ma'am, is my screen is visible?
Yeah. Please make it full screen.
Yeah.
Prabha, please open your presentation.
Yes, sir. Actually, I'm trying to make a full screen, sir. But
Okay, go ahead with this. No problem then. Let's try once. Like in the sense you unshare and share again. And if that doesn't work, then we'll go with the shorter one. So go once, try once quickly, unshare and share again.
is it visible now?
It is visible, but it is very small.
Very small.
Yeah, it's not full screen.
It's still not open, I think.
Can you just unshare and share again?
Okay.
Ma'am, can I present after this? Ankur Agarwal, ma'am.
Ankur, yours is scheduled after.
Is it visible now?
Yeah, yours is scheduled later on. Yeah, 1 second, 1 second. Yeah, it is visible now.
Okay. Okay, good evening, everyone. I'm Snehaprabha Tomar. I'm PhD scholar in MIT University itself under the guidance of Vijay Chaudhary. Today, I'm going to present my work on fabrication of carrier-mediated nanoparticles for efficient drug delivery system. Drug delivery system for cancer therapy.
Please go ahead. change your slide and go ahead.
Yeah. So, my work is focusing on the nanoparticles. I'm using polymeric nanoparticles, and polymeric nanoparticle has emerged as the most promising carrier. They are very good. They are good in using for the drugs which are poorly water soluble, and they are biodegradable polymeric nanoparticles. So they they don't create any kind of harm to the body and to the environment. PLGA, PGA is the most common polymeric nanoparticles which are being used for this work. They are biodegradable polymeric nanoparticles, as they are being used because to enhance the bioavailability, biodistribution of the drug in the drug delivery system. They are more target specific, and they minimizes the non-specific toxicity, and also they enhance the effectiveness of the drug at the target site. The objective of my study is to fabricate a biodegradable target-specific nanoparticle drug delivery system for the treatment of the cancer to improve the biodistribution, safety, and efficacy of the encapsulated agent in it. And it is target-specific. For that, we are using the folic receptors also with it, folic ligands with it. The research methodologies are involving the fabrication of my complex, then the characterization using following tests for that. These are the results of my work. First of all, there is a fabrication of PLGA polymer that included these 4 steps. First of all, there was the activation of PLGA using bisamine, then conjugating it with PEG, then conjugating this activated formulation with the folic acid, and then finally creating my complex that, which is PPF, that is PLGA, PLGA, PEG, and folic acid complex. This is the chemical reaction which takes place during the synthesis process. In this, we can see that activation is the PLGA is getting activated using bisamine, then it is getting conjugated with PEG, and finally conjugating with folic acid and creating a complex. Then for the first characterization which we have done was percentage yield calculation. This was the formula for calculating the percentage yield of my complex. The result was 72.73% for PLGA PEG complex, and 75.91% for the whole complex, which was PLGA, PEG, and folic acid. This was the FTIR graph for my complex, in which it has confirmed the conjugation and successful formulation of my complex. Then that was the previous one was this is the graph for the formulation of PLGA and PEG only, and then this was the graph for the PLGA, PEG, and the folic acid for the whole complex. Then there was NMR study also to confirm my formation. Both first it was for the PLGA, PEG formulation, then it was for the PLGA, PEG, and folic acid formulation. Then there was a quantitative study for the determination of the amount of the folic acid in my complex. The conclusion of my study was that my complex was successfully synthesized using this synthesis process, and the result was acceptable and were successfully optimized. Thank you. Hello.
Yes, Prabha. Why did you incorporate folic acid? What was the reason for adding folic acid with PG and PLGA?
Folic acid is being used because to enhance the target specificity of my formulation, because folic acid's receptors are present at the tumeric sites to avoid any kind of non-specific toxicity. I'm using folic acid with my formulation.
So basically, this formulation, how are you going to use it in tumor cells?
I will use this formulation as a carrier with my drug. Then my drug would be resveratrol. So I'll be encapsulating my drug into this, and then it will be used in the drug delivery system to deliver that drug specifically to the tumor sites only.
So basically, this is a drug delivery system.
Yes, ma'am.
So the whole complex, what is the size and the morphology of this particle which you have made, the nanoparticle?
I have done the morphology part also. But for that the results are I have not put those results here actually, but I have done that study. But I have to do more studies on the size and morphology study. I have to do more about it actually.
Okay, so whatever information you have collected, what is the shape of the molecule which you got, the nanoparticle?
It is spherical shape actually.
Spherical shape. Okay, thank you, Snehaprabha. So Irene, please join. Thank you. Thank you, Snehaprabha.
Yes, ma'am.
Okay, ma'am.
Thank you. Snehaprabha, please unshare so that Irene can share, okay?
Yes, ma'am.
Okay, okay then, thank you.
I hope my screen is visible.
Yes, visible.
Okay.
Okay, go ahead. Yeah, go ahead, please.
So, good evening, all. My name is Aaron Vargas. Today, I'm here to present my review paper topic, that is plant-based meat alternatives, innovations, challenges, and future prospects. Coming to introduction. So, basically, what is plant-based meat analogs? They are usually meat alternatives or meat substitutes that are made from vegan or vegetarian ingredients and is eaten as a replacement of meat. They usually look like meat, taste like meat, and feel like meat products. Some of the examples include seitan, falafel, tempeh, etc. The first plant-based meat analog was introduced in late 19th centuries was the tofu. Now the question comes, why plant-based meat analog? First of all, less animal cruelty. Over 200 million animals has been slaughtered each day for the human consumption. So this helps to reduce the animal cruelty. Also, it is opportunity for the vegans to consume it. It's a matter of sustainability and is a healthy alternative of meat with less fat and more vitamins and fiber. Now, the usual composition of the plant proteins that are used are from wheat, rye, barley, we get gluten, from soybean, we get beta-conglycan, etc. Now, coming into the composition, the main ingredients used are water, texture vegetable proteins, non-textured vegetable proteins, flavor and taste enhancers, fat or oil, binding agents, coloring agents. Each of these ingredients provide different functions. Coloring agents like heme and keratin provides a red color of meat. Binding agents helps to hold all the ingredients together, and fat or oil helps to provide tenderness and juiciness to the product. Flavor enhancers produces meaty flavor, and the textured vegetable proteins, they also helps to hold the ingredients together and give the proper texture to the meat products. Now, coming into the various methods that are used to develop plant-based meat analogs, the first one is top-down method. The specialty of top-down method is that here the existing meat is deconstructed and it is completely understood, and its composition is studied, and we are recreating it to a plant version. That is, we are starting with the finished products here and working backward. Under this comes extrusion, mixing of hydrocolloids, free structuring, and shear cell technology. Now, coming to the bottom-up method, we here you're starting with the individual ingredient and building up into a final product. That is, you're starting from the smallest of the basic animal and gradually assembling them to finished meat. Under this come microprotein, which is highly digestible and bioavailable. It also produces a meaty flavor. And spinning, which under that comes a wet spinning and electrospinning. Now, coming to the advantages and disadvantages, the major advantages are this plant-based meat and oils are having low saturated fats with less cholesterol, and also they contain fiber, antioxidants, vitamins, and minerals. And also they provide wide variety of flavor and texture and produce less greenhouse gases. Now, coming to the disadvantages, most of them would be processed, so they would contain additives and preservatives that are not sometimes healthy, and also most of them are rich in sodium and are expensive than the usual meat. Also, not all the plant-based meat mimic the taste and texture of common meat to everyone's liking. Now, main challenges
Can you
faced.
go to, can you go to conclusion? We are
Yes, sir.
already aware about this.
Iron, is this a review work? You haven't done any results because you haven't done any experimental work?
No, this is my review paper presentation actually.
Review paper. Okay, okay. Because we were expecting results and discussion. Research papers were oral ones, and the poster ones we were expecting the review. And there are even for the poster we were having a lot of research articles. So since it is a review, so okay, so you give your conclusions. Okay.
Okay, ma'am. Okay, thank you. So coming into the conclusion, we know that the demand for plant-based substitutes are being increasing, not only for the vegans, but also for the healthy and environmental reasons. But there are certain challenges to bring the proper texture and appearance, consumer safety, etc. And other challenges include the consumer acceptance. Like most of the people are not aware of it and are not ready to accept it. So proper consumer education has to be given. Also, technologies has to be developed to improve the texture quality of this product, and it has to be marketed well for the people to understand it's important and accept the product. So that's all from my presentation. Thank you.
Oh, okay.
Okay.
Okay, Irene. Thank you for the presentation.
Thank you, Irene. So
Okay,
the
ma'am.
next person is, thank you, Irene. Next person is Anuradha. In between, we were having a request from Ankur. Ankur, are you online?
Yes, ma'am. Yes, ma'am.
What is your problem, Ankur?
Ma'am, actually, I have to go somewhere else. So, can I present?
Somewhere else.
Ma'am, actually,
See, Ankur, your presentation is scheduled after 3 students. Anuradha Barda, Abhimanyu Sharma, Ashok Kumar Yadav, because Dr. Deepti Singh is not presenting, so you have 3 people ahead of you. So right now, Anuradha Barda, Abhimanyu Sharma, Ashok Kumar Yadav. These 3 of you, if you have no issues, then shall we allow Ankur?
मैम एक्चुअली मेरा एनिमल ट्रायल्स चल रहा है मैम, तो मुझे जाना पड़ेगा एनिमल ट्रायल के लिए, तो मैम वो एनिमल
So that's what I was telling. So we are requesting the permission of Anuradha Barda, Abhimanyu, Ashok Kumar Yadav. Are they around? If it is okay, we allow Ankur.
We don't have any issue.
Okay,
Can
okay,
present
Ankur, then go
with
ahead. Ankur, please go ahead then.
Good evening to all of you. Today I am present my topic. This is optimization of this fermentation process, or identify this metabolite profile by throwing HRMS of this.
Ankur, can you please make it full screen?
Right. So first we
And
discuss this black
from where you have joined?
Just
which place you have joined? Which university? BHU. Okay, okay, fine. Please go ahead.
Going. Ma'am, now ma'am, full screen are you? Hello.
Yeah, yeah.
Hello.
Yeah, yeah. We can see your
now,
paper.
yes. Yes. So
Okay,
now
please go ahead.
yes. Now it is what is a black. You can see in this table that what is the difference between conventional wheat and black wheat, and there is a high content in anthocyanin, phenolic content, and some minerals, sodium, iron, and zinc. And this is developed by this NAPI Institute in Wuhan. And this underutilized black wheat can be used for this food product such as idli. We know that idli is made from this rice and black gram. This is a composition in right side, and in one of the data says that the demand of this idli batter is very popular. There is a one company brand ID batter which produce 65,000 kg of idli batter per day. So demand of this idli batter is good. In India, demand of idli batter is very high. So on this, we can use the blackwood, underutilize this blackwood for preparation of this rava, or we optimize through resource surface methodology and artificial neural network, and this is our objective. First objective that we develop this rava idli by this method. In this fermentation is optimized by through response surface methodology and artificial neural network. This is a response that on the basis of batter volume, pH, titrable acidity, and density. This is a flow diagram, and we get a resource surface methodology. There is a 13 type of trial we get by through RSM. There is a temperature 36 degree, and fermentation time is 2, 4, 6 hours, and the response is batter volume, pH, and titrable acidity and density. And we get temperature 36 at this batter volume at 36 degree for 6 hours, and the batter volume is very high and desirability also high. And this is a 3D image that the pH decreases and titrable acidity decreases with the over time, and due to the mainly organic acid production, and idli batter density also decreases with the higher temperature, mainly due to the incorporation of this bacteria. And we use this RSM data for 2018, and we get regression. We get a regression 0.98, and this is a diagram of that regression analysis and performance of ANN used in this work. And this is a comparison of this ANN or RSM. We have seen that ANN having closest to experimental value, ANN having high batter volume, which is closest to experimental value, titrable acidity and density, and R square is very high as compared to RSM. In RSM, there is a prediction value is 0.94, and in ANN 0.98. We use this RSM data in ANN, and there is a comparison by through R square and mean square value, and we done an experimental value which is closest, that the ANN value is closest to experimental. This is a pictorial image that what we have done, that blackwood we have taken, and we done this blackwood inoculated with this 1% of lactobacillus menstruate at this 36 degree for 4 hours, and followed by tray drying and packaging, and reconstituted with the mixing with and this we get the idli, and in left side there is a control, and in this right side optimized. And now at high protein, because of blackwood having high protein dietary fiber also, we get iron content is 23.8 mg per liter as compared to control. So we get the DPPH also is high as compared to control. This is a SAM image of control and final product. In final product, we have seen that the starch granules in blackwood lost structural integrity, mainly due to the high dietary fiber, which restrict the water binding with the protein. And in left side, there is a control, and we see that there is swelling and starch granule with slight floating texture analysis of this both control and optimize, we get the hardness. We get in optimize, the hardness is very high, mainly due to the insufficient breakdown of this starch
Ankur,
protein
please
matrix,
hurry up.
and we get a high adhesiveness. Yes, sir.
Yes, your time is running low. Okay, you have already.
Yes, sir. Yes, sir.
Okay.
There is the last slide. Yes, we get less adhesiveness due to the poor gelatinization. And this is the last slide. We get this HRMS by through high resolution mass spectrophotometry, and this is the component we get as compared to control. We see that amino acid, especially essential and non-essential amino acid, we get and fatty acid and bioactive as compared to control. We done control also, so we get this amino acid and
Okay, Ankur.
as spectrophotometer on the basis of the yes, sir. We conclude that the ANN is optimized for fermentation process, and high resolution mass spectrophotometry identified these 34 metabolites, and this black wheat rava idli can be used for the woman which is deficient for this anemic woman. Thank you.
Okay, Ankur, only one question. Why you have used ENO in that?
सर जो ट्रेडिशनल मेथड है वो उसमें इनो यूज करते हैं सर, हमने बिल्कुल ट्रेडिशनल मेथड यूज किया.
Traditional method.
यस सर, जो ट्रेडिशनल मेथड है, जैसे बेकिंग सोडा यूज करते हैं, तो बेकिंग सोडा में वो मतलब यहाँ का बेकिंग सोडा अच्छा नहीं है, तो इनो ही हम प्रेफर करते हैं. वो और फ्लफीनेस के लिए भी हम सर काफी करते हैं, क्योंकि वो फ्लफी आ जाए थोड़ा.
बट आपने कंपैरिजन में राइस इडली में तो इनो ऐड किया नहीं है, राइस में तो विदाउट उसके है.
सर हमने
So how can you...
हमने सर यस सर, हमने एक्चुअली व्हीट लिया है सर, हमने राइस और ब्लैक ग्राम नहीं लिया सर. हमने व्हीट से सूजी बनाई है और सूजी की रवा इडली बनाई है.
अच्छा, रवा इडली के साथ आपने कंपेयर किया.
यस सर.
ओके, ठीक है. नेक्स्ट डॉक्टर अशोक कुमार यादव सर.
Good evening, everyone. Am I clearly audible to everyone?
Yes, yes, sir.
Okay, thank you for clarifying. Just I'm sharing my screen. Is this visible?
Yes, sir. Yes, it's visible.
Please make it full screen, sir. Yeah, thank you.
Okay, and good evening. My greetings to everyone. Thank you for giving me this opportunity to share some of my work that is going on in my lab at this international platform. So first of all, I would like to introduce myself. I'm Dr. Ashok Yadav, currently working as assistant professor in the Department of Food Technology at Rajiv Gandhi Central University, Itanagar. So this is a part of my work. This is a part of a work that has been performed by one of my students, which I want to share with you all. In the northeastern part of our country, in the Himalayan part of our country, there is a plant known as this one Hutenia cordata, which is commonly known as fish mint here in local area, and it has a lot of medicinal properties, and we are working on the extraction of those main components from this plant, and after that we have planned to develop some kind of functional food from that particular extract. So this particular plant, this plant, how it looks like, it is not very much known, and it is not very much explored plant, and it is not known in those where we generally live in the northern part of our country. But here in the northeast and the Himalayan region, this plant are very much popularized, and people are consuming it in their culinary and used it. Some traditional people are using it as a medicinal importance also. For it is a rich during our research we have found that and from our some of the literature review we have found that it is rich in bioactive components and various health benefits. The reason for selecting this plant for our research is since it is very less explored plant, and it have a lot of bioactive components which are very much less explored, and very much least least research have been performed on this plant. That's why we have selected this plant, and this particular work has been classified into three main objectives. We have selected first of all, our objective was to select a proper suitable solvent through which we could get a best solvent extraction method. Then we have just some of the tests through which we can explore that the bioactive components are best, in which sample we are getting the best bioactive components, and then we have studied the antibacterial effect of this plant extract. This is the complete, we have gone through the extraction process through the help of sonication and obviously through continuous moving shaker. And finally, after centrifugation, we have got the extract in different four different solvents, which is methanol, ethanol, acetone, and water. Then we have gone through some antioxidant activities like the PPH inhibition, antioxidant inhibition, total phenolic content through standard protocols that were earlier identified by researchers. Total phenolic content and antibacterial activity. And this is the figure that demonstrates one of my student is working on that plant extract. This is the result what we have obtained during our research. We have found that the ethanol extract has been found best extract, you can say that solvent in means of all those parameters which we have taken for our study. Either those are yield or the pH or in ABTS or even antibacterial activity. Also, we have found that the ethanol extraction and during the ethanol, whenever we have used ethanol as a solvent medium, we have got the best result in this one, and these results are very much aligned with the previous studies. And what we conclude that the reason for the best yield there, because ethanol have the ability to dissolve all polar and non-polar compounds. That's why it has given the best yield best yield, which we have got 0.46 milligram per 10 gram of the sample. In the same way, DPPH inhibition was also found with this ethanol extract, and the same reason was that this already supported by some of the researcher, and what we have concluded that is the water and methanol have low antioxidant activity because some of the components were not dissolved, could not be able to dissolve in water and methanol components. So that's why our result could not, or the DPPH inhibition or ABTS inhibition could not be up to that mark in water and methanol extract. And ethanol, and this ethanol have the ability to dissolve a wider range of phenolic components and bioactive components into it. That's why we could be able to get those DPPH and ABTS inhibition up to a means we can say that up to a greater extent, so that this 82% inhibition, which is very much high. In the same way, we have got for this total phenolic content also in the ethanol extract, we have got a better, in ethanol extract, we have got a better result for DPPH also, which is near this 210 microgram of gallic acid equivalent, which is also supported by the previous researchers. They have also found that during the ethanol effect of the plant, the total phenolic content, DPPH inhibition were of greater extent. When we found that when we done through this bacterial count, we have found that this in ethanolic extract, the bacterial count in during 5th and 6th evolution, the bacterial count was very much less, and we can conclude from this count that this particular plant have some antibacterial effect as well. So we can conclude that the plant have a lot of bioactive components which can further be used for the development of some kind of functional food. And obviously this after a proper study, some future research is required so that to extract a particular bioactive components, and that can be transferred to pharmaceutical industry for the development of some kind of active drug for particular use. So this is our future research, and I don't want to miss to acknowledge one of my master student who have worked on this, and still some of the work is going on this particular plant. So this is all about my work.
Thank you, sir.
Thanks from my side.
Sir, can I ask you a couple of questions?
Sure, ma'am. Sure.
Sir, first thing which I would like to know is whether you have done the phytochemical analysis of the extracts which you have made, which you have done with the plant.
Yes, ma'am.
Phytochemical analysis of all the type of extracts. If I remember correctly, you had water extracts, ethanol extracts.
In all the 4 extracts, we have gone through this test.
You have done only the antioxidant activity test and phenol content test, or any other phytochemical analysis?
No,
you
ma'am.
had done?
Here here I have shown only this test, but we have gone through this phytochemical test also.
Okay, so when you check the phytochemical analysis, again, there also the same result showed up that ethanol contained the maximum components.
But in some of the tests, we have got the better result with methanol also. But the reason for selecting the ethanol extract as the optimized, because ethanol extract could be used for further when if we have planned for developing some kind of functional food, then ethanol extract is more desirable, or we can say that ethanol extract is more suitable, so that we can incorporate that ethanol extract. But in some of the result, we have got good result for methanol also.
Okay, so
But
for
that methanol
organics,
extract could not be incorporated, ma'am.
yeah, that is okay, sir. So just wanted to know about the acetone extract as well, because organic extract you had done only with ethanol, acetone, if I remember correctly, right?
Yeah, yes, ma'am. Yes, ma'am. Yes.
Yes, and the rest it is like more of a polar nature, the solvents which you had used, water, methanol, and
Yes,
ethanol.
ma'am. The rest of those are polar
Okay,
in
have you
nature.
tried chloroform as well, or is it only the acetone
No,
extract you had done?
we have not gone for chloroform.
Okay, and the last question which I had is the antimicrobial activity you had mentioned, right? So
Yes, ma'am.
is it coming from the ethanolic extract, or any activity you got from the water extract?
I could not understand your question. Means the inhibition of those growth of microorganism.
Microorganisms. Yes, yes.
Yeah.
Was it only
It-
coming from the ethanol extract, or was it coming from all the
No, the
extracts which you had
inhibition was
prepared?
observed in all kind of extracts, but the minimum inhibition, the maximum inhibition was obtained in ethanolic extract.
Ethanolic extract.
But
Okay. And one
one
last
thing,
question.
yeah,
Yes,
please,
sir.
please.
Yes, sir. Please go ahead.
But one thing I want to clear that because during the research or during the question, it may arise that ethanol has its own antibacterial properties, though the suppression may be due to the ethanol. But that is that we evaporated all the ethanol and we have used the extract only, and we have
Yeah,
culture from
you
that
must have
extract only. So we can
yeah, you must have dissolved it back in DMSO, right? When you did the antimicrobial
yes, ma'am,
activity.
absolutely, yeah,
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 that's
that is absolutely fine. So I just wanted to know the comparative effects of the different extracts which you had prepared. And the last question I have is, sir, what about the preparation of the extract? Did you do any kind of warm extracts, or is it the normal temperature extraction you had done?
Ma'am, we have just gone through this sonication process. During sonication, there are chances for the maximum extraction of the plant components. So we have gone through this sonication only.
So the extract was prepared using these solvents during the and along with sonication.
After crushing this plant leaf and comparative study of leaves and stem also, we have mixed with this solvents, and after that we have gone through the sonication. We have kept that sample for 24 hours in sonication, and after
In
that we have gone
sonication.
through centrifugation.
Okay, so during the sonication process, did you do it in ice jacket or in some kind of cold chamber you did the extraction?
No,
It
ma'am,
means in
we
the
have
sense
not used
sonication.
that. We have not used in the cold chamber. It was at normal room temperature. Since our
No.
sonicator was at incubation, we have maintained the temperature at room temperature 35, 38 degrees Celsius. We have maintained.
Okay, thank you so much, sir. Thank you. Thank you for answering our questions also. Thank you for your time.
Oh,
Thank you for your time, sir.
thank you very much. Thank you for
Thank you.
giving me this
Thank you, sir.
opportunity also. Thank you.
Thank you, sir. Thank you. So the next person is Anuradha Barda. Anuradha Barda, are you there? Abhimanyu Sharma, are you there? Abhimanyu Sharma? Ankur is done. Then Manisha? Manisha?
No.
Okay. Then Manisha, not there. Kaushik? Kaushik, are you there?
Kaushik Jaina.
Kaushik Jena?
Yes, ma'am. I'm here.
Okay, okay. Kaushik, then you can start your presentation.
Yeah.
Is the screen visible?
Yes, go ahead, Kaushik.
Okay. Good evening, everyone, and good evening, all the chairpersons. Actually, I am going to present the topic energy exchange analysis and microwave drying of little millets, and the fourth international convocation on green environment technology introduction through innovation, organized by Amity University, Jaipur. And my authors are Prabhu Bairakanu sir and Dr. Siddharth Sinha sir, Department of Chemical Engineering and School of Agro and Rural Technology, IIT Guwahati. And these are my content introduction followed by methods, drying time, energy efficiency, specific energy loss, exergy efficiency, conclusion and references. Introduction: generally, drying refers to the removal of moisture from a substance. The drying mechanism include combined and heat and mass transfer and changes the properties of the product. And efficient drying process has the three following parameters. One is quality of the product, that is retaining all the nutrients, the natural appearance are retained, and initial investment and running cost, and maximizing the energy efficiency. In conventional heating, mass transfer takes place from the center to the outer periphery, and heat transfer takes place from the outer periphery to the center. But in microwave, heat transfer takes place from the center to the outer periphery, and mass transfer takes place from the center to the outer periphery. And introduction: millets are the small fielded grain and the Poaceae family. Millets are rich in very nutrients like dietary fiber, bioactive compounds, protein and resistant starch and vitamins. That's why millets are called smart agriculture product. And little millet, one of the important minor millets, and oval to elliptical in shape. Actually, little millets are prone to insect after harvesting. That's why we are planning to do the sprouting, and then after the sprouting, millets should be dried in the microwave oven. And consumption of little millets helps in controlling the cholesterol and like cardiovascular disease. And though millets are a gluten free, though protecting celiac diseases and also some cardiovascular diseases, millets are protecting. And these are my experimental setup: the soaking of millets, and then after soaking for the 16 hours in the little millets in room temperature, I sprouted the little millets in the incubator, maintaining the humidity at 85% and room temperature. This is the sprouted millet. Then the sprouted millet are dried in the microwave oven by controlling the power, but my highest temperature was set at 50, so that the product should not deteriorate with respect to the power and time. So, and this is the drying time, and I dried the three microwave power. One is 450 and another one 750 and 1050 watt. And the drying time was highest in 1500 seconds for the 450 and the lowest was 240 seconds for the 1050 watt. And the specific energy consumption, I found that at 450 it was 14.573 megajoule per kg, and the lowest was 1050 watt. At the higher microwave power, the more amount of water is evaporated, which leads to the specific energy consumption is less. And energy efficiency of the energy efficiency, how much fraction of heat used for removing water to the amount of heat supplied, that is the energy efficiency. And energy efficiency we got highest for the 1050 watt, that is 49.24%, and lowest was 15.51%. And the specific energy loss, during the drying was calculated. One is 1 minus drying efficiency, and it was calculated. The specific energy loss was lowest in 1050 watts and the highest one in 12.313 watt megajoule per kg. And exergy efficiency defined as the amount of work a system can perform when brought into thermodynamic equilibrium with the environment. So exergy efficiency can be calculated as exergy of evaporation in power input, and exergy of evaporation is calculated by 1 minus temperature of the ambient temperature to the product temperature, and it was found to be highest was 5.31, though we are maintaining at the highest temperature of 50. So the exergy efficiency got some low percentage, 5.31. And these are the conclusion. Drying time was decreased by 2.5 times in each interval from 450 to 750, and then 1050 watt, and the specific energy consumption is decreased by 38.03% and 68.5% at a power level of 750 and 1050 watt compared to 450 watt. The energy efficiency was found to be 49.24 at 1050 watt. With increase in power level, the specific energy loss decreased, and the exergy efficiency was found to 1.2 to 5.31 at this microwave power level. And these are my reference. And thank you.
Okay, Kaushik.
Yes, sir.
Okay. Why you have just used a microwave only? You can use some other drying techniques also.
Actually, conventional drying techniques, the drying time was from 8 hours to up to 16 hours. So the drying
Okay.
time was higher. So in microwave, the drying time was reduced much lesser time. So that's why we have chosen the microwave. And one parameters also we have chosen. We have given the power input, and the higher temperature we also fixed so that the product should not get deteriorated.
But when you microwave it, you are already cooking the product.
Yes, ma'am.
So then have you checked the nutritional difference between the one which is sun dried and the one which is microwave dried?
Yes, actually, we have done the nutritional values, but I have not presented the nutritional quantities.
Actually, you should have done a comparison.
Okay.
The energy composition of the microwave is okay, but you should have also compared it with tray drying or any other conventional drying methods. What is the difference between the nutritional aspect? Then only you can say that microwave drying was better at drying.
Okay, sir.
Okay.
Yes, sir.
Okay, next.
Okay, so thank you, sir.
Next.
Asmita, are you there? Dhruv, are you there? Nikki Bhandari? Nikki Bhandari? Nikki Bhandari, are you there? Nikki Bhandari? She was online a little bit ago, while ago. Maybe she got disconnected somehow. Okay, then Krishna Kumari? Krishna Kumari?
Yes, ma'am.
Okay, so please go ahead, Krishna Kumari.
Yeah, PPT is visible. Start
Okay.
presenting.
Okay.
Make it full screen though.
Make it full screen.
Is it?
Yes, yes. Go ahead, please.
Okay. Good evening, everyone. Myself, Krishna Kumari. I am a research scholar in the field of biotechnology at Banasthali University. I am here to present my work on the topic of efficient degradation and detoxification of reactive violet 5R by newly isolated bacterial strain from dye-contaminated site. Let's begin with the introduction. There is a rapid increase in industrialization and urbanization, which causes increased wastewater production. Wastewater generated only from textile industries is about 17 to 20% of total industrial waste. Several kinds of synthetic dyes and pigments are extensively used in the industries due to their vibrant colors and resistance to fading, but their non-biodegradable nature makes them persistent environmental pollutant. So the treatment of these toxic dyes are necessary for the environmental concern. There are some traditional methods were used to treat them. These methods have highly operational cost due to need for the advanced technologies, chemicals, and energy consumptions involving chemicals that also that also further contribute to the pollution. In contrast to the bioremediation, which is a low operational cost as it is primarily relies on the natural growth and activity of the microorganism, also offering a sustainable and natural solution. There are different microorganisms we can use. Here we choose in our studies bacteria, which is considered as a most effective agent due to faster growth rate. Also, their metabolic diversity makes them capable to degrade a wide range of synthetic dyes. These are the following objectives. First one is isolation of efficient bacterial strain from dye-contaminated site, followed by the optimization of potential strain to degrade reactive violet dye by using response surface methodology. Additionally, the study was conduct a phytotoxicity assessment to evaluate the toxicity of degraded product. The soil sample was collected from Bagru, Rajasthan, and potential isolate was identified by using 16S rRNA gene sequencing, and it was identified as Bacillus paranthracis, which is not explored very much till yet. This bar graph compares the decolorization efficiency of different bacterial strains against this dye over various time period from 24 hour to 5th day. Notably, the B61 strain, which is identified as Bacillus paranthracis, it shows highest decolorization efficiency, reaching up to 95.33% by the 5th day, while other strains exhibited significantly lower performance. Usually, the optimization of environmental factors was done by using one factor approach at a time method. But here we had used RSM for the optimization. RSM can estimate linear and quadratic effect of the factors and predict a model for the response. Here, 4 different factors were used, named as pH, temperature, dye concentration, and the inoculum size. In this study, total 27 experiments were generated by this model, and a second-order quadratic polynomial relationship between all variables were obtained. The experimental results were fitted with the coded equation. The optimal model showed high coefficient of determination, which was 0.9612. And this is the first figure is perturbation plot. The curved lines indicate interaction between the factors. The steepness of these lines indicate the relative importance of each factors. Also suggests that which factor has more significant impact on the response. As we can see that the factor A, C, and the D has more impact rather than factor B. Next is actual versus predicted graph. In this graph, these cluster points closely around this diagonal line, it indicates a good fit between the model and the actual data. Here, the actual data is nearly clustered closely to this diagonal line, which proves that our model was fit. These are the 3D visualization of above 4 parameters interact to the influence the response with each parameter. Each plot represents a pair of parameters. The color gradient and the surface shape illustrate that how change in one parameter affect the response while the other is held constant. The analysis of these plots identified the optimal conditions to achieve highest decolorization, which was 98.91% at pH 6.9, temperature 36 degrees Celsius, dye concentration was 180 mg, and inoculum size was 4%. This slide represents the phytotoxicity effect of the dye, and which was done by 2 plant species. One is Vigna radiata and other is Brassica junecia. Both species showed a decrease in germination, plumule length, and the radical length, when exposed to untreated dye compared to the control. This one. And the treatment with Bacillus paranthracis significantly reduced the toxicity of the dye, leading to a recovery in germination and growth parameters in both the species. These findings suggest that this strain could be a potential bioremediation agent for contaminated sites of textile dyes. In this present work, the azodye-degrading bacterium was isolated from textile dye-contaminated site and identified as Bacillus paranthracis by using 16S rRNA gene sequencing. The bacterium decolorized 98.91% after optimize the conditions, and the toxicity assessment results showed that the toxicity of treated dye solutions was significantly reduced, and the germination seeds were indicating that the isolated strain is potential to decolorize and detoxify the reactive violet 5R dye for environmental safety. I would like to acknowledge Amity University for giving me this opportunity to present here, and the HOD sir of our department, Professor Deepjyoti Chakravarti, and Dr. Sharad Vats, my supervisor, and my lab mates for their support and expertise in the research. Thank you. Ma'am, I'm open for the question and answer round.
Okay, Krishna Kumari. So my first question to you is, how did you isolate the strain?
Ma'am,
What
I
was the
have
method you
isolated by
had?
using serial dilution method.
Serial dilution method.
Yes, ma'am.
Okay, so when you did the serial dilution, what was the actual dilution factor which gave you the isolation of the strain?
Ma'am, -3.
10 to the power of -3.
Yes, ma'am.
You had collected the soil from the dye-contaminated site, right?
Yes, ma'am.
Okay, did you see any difference in the amount of microflora which are present in the soil isolated from the dye-contaminated site and the soil which is a normally soil which do not have any dye contamination from the same area? From the same area, did you see any difference?
No, ma'am, I haven't did this from I haven't isolated from same area, but I have just trial once to see the difference between both. So yes, the microflora is less over there as compared to the normal soil sample, as I have been isolated from Banasthali.
Okay, so you said, if I remember correctly, you had isolated it from Bagru.
Yes, ma'am.
Okay, so you had collected the soil sample from Bagru, right?
Yes, ma'am. From Chippa Mohalla of Bagru.
Chippa Mohalla, Bagru.
Yes,
are you aware of any technique in which we can actually isolate microorganisms, specifically the one you are looking for?
Ma'am, enrichment method?
Okay, you did this. Yes, enrichment technique. So if you were supposed to do enrichment technique,
yes,
what must have been the technique which you have optimized in isolating the microorganism which you got after serial dilution, which you are doing, which you had presented?
Ma'am, I didn't get your question. Which technique I have
Question is,
optimized?
how will you, how your enrichment media will look like when you want to isolate a strain of this much importance, as you were telling that you did it by serial dilution, right?
Yes, ma'am.
So serial dilution, you must have come across a lot of strains, isn't it?
Yes, ma'am. Ma'am,
Yes, a lot of, yes.
Ma'am,
Then how did you?
हमने ऐसा करा था कि सीरियल डाइल्यूशन करा था, देन एक एक स्ट्रेन को टेस्ट करा था डिफरेंट डिफरेंट डाइज पे. जो स्ट्रेन ज्यादा डाइज को डिग्रेड कर रहा था उनको फिर हमने चूज करा था. एनरिचमेंट मेथड में आप पहले ही, क्योंकि आप डाई स्पेसिफिक एनरिचमेंट मेथड यूज करोगे.
बिल्कुल, बिल्कुल. Because you had mentioned the name of one dye, and that is the reason I asked you this question.
मैंने और भी बहुत सारे ये प्रेजेंट यहाँ पे बस एक डाई का किया है, बाकी तो और टोटल 7 डाइज पे काम कर रहा है.
Okay, okay. Then it answers the question, because I was curious why you went for serial dilution when you can go for enrichment technique rather than, spending so much of time analyzing
क्योंकि
each of
पोटेंशियल
the
माइक्रो
microorganism which you get.
छोड़ना नहीं चाहते थे, तो बस इसी वजह से
कैसा?
मैम, we don't want to left any potential microbe which
Okay,
can
leave any
destroy all the
protection.
dyes. So we firstly we isolated, then we done the screening by using dye concentration of 50 mg per liter, and then we had done other experiments.
Okay, are you a PhD scholar?
Yes, ma'am.
You're a PhD scholar. Okay. So this is part of your PhD work, not the entire work which you have presented.
Yes, ma'am.
Okay, okay then. Thank you. Thank you, Krishna Kumari. The next person can present now. Next person in line is Tanishq Khandelwal.
Yes, ma'am. Yes, ma'am.
Nikki Bhandari, are you there? Nikki Bhandari? Okay. Anyway, Tanishq, you can go ahead with your presentation.
Hello, ma'am. I am audible.
Yeah, you are audible. Please share your PPT. And Krishna Kumari, please unshare so that Tanishq can share his PPT. Krishna Kumari, please unshare your PPT so that Tanishq can share his PPT.
Yes, ma'am. I'm looking for the option.
The same option which you had used for sharing. That is the one.
Okay.
The share button is the same button for unsharing. Or if you log out, then also it will be unshared.
Okay, ma'am.
Yeah, Krishna Kumari, it is unshared now. So Tanesh, please share your screen.
Hello.
My screen is.
Hello, yes. Yes, Tanesh, please go ahead.
Okay.
Please make it full screen. Go ahead.
So, hello everyone. Good evening, ma'am.
Good evening.
So my topic is
Please
on
go
green.
ahead.
So my topic is on green synthesis. Green synthesis will nanoparticles from Tedescantia pallida leaf extract, characterization and mechanism of antimicrobial action. So going ahead, an introduction. So what are nanoparticles? Nanoparticles are materials that have at least one dimension between 1 and 100 nanometers. Among various nanoparticles, silver nanoparticles are absorbing due to the wide range of applications, especially in biomedical catalysis and environmental fields. They are well known for their excellent.
Tanishq.
Yes, ma'am.
Tanishq, we had shared the format for PPT. One. Second, you haven't mentioned from where you are, which university, which place you are from.
Okay. So, ma'am, I am from MIT University, Rajasthan, Jaipur, and I am currently pursuing my master's in applied chemistry.
Okay, so who is your guide?
So, ma'am, the guide is Dr. Manmohan Singh Chauhan.
Okay, and this is a research article, research paper you're presenting?
Abstract.
Abstract? No, we are not asking any abstract presentation.
Ma'am, research paper.
Research one? Research paper.
Yes, ma'am.
Okay. Okay, then. So,
yes, ma'am.
Go ahead.
Yes, ma'am. So, talking about green synthesis, the green synthesis of nanoparticles is an eco-friendly approach that uses biological agents, such as plant extract, microorganism, or enzymes for the reduction of metal ions into nanoparticles. It eliminates the needs for harmful chemicals and toxic reagents. Moving ahead to why we choose Tedescantia pallida. So, its common name is purple heart. It is rich in bioactive compounds such as flavonoids, polyphenols, etc. It is eco-friendly in nature. Biocompatible, talking about biocompatibility and availability of plant extract for green synthesis. So,
Tanesh,
if we talk
Tanesh,
about,
excuse me, Tanesh, your
yes,
slide
ma'am.
is not moving and it is not on full screen mode. Tanesh, your
Okay,
slide is not moving. It is still at the first slide.
okay. Wait.
Yeah.
And now is it visible? Hello.
Slide is
It is visible.
changing, but it is still, yeah, it is visible, but it is not on full screen mode. So please share it in full screen mode. Yeah. So now you go ahead.
Now?
Yeah, it is fine. Go ahead now.
Okay. So talking about introduction, nanoparticles are materials that have at least one dimension between 1 and 100 nanometers. Among various nanoparticles, silver nanoparticles are absorbing due to their wide range of applications, especially in biomedical, catalysis, and environmental fields. They are well known for their excellent antimicrobial properties, making them valuable in medical devices. When talking about green synthesis, the green synthesis of nanoparticles is an eco-friendly approach that uses biological agents such as plant extract, microorganisms, or enzymes. Ma'am, I use plant extract to check antimicrobial activity for silver nanoparticles. So my objective is to synthesize silver nanoparticles using Tradescantia pallida leaf extract and explore their antimicrobial activity. So, ma'am, my slide is moving. My
Go
slide is
ahead.
moving.
Yeah, it is moving. Go ahead.
Okay. So why I choose Tradescantia pallida? So its common name is purple heart. It is rich in bioactive compounds such as flavonoids, polyphenols. Eco-friendly choice, biocompatibility and availability of plant extract for green synthesis. So talking about my synthesis of silver nanoparticles, firstly I prepared leaf extract of Tradescantia pallida. Then it
See,
mixed with, yes, ma'am.
excuse me, Tanishk, can
Yes,
you,
ma'am.
because this is the normal way of nanoparticle synthesis. So can you show us the results, what you have got?
Ma'am, I don't have image, but I put an image in background of this. Ma'am, this is ma'am, I don't have results. I make a short PPT on this. I don't have results right now.
See, you had mentioned that you have done the nanoparticle synthesis. So this is basically silver nanoparticle synthesis you have done.
Yes, ma'am.
Is it?
Yes, ma'am.
And silver nanoparticle synthesis you have done using a plant extract. And what was that extract? Methanolic, ethanolic, which extract it was?
It is plant extract.
No, extract must have been done in a solvent, right? Either water or ethanol or
Water
extract,
Yes, ma'am.
right? And in the water
Yes, ma'am.
extracts, you made the silver nanoparticle in how much time?
Ma'am, 3 to 4 hours.
Was it it's a leaf extract that you had made?
Yes, ma'am.
Leaf extract of the plant. Okay. Okay, so you made your nanoparticle, and how did you make sure that you have made the nanoparticle?
Ma'am, by UV spectrometer. Uh, Ma'am,
Only UV
it comes.
spectrophotometer?
Yes, ma'am.
No other method?
What?
Except UV spectrophotometer, did you use any other method for checking the nanoparticle? Formation of
FTI
nanoparticle.
indications of
Yeah,
UV
XRD you have done. So UV, XRD, and then
FTIR.
FTIR. Okay. What about electron microscopy?
Ma'am, I didn't do that.
Okay. So
Yes,
what information you got from XRD?
Ma'am, it is XRD. Ma'am, it peaks has come at I don't know the nanometer of range. Ma'am, it confirms crystalline structure.
Okay, so your nanoparticle was crystalline structure.
Yes, ma'am.
Okay. And then go ahead. What you did after that?
After that,
FTIR indicated what? Because your slide is very black.
Ma'am, okay. Ma'am,
What
mainly
was
my
the
focus is on antimicrobial action of silver nanoparticles.
Okay, go ahead. Which microorganism you had used?
E. coli and Bacillus cereus.
E. coli and Bacillus cereus.
Yes, ma'am.
Any particular reason for selecting these two?
Ma'am, because E. coli is the most commonly used bacteria in antimicrobial action, and so does Bacillus cereus also. That's why.
Okay, what was the strain's name? Where did you get the strain?
Means I didn't get it.
Where did you buy this strain? You bought it from somewhere, or somebody donated the strain to you?
Yes, ma'am. Yes, ma'am. Somebody donated my strain of this, ma'am. Then I revived the bacteria again, and then I do experiment.
Okay. Okay. Go ahead.
Okay. So, ma'am, my methodology is agar distribution method.
See, methodology is fine. What was the results that you got after the antimicrobial activity checking?
Ma'am, results, ma'am. This is my result. It is visible.
Yeah, it is visible. Which organism it is, I don't know.
It is
it E. coli?
E. coli. And I didn't
what?
mention Bacillus cereus disc, but this is E. coli.
Okay, go ahead. Complete your conclusion.
So my antivector results are there, and the advantage of green synthesis silver nanoparticles is eco-friendly,
That's
biocompatible.
all fine. What is it that you got? That's it, done.
And then I performed reduction of silver nanoparticles by NADH4.
And then?
And then
should continue with your slides. How many slides you have?
Okay, ma'am. Ma'am, these are the advantages and these are the applications of silver nanoparticles: environmental water treatment, bioremediation, biomedical bound healing, drug delivery, antimicrobial coatings, industrial. These are the applications, and the conclusion, ma'am, my summary is the silver nano using traditional, eco-friendly, and cost-effective method.
Okay, my question to you is, does it have any antimicrobial activity? Yes or no?
Yes, ma'am, it has.
Okay, thank you.
Okay.
Okay, Puneet Gupta.
Yes, ma'am. Just a second.
Yes. Yeah.
Ma'am, is it visible?
No, it is not visible yet. Please share your screen.
Just a second. Is it visible now, ma'am? Hello?
Yeah, you're presenting from your phone?
Yes, ma'am.
Okay, go ahead.
Okay, so good morning, good evening. So my name is Puneet Gupta and I'm from MET. I'm currently pursuing MSc in applied chemistry, and my mentor is Dr. Manmohan Singh Chauhan. So today we'll be explore how graphene-based composite materials are revolutionizing water-based purification. So basically, starting off with the introduction. So graphene, as many of us already know about, it is a single layer carbon atom which is arranged in a hexagonal lattice. So it also has some exceptional properties like mechanical strength, electrical conductivity, and chemical stability, which gives it more fantastical material, which makes it. Yes, ma'am. Yes,
Puneet,
ma'am.
is it a review article or a research article you're
Ma'am,
presenting?
no, ma'am. It's basically like I have done the research on it. Although
Okay, if
the
you have done the
reduction,
research, then show us the results.
ma'am.
Which
So
is
the thing is, yeah. So the
graphene
thing
we
is
know
that,
already.
yeah. So the thing is that, ma'am, the last slide which I have presentation which I have made is got corrupted. So I made this another presentation in a rush. So I don't have the specific result, but I do contain some of the points. Like in the images, I can tell you about all those things.
Yeah,
Like
so
which
just
material.
in short, you tell us the story, what you have done.
Yes, ma'am. So, so basically, from the very beginning, I have taken up the material called copper for the metal, and the graphene oxide, which I made a sandwich compound of it. By firstly, I just prepared the sandwich compound through the sonigation method and stirring for 2 to 3 hours. After that, when the complete process is done for the making of the sandwich compound, then I go for the reduction process of it, just by checking it with the dyes. I used alizarin red, malachite green, and methylene blue. These were the dyes I used, and with that I checked the reduction of it, at what peak and the absorption level it was giving. So most of the time, the copper was giving something around 575 nanometers of the range, and this is how the whole research went off. So basically, my purpose for that is to make a membrane which actually stops the those of the contaminants and toxins which do not pass by through it directly through the water, or so that it can actually use for the water purification for in the future aspect, and replacing the traditional methods for water purification through the nanoparticle and the graphene compounds.
Have you done the toxicity analysis?
Ma'am, I have checked the spectroscopy, like I use the UV and the FTIR, and like electronic microscopic view. Wait a second, ma'am, just a second. There it is. So it was something related to that. I don't have the clear image. I just got it from my previous slides.
Okay, my question was, you're using it for water purification. Are you using?
Yes, ma'am.
Or is it or the polluted water purification?
So, ma'am, we can actually engineer these types of things, like these nanoparticles, specifically to the contaminant. If we are using just for the water purification and just make the salty waters for the drinkable water, we can actually use it by these methods. But if we are looking for specific toxicants like mercury presence or like toxin component present in the water, we can actually engineer it by different types of filtration methods, or by in the laboratories doing the similar techniques that we have used.
Okay, thank you. Thank you, Puneet.
Okay, ma'am.
Next.
Just a second.
Chahat.
Good evening, ma'am.
Good evening. Please share your PPT, Chahat.
Yes, ma'am. Just a minute. Good evening, ma'am. Ma'am, is my screen visible?
No, Chahat.
No, it's not visible.
Your slides are not visible.
Okay. Ma'am, now? No, this is...
It is coming, but it is not in full screen mode, and it is
Yes,
showing only half of your slide. Yeah, it's still not in the full screen mode. Can you make it full screen?
Ma'am, actually, there was some issue with my laptop. That's why I couldn't join with it. I'm using the phone.
Your phone is fine. Somebody was also already sharing with phone. That is absolutely fine if your phone's network is fine.
Try to change the slides.
A slide is moving. Okay, go ahead. Give your
Ma'am, actually, the
presentation.
images are not properly visible.
That's fine. You explain.
Okay, ma'am. Good evening, ma'am. Ma'am, my name is Chahat. I'm a student at MIT University, Rajasthan. I'm pursuing Masters in Applied Chemistry. Today, I'm going to tell you about the routine analysis of dapagliflozin metformin tablet. By the analytical methods, I'm going to ensure the precise quality and the efficacy of the tablets that I have used. Coming to the introduction, dapagliflozin metformin tablets are basically used to manage type 2 diabetes, and it is also used to reduce the risk of heart failures. It is a combination of dapagliflozin and metformin drug, which gives the benefits of both the drug together instead of different medicines that we take individually. The type of combination that we can is simply treatment regimens and improve adherence by reducing the number of pills our patients can take. And about the dapagliflozin and metformin tablet, dapagliflozin is a sodium glucose co-transport inhibitor. It reduces which are responsible for reabsorbing glucose from urine back into the bloodstream. It reduces this flow, which helps us to reduce the glucose. Dapagliflozin increases glucose excretion through urine, thereby reducing the lowering good glucose blood pressure. And coming to metformin, metformin is a primarily works on reducing glucose production in the liver and increasing the insulin sensitivity in muscle cells. Hence, both the combination of these drugs help us to reduce the blood glucose level and improve glucose taken up by the cell. Ma'am, for the efficacy that we have used here methods, there are a number of methods that we have used, with the help of HPLC, high performance liquid chromatography techniques. With the help of HPLC, we perform specific tests like dissolution test, blend uniformity test, content of uniformity test. And these are the tests that we use to for the quality checking of the medicine. We also perform tests like disintegration test, RS test, to ensure if there are any unknown impurity that can be produced in the dapagliflozin metformin tablet because of the reaction of the ingredients that are already present in that drug.
Chahat,
Yes, ma'am. Yes, ma'am.
These are the drugs which are already available in the market for
Yes,
treating diabetes, right?
Ma'am.
So
yes, ma'am.
Both drugs are being, been in the and they both have been in the market for more than many decades.
Yes, ma'am. Ma'am, actually, in the
Yes.
market,
So what is it that you are studying? Because quality control already it has been done. Otherwise, it will not come into the market, right?
Actually, in the market, we have already in several reference listed drugs. In the formulation project, we are mainly checking in a dapagliflozin metformin tablet, we have different 2.5 mg present of dapagliflozin. Dapagliflozin present can be 2.5 mg to 10 mg presence of dapagliflozin. We check the different types of formulation projects, in any particular batch there is 2.5 mg present of medicine, and in other project there is 5 mg. So we take different samples, and after different samples we analyze what are the results that we have got.
So how I just did not get what you are actually doing. The thing is, like, these two drugs are already available. Just wait. These two drugs are already available in the market. You had mentioned that you did the quality control check. That is obviously not required because otherwise it will not come to the market until and unless you are comparing the quality of different brands giving the same drug, right? That is not you are doing. Then with these two drugs, what is it that you are doing? You are making a totally different formulation. What is the objective of the work?
Ma'am, actually, they are formulation projects. The drug formulation is going on. We are basically analyzing the results that we are getting by changing the composition of the drug that is already present in that medicine. Like we are taking 2.5 mg.
So how are you going to change the composition of the drug?
Ma'am, in any sample, we are taking 2.5 mg of dapagliflozin. In other batch, we are taking 5 mg, and in other batch, we are taking 10 mg. So we are just changing the composition to analyzing the result. But the result that we vary by doing these changes.
So how are you analyzing the result? You said it is for, it is for a anti-glycemic drug, right? It is going to treat diabetes. So where are you analyzing? How are you analyzing that? Whether it is better than, better in taking which dose, and who is getting the benefit? What is it that you're analyzing?
Ma'am, the samples which are already available. We are making new samples by these methods, and by the help of chromatography techniques, we are analyzing what the results that we have got are different from the results that are already in the market.
But what result you're analyzing? That is my question.
Ma'am, like if under different condition, like after 6 months, under a particular temperature or humidity level, what are the things that are getting, there is a possibility by the reaction of the ingredients that are in the drug, there can be any unknown impurity arise at a different temperature and humidity level.
So are you checking the shelf life of the drug?
Sorry, ma'am?
Are you checking the shelf life of the drug?
Ma'am, we are doing many tests like shelf life. We are also checking. We are also checking if there is any unknown impurity on the in comparing to content test, if the medicine has already the proper amount of drug present while its formulation.
Okay, you do one thing. Show us the conclusion.
Ma'am, like this is the dissolution apparatus. Here we see how the tablet, at what time the tablet is giving how much release. Like after 2 hours, after 4 hours, how much the tablet is giving release in a particular condition.
Okay,
And
go
ma'am,
ahead.
now at the conclusion, we have got the test we are performing here. We have taken the 10 mg tablet of dapagliflozin metformin. So it has passed the dissolution test in which we observe the effective release of drug by the time. Like BU test, it is a blend uniformity test in which the blend powder before making the medicines we get and we check if the drug is properly uniformly distributed in that powder or not, or if there any lack of mixing in that. Like in CUSA test, we see after the medicine has formed, if there is a proper distribution of drug over that medicine or not, like if it is giving proper results or not. That's what we have taken this. And the 10 mg tablet has passed all the results, and we have got the conclusion here.
Yes, Chahat, thank you. The next
Yes, ma'am, thank
person,
you.
Kena. Devanshu Sharma.
Yes, ma'am. Yes, ma'am.
Puneet Gupta, you have given the presentation already, no? Yeah, Puneet has given the presentation. So Devanshu Sharma, you are the only one left now, the last person.
Yeah, ma'am.
Because I think Nikki Bandari is still not able to join. Okay, so go ahead with your presentation. Make it full screen, please.
Okay, ma'am. Yeah, ma'am, it's okay. Hello, I'm audible, ma'am?
Yes. Yes, go ahead, please.
Good evening, ma'am. My name is Devan Sharma. I am from MIT University. I'm pursuing right now MSc Applied Chemistry. And my guide is Dr. Renu Padhyay, ma'am. So today I represent my work on screening and isolation of microbes degrading benzopyrene using from lens fill site soils. Okay, so these are the topics I consider today. First, I tell you about the benzopyrene, what is benzopyrene, and why I use the biodegradation process for degrading this. And
Beta, this is done. You go for the experimental setup.
okay, ma'am. Let us talk about the experimental setup. I take the soil from landfill site. So first we take the soil and do some extraction methods like Soxhlet extraction method. From it we extract some polyaromatic hydrocarbons from soil, and we check it by GCMS that which type of polyhydrocarbons are present. Okay, this one first one this is, and for the degradation I make the minimal media of pH 8. In this minimal media I take potassium source, phosphorus source, sodium source, like that, and then autoclave it, and then add the only carbon source, which is benzoypyrene, for the degradation. and then use soil by adding water in it, and put it for uh, till the soil get settled down, and took the microbes of 100 microliters and put it in the minimal media, and then put it in shaker incubator for 37 degrees at 37 degrees Celsius on 110 RPM for several days. And then after 7 days, we check that the turbidity is come, and if the turbidity is come, means microbes are growing. And for direct means is this, the microbes using benzoypyrene as the energy source, which is the only carbon source in our minimal media. And for this, we further analyze that. We take OD of that solution also in 2 hours, which we find that microbes are growing continuously, which means our benzoypyrene is continuously degrading. And then we start a 1.5-month experiment for it. In which we take the sample in every 7, 15, 30, and 1.5 months. And by taking these samples, we do GCMS. And by the GCMS, we found that different molecular weights which come in the degradation part of benzoypyrene. And if we talk about the degradation of benzoypyrene in the several steps, which first is oxidization by microbial enzyme, which are our microbes. We do that because they use benzoypyrene as the energy source. First they have to degrade it in different parts, like bond cleavage, bond breaking in the ring system, like that. Where further yes,
Devanshu, your work is more of microbiology than chemistry. You are doing chemistry, right?
Yeah, ma'am.
And what is the method, the name of the method which you have done, just explained? Can you tell me that?
Yeah, ma'am. We use which, ma'am? I don't get it. Method.
Have you actually got the microorganism, and have you saw the degradation? What is the method for the name of that method?
Ma'am, when we generally use we normally use it by GCMS. We did GCMS, that we found that first we did GCMS of the native compound and get the different molecular weight on different peaks, that retention time. And after that we did the degraded solution analyze, then it will give different peaks and molecular weights. That how we get that our benzoypyrene is degrading.
So how harmful benzoypyrene is for the nature?
Yes, ma'am. Benzopyrene is a, ma'am, carcinogenic product, which means it is a cancer producing compound. And if
where is it present, in the sense, what is the reason for finding benzoypyrene in nature?
मैम different by vehicle emissions हो गए, और कोल तार हो गए, और ये जो हमारा इंडस्ट्रियल पॉल्यूशन हो गया मैम, इस वजह से भी बहुत सारे जो हमारे पॉलीएरोमेटिक हाइड्रोकार्बन्स है वो आते हैं, जैसे कि एंथ्रासीन, फ्रेंथ्रीन हो गया, हमारा फ्लोरीन हो गया.
This is this present in the atmosphere as in the gaseous form, or it is going into the soil? That was my question.
Ma'am, it went to the soil.
Okay, thank you.
Okay, ma'am. Thank you, ma'am.
IT support, are you still available? MET IT support, are you still available, sir? Hello.
Yes, ma'am. Hello.
Sir, sir, there is one student, Nikki Bandari. She's not able to join. I don't know what is the reason.
Ma'am,
She had joined in the beginning, but then she's not able to join again.
ma'am, there might be network issue from their end.
Okay, so we'll try one more time. We'll ask her to join, and then if she is not able to join, then we'll close the session after this. Okay, sir?
Okay, ma'am.
Okay, and sorry for extending the session. We had to finish the presentations because we had to give the results tomorrow.
No issue, ma'am. No issue.
Okay, and your name, good name?
Mobin.
Mobin. Okay, okay. I got your number from Pashori sir yesterday. Okay, so we'll just give her a call and we'll let you know in a few minutes. Okay?
Hello.
Okay, ma'am.
Okay, sir. Thank you.
Hello.
Sir, Nikki is trying to join. So we have asked her to join again. So please let her in if she joins. Okay, sir? Nikki Bhandari. Hello, IT support. Hello. Hello, Mobin sir. IT support.
Yes, ma'am.
Sir, that student is actually trying to log in. She's telling she's in the waiting room. Can you please allow her?
Give me a second, ma'am.
Yes, sir.
Yes, ma'am. She's joining.
She's joining. Okay, okay then. Thank you. Yes, Nikki, please give your presentation fast. Okay. Share your screen.
Nikki Bhandari.
Nikki Bhandari, please share your screen. Yeah. Okay, make it full screen and then go ahead.
My screen is visible?
Nikki, your screen is visible. Please go ahead.
Good evening, everyone. My name is Nikki Bhandari, under the supervision of Dr. Sharad Bars from the Vanasari Vidya Peeth. I'm going to present my research work on the topic of the phytochemical investigation of the plant parts of Tecomella undulata, endangered desert teak. First introduction, Tecomella undulata belongs to the Bignoniaceae family and commonly known as Rohira, desert teak, and honey tree. It is endangered deciduous tree found in the different parts of India, including Rajasthan, Gujarat, Punjab, Haryana, and Maharashtra. It is used to treat various diseases like abdominal pain, cough, liver and spleen, and many more. And root paste is given internally in Leucorium. It is primarily valued for the timber. Various parts of the plant, including flower, leaf, has been explored for the potential antioxidant properties. Second objective. My first objective is to identify various bioactive compounds from the from different plant parts of Tecomella undulata to evaluate, and second one is to evaluate the phytochemical antioxidant potential of the Tecomella undulata. Procurement, the plant parts of Tecomella undulata will be collected from the campus of Vanasthali Vidyapeeth, and different plant parts, leaf and flower will be dried in the 40 degrees Celsius oven and fine powder for the analysis of bioactive compounds. In this slide, this study utilized reverse phase HPLC to identify various phenolic compounds. There are the different standards of phenolic compound, and it is retention times. It is a chromatogram A and B. A is flower and B is leaf. Among the investigation, significantly, maximum 6 compounds, phenolic compounds were identified in the flower, and 5 phenolic compounds are found in leaf. In flower, aspartame, syringic, naringin, cinnamic, and vanillic and ferulic acid were found. In leaves, cinnamic, benzoic acid, and salicylic and cinnapic and ferulic acid were found in leaf. These compounds have been shown in antimicrobial, antioxidant, anti-cancerous property. Antioxidants. Antioxidant is the molecule capable of inhibiting the oxidation of the other molecules. Oxidation is the chemical reaction that transfers electrons or hydrogen from the substance to an oxidizing agent. Oxidation reaction can produce free radicals. In turn, these radicals start chain reaction cause various damages in the body as shown in figure. Determination of the total phenolic content and total flavonoid content. Total phenolic content measure the amount of phenolic compounds which are the antioxidant that helps protect against cell damage and have potential to health benefits. In this slide, you can see in the leaf have higher content of total phenolic content present than the flower. And total flavonoid content measures the amount of flavonoids, a class of the plant compounds with the anti-inflammatory, anti-allergic, and anti-cancerous property. And in total flavonoid content in flower have the higher content of the flavonoid than the leaf. Antioxidant assay of Tecomella undulata, DPPH. Measure the antioxidant potential by scanning free radical decolorization of the DPPH solution expressed as a IC50, including the amount of antioxidant required to neutralize half of the free radicals. DPPH, in the DPPH, leaf have the higher activity than the flower, and NOSA measure the antioxidant inhibiting the nitric oxide generate the sodium nitrochloride expressed as the percentage inhibition of nitric oxide indicating the antioxidant capacity. In also NOSA has the higher antioxidant activity than the flower in leaf. Thank you.
Nikki, basically, what was the importance of the study?
Basically, it is an endangered plant, firstly Tecomela, and it is not explored that much, and it is the highly medicinal importance of this plant. That's why we explored that these phenolic compounds are not reported yet. I am the first who report these compounds.
So basically, what is the activity of these ones? You have done any antimicrobial activity? Or it is only antioxidant?
Antimicrobial activity also, like I'm not showing the result of this antimicrobial result in this PPT.
Okay, you are a PhD scholar?
Yes, I am PhD scholar.
Okay, then Nikki. So basically, you have shown only the antioxidant properties of the phenolic compounds which you have isolated,
Yes,
right?
yes.
Okay. Okay, then. Thank you, Nikki.
Thank you, ma'am.
Thank you. Sir, IT support team. Sir. Hello. Hello. Mobin sir. Hello.
Hello. Yes, ma'am.
Yes. Sir, I have one question. The thing is it the recording is being done for the session?
Yes, ma'am. Yes.
Okay. So will you be publishing it anywhere? Because the reason why I'm asking you is this is an oral presentation session. So many of the results which the students may be showing, because there were many PhD scholars, they may not have been published. So we don't want this session to be published anywhere.
Ma'am, right now we don't publish any event.
Okay. So that means this is going to be very private for us, right?
Yes, ma'am.
This should not come in public domain. That is the main thing, because many of them know their PhD will be under trouble if you publish it anywhere. So all the oral presentation sessions, because yesterday Pachori sir told me again, yesterday session also you were handling from Noida, right?
Ma'am, we record all the events for our records only.
Okay, so we are not submitting anywhere. That is fine.
Yes.
Okay, sir. Then thank you. Because public domain, if it comes, then we will be in trouble. That is the only issue, because oral presentations we are not supposed to publish anywhere. Okay, sir.
Okay.
Okay, sir. Thank you so much. Thank you, sir. And sorry to keep you so late.
It's okay, ma'am.
Okay, then. Thank you. Good night, sir.